inauguration ceremony and also the organizing chairman. Respected Dr. S.K. Sarin, MD, DM, FAMS, President of the National Academy of Medical, has an online guest of honor. Dr. Naveen Thakkar, Honorary Secretary, Indian Orthopedic Association. Dr. Rakesh Gupta, MSc, our guest of honor and President, Indian Medical Association, Raipur. Professor Dr. Ali Mohapatra, Dean Exams and HOD Biochemistry. Professor Dr. Amit Chauhan, Co-Organizing Secretary and HOD Pathology. Dr. Harshal Sakle, Co-Organizing Secretary and Consultant Orthopedics at AIIMS Raipur. A Herculean task was given to us by our director, sir, 3 years back, when he wanted us in 3 years. The first of its kind on stem cells was organized 3 years back. Then last year on artificial intelligence, and last of this series was on role of nanotechnology in healthcare, which we are conducting today. As nanotechnology is not a basic part of medical, getting faculty for the same was really difficult amongst the medical fraternity. But then we got wonderful faculty, and today I know after attending the session, you all will be well trained into nanotechnology. You all must be knowing that Richard Feynman was the first to conceptualize the potential of nanotechnology nearly 6 decades ago. In 1959, he described it as a field in which little has been done, but in which there is an enormous amount which can be done in principles. Nanotechnology was originally defined by the National Nanotechnology Initiative as a study and controlled manipulation of individual atoms of size between 1 and 100 nanometers, which have therapeutic applications at the subcellular level. Fundamentally, at the nanometer scale, nano-engineered materials have the theoretical ability to target and modify these processes. At nanoscopic size, they have unique and advantageous surface characters compared to the original size. High surface area to volume ratio is basically responsible for the infinite property achieved by their smaller size. Mechanical, optical, magnetic, and chemical characters of nanoparticles is achieved due to its particle size, which enables them as ideal choice for wide application in the field of science, electronics, defense, cosmetics, and medicine. Respected Director, sir, I welcome you as well as thank you for providing us this platform where we can all learn about nanotechnology. I'm thankful to Professor Dr. Rakesh Gupta, sir, President, Indian Medical Association, who is here with us and who has taken great pains that the whole city today knows about this nanotechnology CME, and every press and newspaper fellow has contacted us. I welcome all the registered delegates, which amount to more 5 on the Google Forms, and many more coming from the National Academy of Medical Sciences, Indian Orthopedic Association, Indian Medical Association, AIIMS Raipur Alumni Group, and students and SRs who will be attending this function, not only physically here, but also on the and YouTube link, which will be available, if not possible today on a working day, then throughout the year, whenever they find time. So thank you, friends. Let us start with the CME.
Thank you, sir. I like to request Professor Dr. Ali Mohapatra, ma'am, Dean Examination and Head of the Department Biochemistry, to tell a few words.
Good morning, all. Our respected Director, sir, Nitin Ma'am Nagarkar, and our CEO and Director. Then the President of IMA, Rakesh Gupta, Dr. Rakesh Gupta, our Dean, Academic Professor Alok, and all the dignitaries on the dais and off the dais, and dear students, a very good morning and welcome you all to this symposium on nanotechnology. Well, I'll elaborately a little bit about nanotechnology, so I won't be repeating the same things. But just to know, nanotechnology, it started in India recently in 2001 only. It was initiated by, he is being told, the father of nanotechnology in India, Professor CNR Rao. And now we have seen the invasion of nanotechnology in different fields, right? It be agriculture, medicine, and everywhere. It has been used very effectively in imaging, diagnosis, as well as treatment. The target focusing treatment where you, in case of tumors, cancer, and any other complicated diseases. So this type of CMA or symposium will be a eye-opener for all of us. And we can do our research further. We can carry on our research work on nanotechnology, and with the advancement of which we can give more and more benefit to the society. Thank you all for giving me this opportunity. I thank you all.
I like to request Dr. Rakesh Gupta sir, President, Indian Medical Association, Raipur, say a few words.
Our director, sir, Dr. Nitin Nagarkar, Dr. Ali Mahapatra, Dr. Alok Agrawal, and my dear friends. It has been a very pleasant journey throughout since last 7 years with AIIMS. We have been part of the journey of AIIMS since inception, when the almost foundation stone laid by Dr. Nitin Nagarkar. We have seen the growth of AIIMS to a magnificent institution. Being the ENT surgeon, we have been very close with Dr. Nagarkar, and faculty of ENT and all the faculties have grown to leaps and bounds only due to presence of AIIMS here in Chhattisgarh. This word nanotechnology is new to my generation because we are growing with the medical profession. But due to AIIMS, we have been matching with the technology and the newer aspect of medicine step by step. I feel very honored and blessed to be part of the institution, being friend and co-faculty of Dr. Nagarkar. I think all the medical professionals, paramedics will be benefited by the presence of AIIMS in Chhattisgarh and Central India, by the technology, education, and the technical know-how in coming years. I personally congratulate Dr. Nagarkar having laid the foundation of cooperation and blessing with the knowledge in the Chhattisgarh and medical fraternity. I hope that all these traditions are being continued with coming years. I am grateful for inviting me and hope to be part of the institution in coming years. Thank you so much.
Thank you, sir. Is Professor Dr. S.K. Sareen and Dr. Naveen Thakkar with us online? I think so, it's not connected. Okay. I'd like to request our chief guest, Professor Dr. Nitin M. Nagarkar, sir, for the message.
Good morning. Thank you very much and welcome to everybody for this National CME on the role of nanotechnology in healthcare. Professor S.K. Sareen, who has joined us online, Dr. Naveen Thakkar of the Indian Orthopedic Association. My colleagues, Dr. Rakesh Gupta is the president of AIIMS Raipur and my very dear friend and ENT colleague. Professor Alok Chandra Agarwal, Professor Ali Mahapatra, Dr. Amit Chauhan, Dr. Harshal Sakle, all faculty members from various departments. We got invited guests who have come from other institutions. And we have our support staff, nursing staff, and of course, my dear students. Thank you very much, first of all, to Professor Alok Agarwal, that it was our dream that we should always talk of topics which are contemporary or which are going to become important in the coming decades. Of course, we do discuss about the common topics as well, whether it is tuberculosis, leprosy, cancers, what we grapple with. But there are always new technologies, new sciences coming up, and I think AIIMS has a role to play in that. We need to become abreast with whatever is happening in the world, what is new, what is exciting, as well as what would be the challenges posed by these. So I thank Professor Alok Agarwal and our entire team, along with support from the National Academy of Medical Specialties, IMA of the state, as well as various other organizations, your Indian Orthopedic Association, other organizations, association, CSIR, they have also been, like you have the IHBT again partnering today, IIT, and other scientific organizations, the central universities, for example, the Guru Ghashidas University. So I think everybody has willingly participated when we came up with these topics. Now, topics, whether we are discussing, we had decided to start off with these three, and we have done two of them already. Today will be the third one. We first did on stem cell therapy, again, a very important one, a very healthy discussion. It was again conducted by the hybrid mode. And many participants, including from abroad, were so willingly participating in it. Then the second one was on artificial intelligence and machine learning. We had a lot of support Daxa from IIIT. Professor Sinha, P.K. Sinha, the director, he was also giving his address, and a lot of good discussion on that. And I think these are all technologies of the future. We need to understand what these technologies, how they're going to change our lives, and especially as medical professionals, how it is going to touch us. So the third in the series was, of course, on nanotechnology. And again, thank my team and National Academy of Medical Specialties and other organizations who have teamed up with us. It's nice to see participation from different parts of the country. A lot of participation from IHBT Palampur, which is under CSIR. We're going to listen to Dr. Amitabh Acharya. But we've got other institutions of national importance, including AIIMS, the central universities, IITs. So all, I think, would share their expertise along with our faculty members and faculty members from other medical institutions in the city and state. So we know that nanotechnology is the technology which has already started making a difference. It is the technology of the future. And like Dr. Ali Mahapatra rightly mentioned, whether it is in the field of agriculture, whether in the field of environment and technology, and of course in medical sciences. So that is somewhere where it's going to be very, very important in coming times. And these nanoparticles could become, we have again a lecture by Dr. Manjush, who's going to speak about drug deliveries, I'm sure, a very, very important aspect about how nanoparticles will be, reaching your targeted site and targeted deliveries, how we can reduce the dose and therefore reduce the toxicity, because the damage to the other healthy tissue would possibly be reduced. Similarly, how to do imaging with this, because the tissues would light up better when we are doing whether an MRI or whichever radiological investigation. Similarly, creating other types of sensors. So whether it is treatment of cancers, whether it's treatment of infections, we also had an interesting topic recently discussed with the IIT about nanorobots. And I'm sure that that would also today help in diagnosing infections or targeted delivery toward treatment of cancers. Well, at the same time, whenever you have these potential benefits, there are certain challenges, the toxicities, because some of them are metals, heavy metals, so how they can damage either the liver or the kidneys, or how they can be, excreted or removed. So different types of issues would be there, I'm sure, but we will discuss on those. And across the day, I've seen topics from various specialties being discussed from various institutions. So it all goes to show that whenever you have a newer technology, we all need to come together. Whether it is the research organizations, whether it's the technology institutes, whether it's institutes conducting basic science research, and of course, clinical work being done like in our hospital or institute. So we need to team up together, and so many minds would be, and I'm happy to see that the students who are actually going to be facing this kind of changing evolution or technology and how they would be adapting to the new technology. So their minds would actually be ignited and they would be learning so much more from it. So welcome to again, everybody who are present with us physically as well as online. And I'm sure that we'll have a very healthy discussion and we can come out with a compendium of the ideas finally, which we can circulate to all the concerned. So thank you very much. Jai Hind, Jai Chhattisgarh.
Thank you, sir. I'd like to request Professor Alok Agrawal, sir, to present a memento to Professor Nitin Nagarkar, sir. Thank you, sir. I'd like to request Professor Amit Chauhan, sir, to give a memento to Dr. Rakesh Gupta, sir. Dr. Harshal Sakle, sir, to present a memento to Singh, sir. He's a secretary Raipur branch. Thank you, sir. I'd like to request Dr. Harshal Sakle, sir, to give a final thanksgiving.
So very good morning to all of you. So on the behalf of Medical Education Unit, AIIMS Raipur, Department of Orthopedics, AIIMS Raipur, IMA and Indian Stem Cell Study Group. First of all, it's my proud privilege to deliver the vote of thanks and extend our heartfelt gratitude to respected Dr. Professor Dr. Nitin M. Nagarkar, sir, Director and CEO, AIIMS Raipur, our organizing chairman and chief guest of today's function. Sir, sir always inspires us, encourages us for all of such kind of academic activities. Thank you, sir, for your gracious presence and blessings. I'm thankful to Professor Dr. S.K. Sareen, President NAMS, I'm thankful to Professor Dr. N.K. Thakkar sir, Honorary Secretary, Indian Orthopedic Association. I thanks Professor Dr. Alok C. Agrawal sir, Head of the Department, Department of Orthopedics, AIIMS Raipur, and Dean Academics, AIIMS Raipur, who keep all of us motivating, guiding us from time to time, and who is being instrumental in organizing this kind of event. I take this opportunity to thank Professor Dr. Rakesh Gupta sir and Dr. Digvijay sir, IMA and Secretary and President of IMA Chhattisgarh Group. Thank you, sir, for blessing us. I am thankful to Dr. Eli Mohapatra Madam, Dean Examination and Professor Head of the Department, Department of Biochemistry. Thank you, Madam, for graciously blessing the event. I am thankful to Dr. Amit Chauhan sir, Head of the Department, Department of Pathology, our co-organizing secretary, for being instrumental and working hard from last many days for making this event a success. I am thankful to Professor Dr. N.K. Agrawal sir, Dr. N.K. Bodhe sir, and Dr. Vinay Pandit sir, Dr. Virat Golotar sir for gracing the occasion. I am thankful to all my SRs, JRs, Dr. Ankit, Dr. Shubham, Dr. Rudra, all of us for making this event possible. At the end, it's my apologies if I have missed anyone's name. Thank you very much for your cooperation and presence. I am thankful to all participants. Almost we have a registration of 250 participants which are present online and some are physically present. I am thankful to that. I am thankful to all faculties who are present here or they will be presenting their work and their knowledge online. Thank you very much. Thanks a lot. Now we'll start with our first session. I request the chairperson of first session, Dr. Alok C. Agrawal sir, Dr. Vinay Pandit sir, and Professor Dr. N.K. Agrawal sir to come on the dais to start the first session.
We have our first lecture from Dr. Amitabh Acharya sir. He is a principal scientist and associate professor at Nanobiology Lab, Biotechnology Division, CSIR, IHBT Palampur. He has more than 12 years of research experience and he has published numerous in national, international paper publication, books and projects under his name. We welcome you, sir, today.
Yeah, thank you very much. Am I audible?
Yes, sir.
Okay, thank you very much. Just let me know if my slides are visible.
Slides are not visible, sir. Please share the screen. Yes, sir, they are visible now.
Okay, hope my slides are visible and they are moving forward and backward.
Yes, sir, slides are visible.
Thank you very much. Thank you very much. Good morning to all of you. Good morning to the respected director, sir, and other respected dignitaries who are present on the dais. To all the members who were present in the auditorium and the members who have joined through online mode. It is indeed my proud privilege to be a part of this particular initiative. And I'm here very much happy to share my lab's journey at CSR IHBT regarding the work that we have carried out as a small team of nanotechnologists. So the title of my presentation today will be the search of functional nanomaterials and how we have moved forward over a period of time. And I'm indeed very fortunate to get this opportunity from the organizing committee members for giving us the opportunity, giving me the opportunity to present my work. Before I start the presentation, I would just like to share a few things because this is, the most of the what exactly do we mean by nanotechnology? And what you can see is that the nanometer, which is close to 10 to the minus 9 meter, is actually right there where you have this scale bar. You can see this 10 to this nanometer range, which is 10 to the minus 9 meter, is where we are talking about. Now, this is significantly small compared to the bulk material which we day-to-day encounter with, and also significantly high with respect to the size when we are talking about the angstrom range, in which we have all the molecules or atoms in that range. Now, why the synergy between the nanotechnology and the biotechnology happened? Because a lot of biomolecules, they do fall in this category. And that is why, like DNA or protein molecules, they are in the range of nanometer. So the synergy between the nanotechnology and the biotechnology, as well as with respect to any other stream that you can think of, is very easy because the size ranges matches very perfectly. And that is the reason why long back, as has already been mentioned by Sir in the while he was giving the talk, long back in 1959, Professor Richard Feynman has actually proposed this concept for the first time, where in Caltech, in a presentation December 29th, 1959, Professor Richard Feynman has proposed that the principle of physics, as far as I can see, do not speak against the possibility of maneuvering things atom by atom. Put the atoms down there and the chemist, and so they will make the substance. Professor Richard Feynman is a very well-known physicist and also got his Nobel Prize. But when he made this comment, that time this concept was very new, that arranging atoms one by one was absolutely impossible to think. So the very next day, the New York State magazine has come out with a kind of comment. You must be joking, Mr. Feynman. But this over a period of time, it progressed and people understood the concept of nanotechnology. Nanotechnology now has been accepted by different organizations. EU organization has their own definition of nanotechnology, but the National Nanotechnology Initiative, which has also been shared by Sir, is that any material which is having a particular dimension in between 1 to 100 nanometer in one of the dimension, X, Y, or Z direction, will be called as nanomaterial. And it can actually be used by science people, engineering people, technology people, and it is basically an interdisciplinary area. It has been accepted that initially started from physics, moved to the chemistry. Physics and chemistry actually has gone hand in hand. And once they have done it, the manipulation of the matters has been done with respect to the technology that whatever the technology we would like to carry on. So what exactly is nanotechnology? Nanotechnology deals with the creation of useful nanomaterials, devices, systems using the particles of nanometer length, scale, and this one. Exploration of noble nanoparticles, which basically is at the length of nanometer. Sorry. At that length. Here, just I would like to brief you about why this nanotechnology, the concept of nanotechnology is so important. It's nothing but simply because of a very basic principle that through nanomaterials, you are actually increasing the surface area to volume ratio. So you can see here, we have a cube, which is a 4 centimeter length. And if that cube is having surface area of 96 centimeters square, now the same cube has been cut into some small pieces. And the total surface area has now increased up to 192 centimeters square. So in this process, what we have done is that we have kept the total volume of the cube constant, but we have increased the surface area by a significant amount. Now, why increasing the surface area will have any impact on the material behavior? Simply because when you increase the surface area, in a process, you also increase the surface electrons. And these surface electrons are some materials, some elements which which are going to have the maximum kind of reactivity. So since you have the same volume, but the surface area has increased, now this particular cube, though it is having the similar volume, has having more electron-rich surface area. And that is why they are more reactive. And that is why all over the world, people are now more interested about nanotechnology, nanomaterial. Now, initially it was mostly with respect to chemistry and material science. And last, I will say 15 to 18 years, it has been significantly increased in the field of biotechnology, healthcare science, and all these aspects. Just to give you an example, in the left side, you can see the first transistor that is being built by Microsoft back in 1947. And in the right-hand side, you can see the same Intel 32 nanometer NAND chip, which contains much better efficacy, containing 1.9 billion transistors in this chip. And all these 1.9 billion chips, which are being used as Intel 32 nanometer NAND chip, are actually made of nanomaterial. So the power of nanomaterial, we can see that in even our smartphone, the kind of semiconductor chips that we are using have significantly improved. So the power of nanotechnology has grown over a period of time, and it is only going to increase. But we have to remember, before using any new technology, technology is definitely the first content that we need to have, but also we need to have the safety concern. So that part possibly will be covered in the latter part of the symposia. Quickly, I will now just browse through what kind of activities we are involved with in our lab at CSIR ISBT. We are basically a group of close to 10 people right now, 8 to 9 people right now. And we are mostly involved in development of anti-amyloidogenic and fibril destabilization nanomaterials, and then mechanism studies to combat neurodegeneration. We also work in the area of antibacterial and antibiofilm nanomaterials, and we do the mechanism studies to combat antibiotic resistance. We use our nanomaterials for nanogymes, which can be used as a point of care diagnostic for responsive biomedical applications, biosensing, and immunoassays. We try to look into how nanomaterials behave under in vivo conditions and their implications in determining the fate of the nanomedicines. And also, we are into the area where we look into some of the very therapeutically important molecules can be encapsulated and can be used as a targeted delivery for different diseases. The source that we have with respect to nanomaterials are plants. I mean, this is though a very unconventional source, but we use that plants as our nano factories, because we believe that these plants can be used as a factory material where we can be used the nanomaterials. Now, what we do is, over a period of time, novel biocompatible nanomaterials that can be isolated from bioresource for improved biomedical applications. Now, plants can be a good source for different other applications. Plants can be used for isolation of different phytomolecules, but plants can also be a very good factory for different nanomaterials like nanotubes, under which single world carbon nanotubes, multi-world carbon nanotubes, or lignin nanotubes come. Nanofibers like nanocellulose, nanomaterials can come. Nanoparticles, nanosilica, a large amount of plants, they accumulate huge amount of silica inside them. Those can be isolated and can be used for different applications. And you can see that this area has been grown over a period of time with a CAGR of 17.09% for nanotubes, or nanofibers like 19.5%, graphene and graphene oxides, like they are 36.7%. All these nanoparticles from the source of plants have been actually used for different other applications. I will basically go through what, I will briefly mention about the kind of work that we are rightly involved with. So basically, as I have previously mentioned, that our group is involved in developing anti-amyloidogenic material. So just to give you a brief, because most probably all of you must be aware that there's a lot of neurodegenerative disease, there is an amyloid cascade hypothesis, where over a period of time, we have amyloid deposition in cells, which finally leads to the cell death. So our group major focus was how we can inhibit this amyloid formation. And if at all we can do so, by what mechanism or nanomaterials are doing it. So we use different strategies, out of which I have here mentioned about 3 strategies, where in the first phase, we have used one organic inorganic hybrid nanocomposite and used them as an inhibitor for the amyloids which are being produced by a particular protein, model protein, which is human serum albumin. In the second part, what we have done is we have used cellulose as one of the source for delivering our gold nanoparticle to the target sites. And we tried to look into whether our gold nanoparticles can inhibit the initial amyloid formation, which is like the oligomer formation of these amyloids at a very early stage, and can actually lead to demolition of all this amyloidogenic behavior. In the third part, we have encapsulated the cellulose nano onions with a phytosteroid called diazoxanide, which has been known to be an anti-inflammatory. For some other diseases, we use that strategy here. We successfully delivered diazoxanide to the target sites and found that diazoxanide can actually reduce the inflammation. In the process, it can inhibit the protein aggregation in both the early and late aggregates, and can reduce the amyloid concentration in the intercellular condition. I'll just give you a few of the examples. These are the basic strategies by which we have synthesized the nanoparticles. I mean, this is like the different plant source from where we generally isolate that cellulose nanocrystal. Then we use some chemical template to form these spherical nanoparticles, which are hydrophobic in nature. In this process, we conjugate the gold nanoparticle onto this one, and then encapsulate the diazoxanide onto this one, and which will look like more or less like this way under transmission electron microscope. As you can see, there is a layer by layer structure formation with these cellulose nano onions, and they have size of 60 to 150 nanometer based on what kind of cellulose that you are using. And you have embedded diazoxanide inside this one, which can be monitored using different spectroscopy and microscope techniques. You can see that the cellulose nano onions, these are nano onions because they have layer by layer structures. That is what we call them. They have interlayer structure of 3.6 to 2.7 to 3.6 nanometer. And when we attach them with gold nanoparticles, they form these kind of spherical particles. We monitored their inhibition potential using A-beta 42 aggregation. A-beta 42 is a hallmark amyloid, which is responsible for Alzheimer's disease. We saw whether under normal condition, this is how A-beta forms the amyloids. This is what the deposition we have. And when we treated our nanoparticles with these A-beta amyloids, you can see that they do have very disintegrated fibrils, which were not present. Even that can be seen in the fluorescence microscope images also. We carried out n number of studies just to confirm that how our nanoparticles inhibit the A-beta 42 aggregation. And we came out with a kind of solution where we found that this A-beta 42 aggregation, which is also being reported in literature, has also a very strong effect on inorganic metal ion dyshomeostasis. Our nanoparticle was able to maintain the ion homeostasis like sodium, potassium, and calcium ion homeostasis. And also they can inhibit the reactive oxygen species inside intercellular cell. Generally, when protein aggregates, it creates huge amount of intercellular oxidative stress. That can also be reduced. And finally, we came to the solution that more in-depth analysis are required for neurodegenerative disease, but this cellulose nano onions based gold nanoparticles can be a good solution because they can cross the blood-brain barrier through some receptor mediated endocytosis process and can also be used for targeted delivery. I will just give you a few other glimpses like the kind of work that we are presently being involved in our lab. One of that work involves how we can combat bacterial infection and whether we can have any strategy to find out that the bacterial infection can be monitored in the very early stage. So this has been our long interest over a period of time. We mostly try to look into some point of care diagnostics. Here I have briefly mentioned about the 3 approaches that we generally use. And one of them, most of the nanoparticles, because what has happened over a period of time is that antibiotic resistance, you all know, has become a major problem. And this has happened mostly because the bacteria has evolved over a period of time. We need new materials or new drug candidates which can actually follow some other mechanism compared to in which bacteria has developed their resistance. So what we found over a period of time that small nanoparticles such as carbon dots can be a very good solution to this. We have also used inorganic nanoparticles like gold nanoparticles or copper nanoparticles for the same. As you can see in this case, the first approach was development of some carbon dot nanoparticle, which can finally kill the antibacterial, and looking into those nanoparticles through transmission electron microscope, what is the mechanism by which they adsorb on the cell surface. In the second one, it was mostly like, kind of detection of a biomarker, which is pyocyanin, selectively produced and specifically produced by Pseudomonas aeruginosa while it grows. And Pseudomonas aeruginosa has been, responsible for nosocomial infection. How can we detect the Pseudomonas aeruginosa infection at the very first place? So that was one of the target, whether we can do any kind of, point of care diagnostics making or not. In the third one, again, we have find out how copper nanoclusters can be used, which is loaded on graphene oxide nanosids, and they can be used for the killing of bacterial infection, and also like how the lipid bilayer interacts with the copper nanoclusters and what are the mechanisms. So I'll just give you a brief about how we have synthesized these nanoparticles. The carbon dots were being synthesized from a very natural source, which is like orange peel. And then there are some chemical methods that we have used. We functionalized that with some kind of functional groups, which finally leads to positively charged nanoparticles. And then we have attached gold nanoparticle to that one. Now, here I would like to draw your attention to this particular moiety, which is pyocyanin. Pyocyanin is a kind of phenazine molecule, which is being selectively produced by Pseudomonas aeruginosa, just as a defense mechanism. It has also been used by Pseudomonas aeruginosa for quorum sensing, that is for bacteria to bacteria communication. And also because Pseudomonas does not allow because it's a very dominant strain and it does not allow other strains to grow. So pyocyanin to some extent is very toxic, and it actually acts as a killing agent for the rest of the bacteria. So while we were going through this one, we thought, why this metabolite cannot be detected? Because this is being produced at the very early phase. And if this can be detected by Pseudomonas aeruginosa, which is responsible for large number of infections in our body, possibly we can detect the bacterial infection in the very early stage. These are some basic characterization techniques that we use, by which we synthesize our nanoparticle and characterize through transmission electron microscope, just to find out what are the sizes, because the nanoparticle is all about determining the particle size and shape over and again. As you can see that in this case, the pyocyanin has been isolated, and it has from the Pseudomonas aeruginosa strain, and it has been treated with sequential titration of our gold nanoparticle. You can see the gold nanoparticle is basically of this pink color, and the pyocyanin is basically a blue kind of dye. But when we add our nanoparticle into this one, there is an immediate color change to purplish. So that actually proves that the pyocyanin detection has been done. Now, this pyocyanin detection has been checked through different other mechanisms like FTIR, NMR, MALDI studies. And also fluorometrically, we have monitored this by looking into them under UV radiation, where you can see increased amount of fluorescence intensity from the pyocyanin based samples. So what we have come out is that if this is the mechanism in which our nanoparticle detects pyocyanin, possibly the nanoparticle should also have some role in their killing behavior. So as you know, that pyocyanin forms a very strong biofilm in the area where it actually grows. So what we have to try to look into, whether our nanoparticle can also break the biofilms, and you can see there is a uniform biofilm growth. And then when we have treated our nanoparticles with this biofilm, so that there is this one specifically is having some very kind of porous nature, which is actually because of our nanoparticle being penetrated inside the biofilm. Because the nanoparticles contains gold nanoparticle, so they are actually SPR active, surface plasmon resonance active. So that means you can use a laser to activate its surface plasmon resonance and degrade the biofilm in a much better way. That is exactly has happened in the fourth picture, where you can see there is very high degree of degradation. We have calculated how much it can be degraded. So over a period of time, what we understood that possibly pyocyanin can be a very good moiety which can be targeted for Pseudomonas aeruginosa, and can also be used as a simultaneous targeting as well as killing target. And we can use our nanoparticle to kill the Pseudomonas aeruginosa as well as its biofilm. And then it is having multiple applications. During the pandemic time, we have now understood that, how good, how important it is to understand our basic hygiene. So the nanoparticle can be used for different other applications, like for face mask or for some, food coating agent and all these things, because we have studied the toxicity and could not find any significant toxicity for these nanoparticles. The next part that I would like to take you to is an area where we are working significantly on developing some artificial enzymes. Now, these artificial enzymes has been tried long back. I mean, people are working on that one because of the biological enzymes. They have very high specificity, but unfortunately, there's cost of production, and also because they cannot be reused. So people are looking into some different options. So our main idea was to look into developing some nanoparticles which themselves show some biological activity through enzymatic application. So we have used two different approaches, in which in nano enzyme approach, what we have done is we have attached the enzyme on the nanoparticle surface and used it as it is. In the nanozyme application, what we have done is we have used the nanoparticle as enzyme where we do not have any biological unit, and then we have used whether they have any significant role to play for catalase and for oxidase activity. We all know, why people are slowly moving towards this enzyme applications. It's most probably because they want reusability and to reduce the cost and everything. This particular area has grown over a period of time, where 2007 first iron oxide nanoparticle has been reported to have a peroxidase mimic. We all know that under oxidoreductase class of enzymes, there are multiple enzymes which have shown, there are multiple materials which have shown this catalase and peroxidase activity. So what we try to look into is whether we can use our nanoparticles for nanozyme application. In this case, like this is a tree which we have used as a source. From there, we have synthesized our nanoparticles to different chemical conjugations, and these are the sizes of those nanoparticles. You can see they are close to like 110 to 200 nanometers. We have carried out the enzymatic activity assay through TMB, and both catalase and peroxidase activity we have studied. We found out good Km and Vmax value. Now, the idea was not to just check its enzymatic activity, but also to use them for some practical applications. In case of tumor microenvironment, there are two predominant things that happen. One of them is they are very highly acidic in nature, and number two is hypoxia condition. Hypoxia condition is because tumor tries to be in a hypoxic condition so that it can grow in a metastasis state. So all the chemotherapeutic drug that we use, they do not act properly if at all we if we are not able to remove this hypoxic condition. So what we have done is this hydrogen peroxide that is there, it has been found that tumor microenvironment contains very high amount of hydrogen peroxide. So what our strategy was, whether we can use that hydrogen peroxide itself as a source for developing oxygen so that we can relieve the hypoxia. And exactly that is what we have monitored through different other mechanisms, and we looked into different parameters. We found out ruthenium bipyridyl tribipyridyl chlorine complex is actually one of the complex which binds with oxygen and gives you the red color. And when there is no ruthenium oxygen, then it will not bind. And that is what exactly you can see, that when we have treated our nanoparticles which are having catalase and peroxidase activity, which has converted the hydrogen peroxide to, when mostly hydrogen peroxide is being converted by peroxidase to produce good amount of oxygen. Then we have checked whether a sulforaphane, which is a very good chemotherapy drug molecule, can be used for cell viability, and we found that before and after the treatment, the cell viability has drastically decreased. So that means relieving the hypoxia condition can be a good strategy. We are working further on this particular aspect and hopeful to come out with some solution in future. We looked into different, this hypoxia-induced factor and through Western, and found that after treatment with our nanoparticle, there is a reduced expression of those one. Now, this particular area can, which our focus is to use them for different other enzymatic applications, mostly move from this oxidoreductase class molecules to some other enzymes to some other enzymes. And hopefully we'll come out with some solutions where this tumor hypoxia condition can be relieved and can be used for better therapeutic strategy. The last part that I would like to quickly go through is a particular area in which we look into how nanoparticles under in vivo condition, they interact with the biomolecule selectively. Now, when we have any nanoparticle, under in vivo condition, there is supposed to be a protein corona formation. A protein corona is nothing but a assembly of proteins which are bound to the nanoparticle surface the moment it comes in contact with the blood. That is because of a very basic principle that the adsorption property and the electrostatic interaction of the proteins with the nanoparticles. This is actually a major drawback while designing any nanomedicine because the off-targeting results to a very high extent. So our idea was to find out to few of the proteins which can be specifically designed and put on the nanoparticle surface so that we can look into those proteins and make for targeted delivery. So specifically, we have designed some citrate and CTAP coated gold nanoparticles, and we looked into their characteristic technique and find out their size. So what has happened is that now these nanoparticles initially are being, what we have done is we have coated these nanoparticles with human serum albumin or red blood cell proteins. Now, through this mechanism, what we have done is we have made a coat on the nanoparticle surface by which the macrophages will not be able to detect. So this is called stealth effect in nanotechnological term. And what we have found is that when we treat these nanoparticles with the biological system under in vivo condition, the proteins which are attached, they are not recognizable by the macrophages. That is the don't eat me proteins. But when you use the nanoparticles in as it is form, they are being recognized very initial phase, at the very initial phase by the macrophages, and the macrophages finally degrade them. So before entering to their desired kind of work, they basically being eliminated from the body. So our strategy where we have put the nanoparticle with the proteins can actually inhibit this macrophage detection and can go for a much better option where we can use these nanoparticles for targeted drug delivery. So this, just to give you a brief, you can see the protein coating on the nanoparticle surface, which is close to 1.5 to 1.6 nanometer coating. We looked into thoroughly LCMS studies to proteomic analysis we have carried out, and we found out what are the proteins, and majority of the proteins we have analyzed that once the nanoparticles are being coated with the proteins, there are higher chances that possibly the immune systems will not recognize. And this is having a significant impact on the future, both nanomedicine applications as well for diagnostics, because now in nanotechnology, which is related to biomedical applications, we are mostly talking about the personalized medicine, and this coating of personalized proteins of individuals' body, the blood from which the donor can actually give its blood and can coat the nanoparticle using this one, and have a much better application with respect to a lot of biomedical applications. So I hope I have not gone beyond the time that I was allotted to. Lastly, I would like to acknowledge the real superstars of my lab group. I'm here only present on behalf of them. A few of them have already got their degree, and they got their, they are now not present, but I'm on behalf of them, and few of them are still working. The work that I have presented is a collective work of all Dr. Avnish, Rubel, Shanka, Anika, Chandni, Ashish, Sayyed, and few of them who are already present in this lab like Aki, Mohini, Shivani, Trilok, Manik, Anjali, Rahul. I would also like to acknowledge our Director CSR HPT for his support, constant guidance, and also the instrumental facility of HOD Biotechnology and other departments. And also coordinator ACSR for giving me very brilliant students. It is only because of them that the work has been possible, and also the financial support from CSR HPT, CSIR, DST, and DBT. Lastly, I would like to thank you, and I'll be happy to answer any of your queries if you have.
Thank you, Dr. Amitabh. It was a wonderful lecture, and being the first, it had a great importance to make the people introduced to the subject. Now, you must have seen from the ancient culture in India, we have been using different type of churns, rajat bhasma, swarna bhasma, lep, kadhas. So all these are aiming at making the molecule at a smaller size So it is so that from the ancient times, we were knowing nanotechnological applications and now we are refining it.
Yes, of course. Of course. I mean, thank you very much for asking these questions. Actually, yes, of course. I mean, the bhasma, which is being very heavily used in Ayurveda, that has been now being accepted as one of the composition material where you have actually nanomaterial composition. Bhasma also contains a lot of inorganic metal ions, which is actually has been seen through microscopy that they are in different oxidation state, and those metals are actually present, like mercury and other metal ions. Traditionally, it has been reported, even a few of the literature reports suggest that in the very first phase where, in our ancient days where a kid used to be fed through this silver spoon and all these things. Silver is known to have antibacterial activity over a period of time. So few people believe that our ancestors possibly they knew this particular aspect and have actually gone through that one. Similarly, nowadays, this high dilution effect in homeopathy has been a subject of very great deal of research. People are working on that. And I believe, yes, all these aspects, it's nanotechnology, possibly the term has come very recently, but it has been, it is there, it was there, and it will be there in future, obviously.
Thank you, Dr. Amitabh Acharya. When science, health, and technology joins hand, it will always benefit the public and their health. And it will help AIIMS also to improve the services to the mankind. Thank you.
Thank you very much. Thank you.
Thank you, sir, for your excellent presentation. So if there are any questions, I think it can be put in the chat box. We can discuss at the end of the session after this for the participants who joined online.
Dear friends, we have got with us the President, National Academy of Medical Sciences, Dr. Shiv Sarin with us. I'll request Dr. Shiv Sarin, sir, to unmute himself. And bless us with his speech. Please unmute Dr. Shiv Sarin, sir.
I think we have lost contact with Dr. Shiv Sarin. So we'll continue with the next topic. So next topic will be on nano encapsulation and cure of diabetes mellitus, a therapeutic option for insulin dependent diabetes. So the lecture will be delivered by Dr. Ravikant, Professor, Department of General Medicine, AIIMS Rishikesh. So over to you, sir.
Hello, am I audible?
Yes, sir, you are audible.
Yeah, so is my screen visible?
Yes, sir, screen is visible, sir.
My presentation is visible?
Yes, sir, we can see the slides.
Yeah, thank you very much for inviting Professor Agrawal to this August gathering and wonderful conference. I am Dr. Ravikant to deliver my talk on nanoencapsulation and cure of diabetes mellitus, a new therapeutic options for insulin-dependent diabetics, whether it's type 1 diabetic or type 2 diabetic with primary or secondary OH failure. So greetings from the Devbhoomi and AIIMS Rishikesh to AIIMS Raipur. I think we started our journey way back with this kind of a, vehicle. We had some destination to move, but we needed something better versions. So this is typical, I mean, one dietary modification and diabetes management. Then we switched on to OHAs in the form of cars. There were ups and downs, but we could do fairly well. Then we even invented insulin therapy to control diabetes. But is it really the problem of insulin with the diabetics? So you need to think differently to target population who are diabetics, who definitely requiring insulin to a larger extent. Now to come to the development of drugs for diabetes and the encapsulation in diabetes, basically, what are the milestones that we have achieved? So discovery of insulin and successful methods of beta cell replacement was one of the major breakthrough in the development of anti-diabetic treatment modalities. The central roles of pancreas and insulin producing islets of Langerhans in development of diabetes were established in 19th century itself. For over 50 years, islet transplantation has been rigorously studied in animal models with hope of establishing a basis for human transplantation. The pancreas transplantation remains gold standard for patients having difficulty in controlling their symptoms with exogenous insulin. But it is cumbersome and not possible. Most of the centers in India, they are not well equipped with the transplant surgeons, and the pancreas is short in supply. The Edmonton Protocol, which was developed 20 years ago, incorporated a large dose of islets from multiple donors with a steroid-free immunosuppression, helped to establish the modern era of islet transplantation almost 20 years ago. And currently, we are basically following the Edmonton Protocol for the islet cell transplantation. While islet allotransplantation is recognized around the world as a powerful clinical therapy for diabetics, it is not yet recognized as one of the main armamentarium to treat diabetes mellitus. In fact, islet allotransplantation is able to provide similar results with some advantages, making it an attractive potential alternative for the main therapy for diabetic patients requiring insulin. Recently concluded in successful phase 3 clinical trials, we have the best chance yet to establish islet transplantation as a treatment for diabetes in India as well, as it is already recognized in other parts of the world. Now, this is the timeline. Diabetes was given its name in 150 AD, and a lot of time has passed since we, came up with the oral drugs, insulins, and nowadays in 2016 onwards, clinical trials on islet cell transplantation, in fact. Now, what is this islet cell transplantation we are talking about? In islet cell transplantation, beta cells are removed from a donor's pancreas and transferred into a person with diabetes. Beta cells are type of cells found in the islets of pancreas and produce insulin, which regulates blood sugar levels. Depending on the blood sugar level, the insulin is basically secreted from the beta cells. So once transplanted, donor islets begin to make and release insulin on its own. Now, what are the benefits of islet cell transplantation in diabetic patients? So once transplanted, islet cells resume their role of releasing insulin to maintain normal blood sugar levels in response to food, exercise, and other changes in body. Now, successful islet cell transplantation can provide following benefits. They restore or improve body's ability to regulate blood sugar levels. Need for frequent blood sugar measurements are definitely lessened in this population. Daily insulin injections can be reduced or can be obviated and can be eliminated from the patients who are receiving. So being free from insulin injections may only last several months or a year. So one single injection can last for months or years together. Islet cell transplantation reduces episodes of blood sugar which are really low and high, because this is basically a physiological treatment and not something which is administered from outside. It definitely improves quality of life because the patients are free of insulin injections 3 or 4 times a day, or maybe single time. It's bothersome to patients. The reduced progression of long-term complications of diabetes, including cardiac disease and many more micro and macrovascular complications associated with uncontrolled diabetes mellitus, because the time in range becomes very high, maybe 80%, 90%, which is recommended by ADA basically. But there are, of course, risks of islet cell transplantation. What are these risks? With all organ and tissue transplants, the rejection of donor cells is the greatest challenge. This is the challenge which we need to eliminate from our nanotechnology methods, basically. All transplant recipients must take, for the rest of their life, strong drugs to suppress the immune suppression and prevent rejection. So with these challenges, how islet cell transplantation is possible? Because it's, of course, increasing the cost of therapy to a huge amount, which of course is not possible in Indian scenario. So now how these problems can be taken care, and how we can develop a much more successful technology in the form of nanocapsules that can deliver the insulin or beta cells in much cheaper cost. Now, how successful is islet cell transplantation for diabetes? Let us see and have a review of the literature. Scientists developed a procedure for transplantation islet cell to treat diabetes in 1960s. It was very old kind of a system. First transplantation was attempted in 1990s and succeeded only 8%. In 1999, a clinical trial conducted at the University of Alberta in Edmonton achieved 100% success rate. So you can see over a year of 9 years duration, they could claim from 8% to 100% success rate just by improving the methodology. All the patients in their trial were freed from the need for insulin for at least a month. So all these 100% patients did not require insulin for a month or so. In 2009, 70% of adults with diabetes were free of insulin injections for the first year, 50% for second year, and 35% for third year. So this is the huge success, right? From 8%, from 1 month, we have jumped to 3 years. So this technology really it can change the future, the way we see at the treatment of diabetes. Now, why isn't islet transplantation used more often to treat diabetes? Because donor islets are in short supply. Islet transplantation is considered as an experimental procedure yet. Islet transplant recipient must take immunosuppressants for a pretty long time. So how we can take care of all these shortcomings? Basically, that's a challenge. Now, what is the future of islet cell transplantation research, especially what we are doing at AIIMS Rishikesh and the country at large? There is a need to collect enough islet cells to do the transplant. Prevent rejection and need for immunosuppression has to be brought down to minimal or to negligible, so that patient doesn't require any immunosuppression throughout life. One approach involves coating islet cells with a special gel that prevents immune system from recognizing and targeting donor cells. This is one way of taking care of the immune cells or immune attack of these beta cells. Now, this is how we are working at AIIMS Rishikesh, and we have developed these nanocapsules. Now, to introduce nano encapsulation procedure, the specific islet cells are contained in a protective capsule. Capsule is implanted into body where cells are capable of producing insulin. Capsule is especially designed to allow blood to feed cells within capsule with oxygen and nutrients which are required by the beta cells, and allowing hormones to leave the nanocapsules, which is required by the body, basically. So this way, it can be prevented. The immune system can be, prevented from attacking the nanocapsule by the cytotoxic T cells and entering capsule and killing of islet cells. So this is one way of, doing the nanocapsule and preventing the immune attack of these nanocapsules. There are various types of encapsulation: macro encapsulation, micro encapsulation. We are basically working on nano encapsulation. So the approach is to enhance oxygenation and improving graft survival, reduce microcapsule size to decrease diffusion distance from oxygen to reach islet core from the capsule surface. And there are two methods by which we can do it: by conformal coating and layer by layer coating. What we are doing, we are basically doing layer by layer coating of these beta cells. We have started working on layer by layer nano encapsulation procedure, and in few slides, I'll let you know how we are doing. So what is the future and the scope of future research in diabetes? This is an open area of research to establish standard nano drugs, explore more advanced insulin therapy, the nano formulations, delivery, and the pathway through which they act, basically. drug capable of bringing about corrective modulations of some parameters can be explored. So use of alternative drugs, they can be one of the potential through the nanotechnology formulations, as my previous speaker has already told. I'm not going into the detail. Now, how the nanoencapsulation is advantageous in diabetics. Number one is a protect core material by preventing environmental oxidation or deactivation, separate incompatible materials, modify properties of the core material, convert liquids to free-flowing solids, controlled and targeted release of encapsulated cores, modify core material by a release, develop novel products. These are the aims. Our goal from lab to the patient, realistic research. The idea and the challenges were to design an capsule, to develop in the lab, to have a quality control of these capsules, and scale up to commercial quantities may be the next step that we are looking forward. So what are the encapsulated islet cells? I've already told the specific islet cells contain protective capsule. Capsule is maintained in the body where cells are capable of producing insulin. So they are designed in such a way that no cytotoxic T cell or immune complex can attack beta cells. Rather, the nutrients, oxygen, and other nutrients can, pass through the nanoencapsule and reach the beta cells. And in turn, beta cell releases the insulin and it is given to the body, basically. It will be evidence-based to first conduct a study in vitro before directly conducting it on humans. This study will determine whether encapsulation of islet cells in subcutaneous region could protect implant from rejection and organ destruction in diabetes. In this study, we'll be able to develop encapsulated human islet cells for commercial production. So the aim was to see the effect of encapsulated human islet cells implantation on insulin dependency in diabetic patients. There are a few objectives with that. Objective one was to culture human pancreatic beta cells in suitable culture medium, to encapsulate beta cells of pancreas in capsule and check beta cells viability and insulin release, the dynamics associated with glucose and insulin release therein, to implant encapsulated pancreatic beta cells in subcutaneous region of Wistar rats, and to replicate encapsulation process and implantation subcutaneous region of the diabetic patients. There are 4 objectives with us with which we went. So we initially thought we will take the beta cells from the cadaver, which is easy, but then we tried a lot. We could not get the cadaver well in time. So then we switched on to, surgically remove pancreas by the gastro surgery department and one colleague from our surgery department. So we treated this pancreas with collagenase enzyme and many more enzymes. We isolated the islet cells from the pancreas. We encapsulated the islet cells. The histopathological examination of encapsulated islet cells was done, and the cell viability of encapsulated islet cells was done in the lab. Gene expression analysis of human insulin was done in this nanoencapsule, and we saw the dynamics of insulin release with varying concentration of sugar in the media that we provided. Then we implantation and encapsulated cell in the abdomen thigh subcutaneous region of the Wistar rats. This is phase 1. Phase 2 was the post implantation monitoring and explantation to ensure that the beta cells are still functioning, analysis of the islet cell function and statistical analysis. So we have completed the phase 1. We are going for phase 2. Phase 3, of course, the patient trial. The study design, preparation of capsule, phase 2 standardization of capsule and implantation in the Wistar rat, and the phase 3, of course, is implantation in human diabetic patients. So what we did prepare the beta cell and capsule, right? We are yet to implant it into the Wistar rats. The hypothesized success rate is 90%. Diabetic patient will show decrease in dependency on insulin from outside. Without secondary effects, 70 to 80% we are presuming the success rate will be. This is the real life lab parameters that I'm showing with you. They are results. Isolation of islet cells. You can see the figure A is having mechanically chopped tissue pieces of pancreas from before digestion, and B is digested pancreas, which is homogeneous tissue suspension of collagenous perfused pancreas after 30 minutes of incubation, shaking water bath at 37 degrees Celsius, followed by 30 seconds of mixing by hand. And in figure C, you can see that islet suspension layer is formed between HBSS and the densely gradient after centrifugation. Now you can see the islet morphology. In figure A, you can see a freshly isolated islet shown among pancreatic acidic tissue on the day of isolation. B is islet purified from acidic tissue after incubation 37 degrees Celsius. And C is islet incubated at 18 to 20 hours with darkened hypoxic center. So now the representation of islet cells, there are good islet cells which were separated. There could be bad and exuberant tissues. So we need to take out good islet cells. Good islet cells means they have a regular circular surface, whereas bad one is having a ragged or irregular surface or margins basically. So this is the result cell viability test in the lab. You can see that figure A and B, they shows islet cells after 24 hours of incubation. C and D shows treated islet cells with fluorescein dictate. E shows one encapsulated islet. So you can see the islet is in black and white, and FGH shows outer layer of encapsulated islet cell, indicating well permeable membrane to allow movement of different molecules in and out of capsule. And in figure I and J shows nucleus of islet cells, indicating live islet cell even after 48 hours of incubation. So these are the glucose and insulin release dynamics at 3.3 millimole glucose concentration and 16.7 millimole concentration, almost 5 times. So you can see that uncoated and nano encapsulated, the response rate is quite predictable at variable glucose concentration. You can see the two peaks. The first peak in figure B is basically preformed insulin, and second peak, which is basically following the nano non-encapsulated islet and blood glucose dynamics, whereas uncoated islet cells, they have similar dynamics so far as the varying molar concentration of glucose is concerned, right from 3.3 millimoles to 16.7 millimoles. So it is a very predictable insulin release in the lab itself that we have done at AIIMS Rishikesh. So conclusions are, if encapsulated islet cell work to prove, it will prevent too high and too low blood sugars from occurring, eliminate the need to take insulin, prevent need for daily blood glucose monitoring, and encapsulated islet cells would represent a near cure experience that could see people control their diabetes with minimal management for many months. So this is a video as to how this will work.
Hello. Nice presentation, Dr. Ravikant. Just
Yeah.
please tell me the last study, whether it has been done at Rishikesh?
Yeah, it is being done at Rishikesh, sir. I just finish up my study with my concluding remarks.
Okay, nice. And who, how many cases have been done?
Sir, actually, to be very frank, we did not get the human approval. The file was with the, with AIIMS Rishikesh, then PGI, then to Government of India. But we did not get a human approval. So we are doing it on, rat model first. And we will be, implanting this, material by next month, basically. And now our nano encapsule have survived for 2 months. So we have the study for last 2 months. The nano encapsule is still producing the insulin. The viability is, I believe, will be more than 3 months. So once it is injected in subcutaneous tissue or diabetic patient, it will be there for 3 to 6 months. So they will not require any insulin for 3 to 6 months, hopefully, which they are requiring every single day. So with this, I thank you all very much. I think we started with that rocket that is having no target, but now we are targeting. With this, I thank you all very much for patient listening and giving us the opportunity.
Dr. Pandit has some queries.
So thank you, Dr. Ravikant, for such a lucid presentation, and you have gone through the islet cell transplantation, encapsulated islet cell, and then the release of insulin, and other things. So what is the future, sir? How much time it may take for something which is where our patients with IDDM and other things maybe really benefit, like
So if you Google, only 3 days back, the US FDA has given approval of this, stem cell therapy via nanotechnology, wherein they are converting stem cell into beta cells, and that is producing insulin. Now US FDA has granted approval for type 1 diabetic patients 3 days back only. So our nano encapsule is ready for use, sir. This is ready for use. I hope the moment in next 3 or 6 months we are through our animal model studies, the human study will be started, because till now there was a hue and cry throughout the ethics committees wherever I sent my proposal. They said it is in the experimental phase, we can't approve it. So I did not get the approval for approximately 5 years. But then we have to go for the rat model, and now the rat model, we have prepared our nano capsule which is working. Maybe next week we'll be injecting it, and we'll be seeing the dynamics for a month or so in this rat model. And once it is through, I think we can definitely go ahead for the human study. I don't see any hitch as of now, because US FDA has already given approval for type 1 diabetic patients.
thank you so much, Dr. Ravikant, for such a wonderful presentation. If anybody has got any questions, I think they can put in chat box. So we can take it after the one more session is pending. So since we are
Thank
short
you.
of time, we'll finish the next session.
Thank you, sir. Thank you very much.
So next session will be on role of preclinical studies in drug discovery and pharma research. So the faculty we have is Dr. Yogendra Padwad, Principal Science Engineer, IHBT, Palampur. So I welcome you, sir, Dr. Yogendra Padwad.
Thank you, sir. Just I will.
Thank you so much, sir. Thank you, Ravikant.
Thank you, sir.
My slides have got shared.
We are not able to
No,
see the
unable to
slides.
see your slides, sir.
Okay, I have shared them.
Yes,
Now
now
we
it's
are able
visible.
to see.
Okay, fine. So now I made them a full screen, and moving forward and backward. Is it okay for you?
Yes, sir.
Okay. So good morning all, and respected chair, respected director, sir, head departments of different head of different departments at the AIIMS, and dear students. First of all, I would like to give my thanks to the organizer who gave me an opportunity and to interact with you and present some of our concepts as well as some success stories, specifically emphasizing on the preclinical studies in our setup at CSR IHBT Palampur. The preclinical study, as everybody knows, are the very integral part of the any. Am I audible?
Yes, you are audible. Please
So,
continue.
integral part of any drug discovery program, the pipeline of the drug discovery program, and also with the specific emphasis of this particular symposium, this particular program, that is the nanoparticles and nano-based pharmaceuticals, as well as the nano-based diagnostics. So as my colleague Dr. Amitabh, and after that Professor Ravikant also emphasized, the importance of the testing them at various levels before entering into the clinical pipeline or the clinical studies pipeline. It is very, very important to establish its efficacy, its safety, its bioavailability, its mode of delivery, and everything. And for this particular aspect, the role of preclinical studies is very, very important, for which again comes under the very well part of the Schedule Y specifically prescribed in the Drug and Cosmetics Rules in 1945 and modified further as time again and again. So it's a very much part of the CDSCO guidelines to arrive at the conclusions and the submission of data where it requires to submit for conducting and getting approval for the clinical trials, and or in other words, we can say the human intervention studies and established its clinical relevance to further bring it into in the form of drug or nutraceuticals or the pharmaceuticals in the market. So for this, the CSR specifically at the CSR IHBT Palampur, where we have a specific emphasis on the developing naturals in the form of pharmaceuticals, phytopharmaceuticals, as well as nutraceuticals targeting different disease conditions. Different conditions arise due to the deficiencies of different nutritions what we required for the healthy functioning during the day-to-day life. So the CSR IHBT is specifically focusing on these particular aspects, which emphasizes on the central nervous system, enhancing nutrition to combat the bone-related issues, targeting non-communicable diseases like diabetes, inflammation, cognition, and immunity. So targeting specifically for immunity modulation and immunity enhancement. The approach is to find out a best natural source in the form of the medicinal plant. And as we all know that the Western Himalayan bioresource is harboring hundreds of medicinal plants, thousands of medicinal plants, which contains such wonder molecules which have a very important pharmaceutical and nutraceutical values. But however, they are also traditionally being used in different parts of the region. But the most important thing and the demand of the today's is to validate this traditional knowledge, validate their importance, applying modern techniques and modern scientific basis, which is also one of the mission of the government of India, specifically the health sector. The standard to develop a standard extracts and characterize them in the form of specifically emphasizing on important bioactive compounds is one of the very important activity in this direction. Then testing them in silico and in vitro evaluation for them. If you have 1,000 of molecules, it is not like possible to take them in the cell-based as well as animal model-based studies, which are a part of preclinical evaluation. And therefore, using artificial intelligence, screening them through a different computational platforms, which is very much available in our setup, and bringing out the most valuable molecules out of this particular activity, and then testing them further into the cell-based models for in vitro evaluation, and then taking lead further a very small number of molecules which we have evaluated through this particular pipeline in the animal model based studies. So testing the putative leads in vivo models of disease, specifically targeting the diabetes, epilepsy, liver and kidney dysfunction, and also going further to generate a knowledge around it by studying underlying molecular mechanism, that where these particular molecules are targeting in the system, in how it is modulating the pathophysiology of a disease, how much they are beneficial, and how much dose response we will be getting in the form of treatment, as well as their bioavailability and pharmacokinetic behavior. Now, in the interest of the students, I will just go through some of the very important aspects of the preclinical studies, like what is in vitro and in vivo studies, because many people understands, or some people may not be able to understand those who are not from a very basic science background, that the what are in vitro and in vivo assays calls for. So in vitro, the animal within the particular part of experimental procedure, when we are performing the experiments outside the living cells, we call them as in vitro experiments, whereas when we are using the living animal models, the living animals for studying and testing our molecules for their efficacy, for their toxicity, we call it as in vivo experiments. So cell-based cell culture experiments in a petri dish, in experimental setup of the animal cell culture lab, comes under the part of in vitro, and when we are using the models such as mice, pig, rabbits, apes, and similarly the lower animals like the C. elegans, as well as the fish, for example, the zebra fish, comes under the in vivo studies. And when we go for logistics, like the in vitro are a little less expensive studies, however, in vivo require a relatively expensive platform, and this provides the results we get using in vitro studies is quite faster. However, in vivo studies, we need to completely study the drug treatment regimen as that of the clinical trial. However, in respect of clinical trial, in vivo studies are again low time consuming, and this is however, the in vitro studies are considered to be the less precise. However, they are giving a valuable information to arrive at in vivo experiments. However, and the in vitro, however, the in vivo experiments are being considered as the very relevant data producing the methods to bring us closer to the start of the clinical trials. Now, in vitro states are experimentally controllable. The advantage are they are repeatable. We can repeat them many times because of the affordability. However, we can't do it with the animal studies because a lot of regulatory issues are involved, with the ethics committee approvals of involved, and therefore the repeating the experiments with animals have a very limited chances. However, and the in vitro tests are fast, inexpensive, and simple, and the small amount of material is required for in vitro analysis. For example, the molecules which are being isolated from the plant sources, as well as the derivatized and synthesized molecules, they synthesize in the laboratory conditions at a very low quantity, and therefore testing them in a larger context, specifically in the animals, and having 6 animals or 6 to 10 animals per group require higher quantity, and specifically giving repeated doses. Therefore, to screen out the molecules when they got synthesized in a smaller amount, and then is very much feasible in vitro condition, and then once the molecule qualified for the in vivo study, then its manufacturing or its production can be further enhanced for the in vivo studies. The in vitro test can be very well compared for their quality with different standards, and ranges of application for in vitro study is quite vast. We can ages of the in vitro.
Please check voice is not coming.
Yeah. Is slide moving?
Yeah, you can now speak, sir.
Yeah. I mean, am I audible?
Audible, sir, audible.
Okay, fine. So I was here at this particular slide, and I'm moving to next now. So to bring the to bring the tissue in the form of cells and then studying them for different effects. We called it as a primary culture, and then subculturing it at different stages, and we ultimately result into the continuous cell line of different tissue representatives. So this particular exercise is very, very important in preclinical research. Specifically, when we have a tissue in hand, we can convert it into the primary cells and then test our molecules at different doses at different concentrations to these particular primary cells. And when we continuously go on cultivating primary cells under different conditions, we ultimately result into the continuous cell line. This continuous cell line have is gain a phenotype of transformed cell line, which actually lost its control on its proliferation and will ultimately result into the cell line which continuously grows in the artificial conditions, the laboratory conditions in the CO2 incubator. And we can use them further for testing different molecules for different disease conditions, creating different disease models also in a cell-based studies is also very much possible, as I will show you some of the examples in my further slides, and then testing them at different concentrations and different doses is possible. So, so skip this. So this is just just how the tissue can be converted into cells, and then this particular cells can be further grown, and we can obtain a secondary culture in the form of continuous culture, and the primary culture can be tested for different effects, for specifically the effects of the pharmaceuticals at different concentrations. So if the tissue is of the cancer origin, the primary culture what we obtain from these cancer tissue is a transformed cell. So ultimately we get we get the continuous cell line directly from the transformed tissue, whereas if the tissue is a non-cancerous normal tissue, we need to grow it further and select the cells which have lost their control on the proliferation, and ultimately results into the secondary culture and further into the continuous culture. So primary culture is nothing but it refers to the stage of the culture after the cells are isolated from the tissue and under appropriate conditions occupy the substrate which has been provided for them to grow. And at this particular stage, the cell need to be subculture and then transform into and transfer them into the new vessel for providing them further nutrition and a space to grow. Now, after subculture, when the primary culture become known a cell line or subclone, and this particular subclone or cell line to be further divide into a different culture vessels and further grow them and passage them. Passage means to provide them more better conditions, fresh nutrients, and fresh plants, everything. So cells with the highest growth capacity predominates, and by resulting into the genotypic and phenotypic uniformity in the population. And this is a very important aspect to ultimately result into a best cell line or the best cell culture, which have its uniformity in genotypic and phenotypic aspects. So the population of the cell line, which is positively selected from the subculture by cloning on some other method, this particular cell line becomes a cell strain. And this cell strain we can use as a specific representative tissue cell to study any disease conditions in vitro. For example, if you have a different cancers, different tissue representative cancer cells, we have the cells presenting beta islets of Langerhans. We have a cell line established like the adipocytes. We have a established cell line for the muscle cells to study the antidiabetic potential of the molecules, and many such hepatoprotective effects also can be tested by using the hepatocytes, the hepatocyte cell line, and all different parts, specifically neuronal, also brain cell lines. So this particular cell strain is very, very important to screen out the most effective molecules which we are screening against different disease conditions. this is how the cell, this is how we generally process the tissue to arrive at the cell, arrive at the particular tissue representative cells. Like for example, if you have a particular tissue to be converted into a cells, then we have to test it with the different enzymes which dissolves the matrix in between the cells, which by which they are attached with each other in the form of 3D tissue, 3D level of tissue, and then test them, treat them with this particular enzyme cocktail, which converts it into cell or tissue cells or the primary cells, and then subculture them in the form of secondary culture, and then as I explained earlier, the cell line. So the selection of particular enzymes to treat the tissues of different organs is also very, very important, because different tissues are organized in different ways with different components of the extracellular matrix by which they have attached with each other. So accordingly, the different enzyme regimens need to be selected to treat them and safely dissolve the matrix in between them, and ultimately convert them in the cell in the form of free flowing cells, and result ultimately getting the immortal cell line as well as the primary cells. So this is how we ultimately get the cells. For example, the fibroblast cells, the epithelial cells, endothelial cells, and neuronal cells. Some of the cells representative photographs I have just put here in this slide. How the fibroblastic cells we ultimately get, and then epithelial cells, the endothelial cell lines, and the neuronal cells. When we observe them under the phase contrast, inverted phase contrast microscope, how these cell lines would look like. So this is the particular representative graph that each and every cell line present in our system of animal cell culture lab should have this particular graph plotted for the every cell line, which represents the growing pattern of each and every cell line. So it's like the cell number versus the time. How can we particularly grow the cells and then passage them to make them to keep them and maintain them metabolically very active form, so that if the cell lines loses its metabolically active status, then we may not able to get the desired results what we are actually looking for. So we may ultimately result into the erroneous outcomes, and therefore it is very important to study each and every cell line, its growth pattern, and establish such a curve for each and every cell line, and actually study when we are growing them in the cell culture conditions, we have to follow this particular pattern. Now, the applications of cell-based studies is very, very important in the pharmaceutical pipeline. So the cell lines can be used as a model system for different diseases. For example, in case of cancer cells, in case of anti-cancer activities, the representative tissue cancer cell line can be used to find out the very minimum inhibitory concentration of the molecule at which the 50% cells ultimately dies, ultimately can be destroyed from the population. And and this particular activity value, when we obtain, it is nothing but a concentration at which the 50% cells can be targeted. And this particular concentration can be further studies for underlying mechanism that how the cell death occur, whether it is apoptosis, whether it's necrotic, or it has created a cellular senescence ultimately. And then studying its molecular mechanism, whether which particular pathway it induces for the cell death, whether it is interfering into the cell cycle regulation or not, it is inducing apoptosis, and which particular pathway it is targeting, which particular molecule it is targeting, whether it is kinase inhibitor, whether it is whether it is nuclear localizing or whether it is cytoplasmic localizing molecules. So all this can be studied as a model system. Specifically in the case of diabetic condition, also we can induce a differentiation of the adipocytes into the very mature adipocytes and study different the anti-diabetic effects by specifically treatment with the anti-diabetic molecule in question at different concentration. Toxicity testing is also a very important part of the preclinical study pipeline, and the highest concentration which is, or the lowest concentration which is injurious to the cells also can be determined, and find out the safe concentration at which we can study the molecule for its efficacy further also can be found out. So as I already explained within the cancer research, virology, the well-established virology lab cannot be possible without animal cell culture based studies or animal cell culture based lab, because these viruses are obligatory parasites and need to, even for their growth, require animal cell culture based setup. So studying the virus pathogen, virus host pathogen interaction and the antiviral immunity, for that this is very important to have a viral infection based model in different target cell lines, and then studying its outcomes, the productive infection, and measuring the infectivity of the viruses. And when the molecule is tested for the antiviral molecule, then how much the molecule is capable to inhibit the viral replication as well as mounting antiviral immune response. Then cell-based manufacturing, as earlier talk, as Dr. Ravikant explained about the insulin production, which is being done nowadays in the form of the biotherapeutic in a cell-based manufacturing, specifically the CHO cell line based manufacturing, and then bring it in the form of injectables in the market, studying different immune responses, cell interaction in immunity and cell differentiation to mount effective immune response in for the against infectives and maintaining it in the inflammation. And that is very important to study in the form of cell-based studies. Then genetic engineering in these cells are, as I told you, the cell-based manufacturing, the different bioengineered molecules can be generated and expressed in the cell line based model, and they can be expressed and purified and characterized for their biophysical, biochemical characterization as a biotherapeutics. Gene therapy, although not much have gained success, but still the gene therapy is one of the very important aspect which we can interfere into the stem cells and then further differentiating them into the very last stage committed cells, and then transplanting it into the tissue where the defective tissue, or replacing it with the defective tissue, so that we can gain the best performing phenotype there, and intervene into the disease conditions to use it as in the form of treatment. Drug screening and development, so screen a lot of molecules. For example, in the drug discovering industry, their chemical technology or the chemistry department manufactures thousands of molecules, but then the molecules has to be identified. The best molecule, the best pharmacophores has to be identified for the most effective molecule or the pharmaceutical. And for that purpose, the cell-based screening is very important at the industrial level also. So moving into the some in vivo part of our setup. So like this is particularly regarding the success story of Picrorhiza curva, very important medicinal plant which harbors the molecules like picrosides, the apocynin, vanillic acid, and all. And after chemical characterization, we studied it for its anti-diabetic activity, specifically in a type 1 diabetic condition, where we studied that how much it is actually capable to induce the insulin response. And to our experience, we could find out that the gene expression we studied for specifically the insulin gene expression after the treatment of the model, right, like the type 2 type 1 diabetic rat model, we found out that the destructed tissue of the beta islets got regenerated by the different concentration of the Picrorhiza curva extract and its enriched fraction, what we called it at the Kutkin, at a different concentration. And when we studied for the insulin gene expression analysis, we found that more than 100 times the insulin gene expression could be enhanced under the treatment at different conditions. And it is better responsive than that of the standard what we use for standard when we compare it with the glibenclamide drug. Similarly, for type 2 diabetic mellitus, when we tested the enriched fraction of the Picrorhiza curva medicinal plant, we found that the Kutkin is enhancing the glucose uptake as well as GLUT4 translocation at the membranes of the differentiated adipocytes. This is important to facilitate the entry of glucose to inside the cells and reducing the hyperinsulinemia, so that the sugar from the circulation can be consumed by the tissue, specifically the adipose tissue, as well as the muscles, and maintain the blood sugar level in the circulation. So like our observations, that it augments the GLUT4 expression and facilitates the entry of glucose inside the cells, thereby improve insulin sensitivity, enhances lipid accumulation. Enhanced liquid accumulation indicates the consumption of the circulating sugars, and then recover the obesity induced fat accumulation in diabetic animals. Specifically in this particular study was done specifically in the differentiated 3T3L1 adipocytes. Further, the other very important molecules like ferritin and fluoridazine, these particular molecules are present in the apple peel, and the industrial waste output, what we call it as apple pomades, from where these particular molecules were isolated and characterized in our setup. And we found that when these molecules, when we are using individually in the form of or in the combination, it enhances, it promotes the accumulation of cellular lipids, cellular glycerides, which indicates that which indicates that the glucose is being consumed, or the sugar is being consumed in the circulation, and which ultimately resulting into the reducing hyperinsulinemia and the glucose uptake. And further, when we tested directly the individual and combination of these molecules, it enhances the glucose uptake by adipocytes very drastically, even when we compare it with the rosiglitazone, which is one of the standard drugs is being given in the clinical setup. And the mechanism of this particular molecule, how this molecule actually targets where it targets, and how it interacts with different factors involved in the regulation of GLUT4 translocation, as well as triglyceride accumulation, and all was further studies with specific emphasis on specific transcription factor for PPAR gamma and its phosphorylated form at serine 273 amino acid. And this particular interaction was studied in silico, as well as it was validated in vitro conditions, and further also in vivo condition in type 2 diabetic mice. And when we found that the PPR gamma levels are differentially regulated by these molecules and in combination. Similarly, the one of the another important factor, the CDK5, cyclin dependent kinase 5. This particular CDK5 is also important when it as a target molecule with PPR gamma and having its potential, this particular interaction like the CDK5 and PPR gamma interaction is very, very important in the pathophysiology of the type 2 diabetes mellitus. And when we study it by different in silico as well as in vitro cell-based methods and by immunoprecipitation as well as their interactions at different under different conditions and under different concentration of the treatment as well as in combination, we found that this particular interaction is being abrogated by the by our treatment and ultimately result into the breaking of insulin resistance, breakage of insulin resistance, supporting glucose uptake and glucose uptake as well as glucose consumption from the in the circulation. So this is all about our experiences and my particular aspects to be discussed at this forum. So I am very thankful to CSIR, Department of Biotechnology, DST, and all other funding agencies for supporting funding for this particular activity in our setup. And thank you all for your patience hearing.
Thank you, Dr. Yogendra.
Thank you.
2 questions. Okay, people can enter their questions in the chat box to be replied by the speakers. Thank you.
Okay, thank you, sir.
Thank you, sir, for the wonderful lecture. We will conclude the first session for the today. I'll request Professor Amit Chauhan, sir, to present a memento to our chairpersons. Thank you, sir. With this, we'll start our next session. And I'd like to request Professor Dr. AIIMS Mohapatra ma'am, Dr. Neeta Mishra ma'am, Dr. Manju Agrawal ma'am, and Dr. Seema Shah ma'am to kindly chair the next session. Our first speaker will be Dr. Jose Emanuel, who is an outstanding young faculty chair and assistant professor from Department of Mechanical IIT Bhilai, and his topic is on nanostructured metal alloys for enhanced biomedical application.
बड़े सर का कहा वो तो ना चेयरपर्सन को वो गिफ्ट दिया निकल लिए वो
Am I audible?
Yes, sir.
Okay. So can I share the slides?
No, sir. Please share your screen.
Yeah. So is the screen visible now?
No, yes, sir. Now it is visible.
Okay. So you're not seeing that small icons of Zoom, right? Is it fully clear? Is the screen fully visible here?
Yes, sir.
Okay. So let me know when I can start.
So you can start, sir.
Okay, so very warm welcome and good morning to all the participants who are present over here. And I take this moment to thank AIIMS Raipur for giving me an opportunity, right, where allowing a metallurgist by practice to speak and share some of his experience in front of doctors and clinical professionals. So for next 20-30 minutes, I will make you all to go through some metallurgical aspects that you will forget yourself as a doctors and see yourself like a metallurgist. So the topic that I have chosen is on how nanostructuring of materials is going to help the medical domain, particularly in the context of implants, is what is going to be the topic for the current session. And I am currently working as an assistant professor in the Department of Mechanical Engineering at IIT Bhilai. And currently, I am also an adjunct faculty in the Department of Material Science and Metallurgical Engineering here. And prior to joining IIT Bhilai, I was working as a postdoctoral researcher at IIT Madras, where I did my master's and PhD. And during this entire journey of my career as a researcher, I have been working on several materials, especially lightweight alloys such as aluminium, magnesium, and titanium. And out of these three, you will easily recollect and relate materials such as magnesium and titanium, which are very common materials used in the field of biomedical engineering. And today I am going to share some of my past experience on these materials and how we try to tailor the material's performance to improve its applicability in biomedical domain. So when we talk about this nanostructuring, typically nanostructuring is given, the name nanostructuring is given to those materials where the structure of the material falls within this nano regime in one form or the other. And according to the way where we are doing this nanostructuring, these nanostructured materials are broadly classified under different major domains. Just a minute. The first one is about bulk nanostructuring. This is where we take a bulk material and whose microstructure is tailored and refined to the nano regime. And today's focus will be on this bulk nanostructuring and how it is going to help us. Other than that, you also have certain materials where we do not disturb the material's bulk property or bulk microstructure. Rather, we only perform this nanostructuring on the surface. So these are called as surface nanostructured materials. And these two are in terms of refining the material's microstructure. But other than that, we also have certain other forms of nanostructuring. One is actually creating textures, creating patterns on the surface where the patterns are in the scale of nano level. And the other one is generating, different type of forms onto the material. For example, material which is having lattice structures, and these lattice structures are having units in nanometer range. So these are the different type of nanostructuring. And today we will focus on the bulk nanostructuring process. And before we go on to it, we need to know that what is bulk nanostructured material. And I'm going to take an example from the medical implant. We are all very much aware, and some of us also might have undergone, right, this implantation, which we do it on dental domain. So this implant material is primarily titanium alloy. And we normally go with a special class of titanium alloy, which is more biocompatible. And what we know from the external perspective is the form and structure of this alloy. But if you look into the microstructure, a typical material will have grain structures, which we call it, these hexagonal patterns, we call it as grain structure of the material. And these grain structures will typically be in the order of few tens and hundreds of micrometer. And this is some kind of a grain structured material, and this is the microstructure for a commercially pure titanium. And certain grades of titanium will also have a similar kind of a microstructure, but it will have some fine lamella. These are multi-phase materials. But here also you will see the grain structure is typically in the order of tens and hundreds of micrometers. But now, what is nanostructured material? Moving the material's microstructure, which is typically in the micro regime, to the regime which is far lesser than micrometer. As what you see over here on the right side of the slide, the grain structures is refined from about 20 to 30 micrometer to about 100 to 200 nanometers, right? So here the grain structure is very, very fine that you need special equipments to see. A typical conventional materials microstructure can be observed even under an optical microscope, and to certain extent, certain materials, the microstructure can even be seen by naked eye, provided the grain structure is far higher than the micrometer. But when it comes to these nanostructured materials, we need advanced electron microscopies. We may require scanning electron microscope or transmission electron microscope in order to understand and see and quantify the microstructure of nanostructured materials. Yeah. What is the benefit that we will get? So in this particular graph, we have plotted the strength of the material, which we typically call it as flow stress of the material versus the strain, right? So how much the material can withstand the load typically when it is subjected to external strain. The red line indicates the maximum strength of a typical coarse-grained microstructure or coarse-grained material, whereas which is about 200 MPa, as you can see over here. The same material, just by refining the microstructure, making it in a nanomaterials form, will significantly improve the strength of the material. So the strength of the material has increased from 200 MPa to as high as about 1100 MPa. So this is 5 times the typical strength of the material. And this is the typical benefit that we are expecting. And that is the reason why we are more interested to look into nanostructured material and study how this nanostructured material is performing in different domains. We may take mechanical domain, or we may take, how it is interacting with the biological fluids, what is its biocompatibility, etc. So that is the major research focus that we currently work on. But now, if you ask me how such nanostructuring is improving the performance of these materials, so this strength contribution is attributed to a phenomena which is called as Hall-Petch effect. So where the material's grain boundaries will act as a barrier to the deforming feature. So the material at room temperature is typically, the deformation is aided through a microstructural feature which is called as dislocation. So these dislocations, how easy it is able to move decides what is going to be the strength of the material. In a typical material with a coarse grain microstructure, the grain boundaries which are acting as a barrier to these dislocations movement are few in numbers, and thereby the resistance offered by the material in general towards the movement of dislocation is also low, leading to a lesser strength of the material. But when the microstructure of the material is refined from micrometric regime to nanometric regime, we are generating more number of grains within a given area. So more number of grains, meaning more number of grain boundaries, right? So I'm talking about these boundaries, which are separating different grain structures. So here, this is a macro or a conventional material. And here you see this is the total number of grains and this is the number of grain boundaries that we have. And when we are moving down to nanometric regime and try to understand this scale, the scale over here is about 20 micrometer, whereas the scale over here is just 200 nanometer, which means for a area which is 100 times smaller, I have almost same number of grain boundaries. Thus, the resistance offered by these grain boundaries is significantly higher. Thereby, the strength of the material is increased multifold. And here there is one catch, that this Hall patch effect, cannot be experienced, or we cannot take benefit out of this Hall patch effect for any number of given grain size, right? So that is a limitation up to which we will get benefited from the Hall patch relationship, which is typically in the order of about 10 to 50 nanometer. So we can achieve extreme strength by nanostructuring the material, by refining the grain size down to about 50 nanometer, beyond which we don't get strength, rather we start reducing, getting our strength decreased, and which is governed by another relationship called as inverse Hall patch relationship, where the material can no longer deform through these dislocations and it start deforming by sliding of one grain over the other. So that is not the matter of our subject here. The next one is, how do we fabricate these bulk nanostructured materials? Primarily, there are different processes which are clubbed into two major domains. First one is called as a bottom-up approach. The approach is very intuitive, where we know that our material should have grains which are in the order of nanometric range, right? So here the manufacturing strategy is to develop the material which is built atom by atom, right? So we can have precise control of what is going to be the grain size, and accordingly we can build the material from atomic level till the required bulk level. And there are different techniques, predominantly deposition techniques, which are either physical based, or it is electron based, it is magnetic based deposition techniques. But in these techniques, in this bottom up approach, we have several disadvantages associated with it. What is that? First one, since it is an atom by atom building up, right, where we are building the material from lower level to higher level, we end up getting a large amount of porosity onto the material. And porosity is always detrimental to the material's mechanical performance. And the second one, as we are building the material atom by atom, layer by layer, we cannot develop very large sized material. So it is limited only to few micron level components. So these are the drawbacks when we go on to a bottom up approach. And on the other side, there is something called as a top to bottom approach. Here we have bulk material, and without modifying the entire form of the material, we will be able to break down the microstructure of the material. So this is the process of top to bottom approach. And what are the advantages? Here we are working on the material which is in its bulk form, right? I can take a cube of say 1 meter cube, and the size and shape of the cube remains the same, but the microstructure is refined from the conventional micron sized features down to nano sized feature, right? So there is no change in the shape of the material, and the material in general is bulk in nature without having porosity. And even if the material in its initial form is having certain porosity, those porosities are also eliminated during this top to bottom approach. And the second one, as we are working on the material in its bulk form, it can be processed to meet any requirement, starting from the micron level devices up to any macro level conventional products that we wanted to develop, right? So there is no size limitation, and The second one, we are not having any drawback coming from the porosity generation, which was one of the major drawback in the bottom-up approach. Now, what are the ways by which we can go for this top to bottom approach? The techniques that we employ in this top to bottom approach are broadly classified as severe plastic deformation process. Now, how this severe plastic deformation process helps in refining the microstructure? The severe plastic deformation processes are mechanical techniques, rather thermomechanical techniques, where the material is subjected to large amount of plastic strain. No, it is more similar. If I have to just help you visualize what exactly this process, it is more analogous to the dough making process which we employ for making, a chapati. So where we keep on, giving deformation onto the dough, right? So the objective is to make the dough more and more softer, right? So in the process where we make the dough softer, the same kind of a process is applied onto structural materials, primarily metals and alloys, where we are deforming the material, and with more and more deformation given onto the material is actually getting stronger and stronger. And the metallurgical principle goes something like this. The material which is having initial coarse grain structure with increasing amount of strain, which means by giving more and more plastic deformation onto this material, it goes through a series of steps. First one, it's called as accumulation of dislocations. As I said earlier, the plastic deformation in the material happens through these dislocations, right? And in this SPD technique, as we are giving plastic deformation onto these materials, the dislocations will start getting mobilized, and these mobile dislocations will encounter certain restrictions which are present in the material. For example, over here, the grain boundaries of the coarse grains, right? And it starts getting accumulated with increasing amount of strain. And then there will be a stage reached where this dislocation will entirely strain the entire material, right? So you will no longer have any room for further generation of dislocation, or mobility of this dislocation is completely arrested. It is at this particular stage where these dislocations will start entangling and form certain features which we call it as cell structures or subgrain structures, right? And in order to get converted to these cell structures, the material also requires some amount of energy. So because of that reason, many severe plastic deformation processes are conducted at a relatively higher temperature, right? Because this thermal energy will help the material to gain these cell structures. And now the combination of additional strain along with high temperature, which is given in the SPD process, will convert the cell structures. These cell structures are certain small entities which have boundaries, which we call it as low angle boundaries, right? So the additional strain along with the high temperature will lead to a process which is technically called as recrystallization. And because of this recrystallization, these cell structures gets transformed to high angle grain boundaries defined structure, which will be nano grains. So this will be the end result where the same material with the same form will have a fine grained microstructure, right? So this is the entire principle behind how grain refinement is affected in nanostructured material. And I will just quickly go through some of the well-known SPD processes. The first one and the most commonly utilized process is called as equal channel angular pressing. So here you just pass on a material to an angular die. So in this particular point where the material has to change its direction from verticality to some angle is where the strain is happening. The dough making process is actually happening in this region, right? So now this process is repeated n number of times just to add more and more strain onto the material. And as a net result, you see the material's performance, significantly increases, not only in terms of strength, but also in terms of ductility. The product of strength and ductility is what we call as toughness of the material, which is very much important, right? A material with less toughness will fail abruptly in a brittle fashion, which is not desired, no matter what is the domain of application. And the next process in this route is high pressure torsion, where the material is subjected to a constricted rotation, right? So where this black color portion on my left side is the material coupon, which is subjected to a very high pressure, and along with this high pressure, it is also given a torsional load, right? And because of this combined pressure and torsion, the material's microstructure is refined. This is a very interesting process. In the beginning, I was talking about two different types of nanostructuring. One is a bulk nanostructured process, and the other one is a surface nanostructuring. This method helps us in generating surface nanostructuring. And again, the principle of this process is same as that of SPD process. So that is why I have just put up over here. Here, a rotating tool, right, with a shoulder and a pin is inserted onto the material of our interest. And this rotating tool is allowed to traverse over the material. So this stirring action on the surface leads to grain refinement, making the surface much stronger than the interior by formation of this nanostructured microstructure. And the last one maybe is accumulative roll bonding. This is more like making chapati, right? So we roll the material, and in this case, the rolled material is cut, stacked, and again rolled. And this process is repeated n number of times in order to generate refined microstructure into the material. So now that we have talked about the metallurgical side, how we are generating the nanostructured material, and we also talked about the mechanical side, how the nanostructuring, just refining the material's grain structure, is helping improve the strength and ductility of the material. We will also see how it is benefiting the biomedical field experts, or what a biomedical expert can expect from such nanostructuring. So this titanium and its alloy is a very commonly known name among the biomedical field professionals. But if you see, the titanium is most commonly used only in unalloyed form, because the unalloyed titanium, the commercially pure titanium, is known to have bioactive property, right? And it has a very good biocompatibility. The commercially available titanium alloy, which we call it as titanium 6 aluminum, 6% aluminum and 5% vanadium, is called as grade 5 titanium, right? It is a commercially viable titanium with very good strength and ductility, but it is not very much biosuitable, because upon release of this titanium alloy, when it is biodegrading, right, the titanium ion is okay, which is compatible with the body, but the vanadium and aluminum ions are detrimental for acceptance of our bodily fluid, right? So the grade 5, which is having better strength, is not practically viable for use in biomedical application, and the commercially pure titanium is not having very good strength, so it is not used in orthopedic application. It is primarily limited to dental and similar other application where the strength is not of major concern, right? And whatever I said, this is the technical data, right? So the strength of commercially pure titanium is just about 170 MPa, whereas the strength of grade 5 titanium is 795 MPa. Now, on the same hand, the commercially pure titanium, which is subjected to severe plastic deformation to form ultra-fine grain microstructure, which is reported here as UFG titanium grade 2, right, is having 796 MPa, which is close to your grade 5 material, right? So now, without the need for adding any detrimental alloying elements like vanadium, the commercially pure titanium is raised, the performance is raised to meet the requirement, right? Thus, this helps and this becomes a suitable addition, right, to replace various, high cost, heavy material in place of, materials such as, stainless steel and various other, biocompatible orthopedic material, right? So this is one example. The other example is actually magnesium alloy. I'm not aware much about the usage of magnesium alloy in the biomedical application in India, but I could see through various literature that there are a lot of studies which are currently going on, and also in few cases they are using magnesium in biomedical application, primarily because it is, it is having the property to get dissolved in the body, right, within a period of time. And also there is now extensive research going on to control such, assimilation into the body, because the biodegradability of magnesium is much faster than it is required to be disintegrated as per the requirement. Now, that is one problem. But another problem associated with the magnesium alloy is its problem of yield asymmetry. The material have different property when it is loaded in tensile direction and it is loaded in compression direction. For example, this black graph is the material's mechanical response when it is loaded in tensile direction. And the same material, when it is loaded in compression direction, you see this is the material's response. And this square, which we have marked over the curve, is actually the indication of the yield point of the material. And this yield point is different for this material in different loading condition, which is not a favorable performance of the material. The objective is to have a magnesium alloy which will not only have better performance, but at the same time will also not have this yield asymmetry, right? So where nanostructuring of this magnesium is found to solve both the problems, both the strength problem as well as this asymmetry problem. And that is what we have seen over here. The 3 and 4 are tensile and compression tests of same material, but which are subjected to cryo-rolling, a process which is again falling under SPD category, to refine the material's microstructure, right? So this is the refined microstructure, and you could see that in both the condition, the yield point is almost same, right? Eliminating the yield asymmetric problem. So with that, I will conclude with whatever I wanted to present. So a very quick overview of what we do at IIT Bhilai in our research group. So we work on certain materials, and currently we are working on certain materials for bulk material for its usage in biomedical application. For that matter, we are working on two newer class of materials. One is called as high entropy alloys, and the other one is on titanium intermetallic class of alloys, right? This is more like a metallic glass kind of a property. It will have very high strength, but at the same time it will have very low ductility. So our group is working on improving its performance. And then we also work on microstructural engineering, that the things that I was discussing in this lecture, right? So we work on magnesium alloys, where we are trying to enhance its biodegradability. That is one area where we are working. And the other one is to develop titanium components through additive manufacturing, right? So there is a lot of trust going on in this area, right? So where we can develop the implant as per the requirement on case-to-case basis directly using this additive manufacturing, right? Preventing all the complex requirement of machining and grinding, and also to prevent unwanted thermal stresses which are built onto the material because of this conventional manufacturing process. And then lastly, we also are working on advanced coating for improving the biomedical application. So one example that we are working on is to develop, I don't know the technical term, but to have silicon nitride coating on magnesium alloy to improve, its interaction with the bone and the tissue, right, for superior tissue growth onto the surface. So that is also one area that we are working at IIT Bhilai, right? And these are some of our technical expertise which we use to solve the problems, right? And with that, I will conclude my session, and I will eagerly look forward for any discussion, to take this up in any further additional forum or for any joint collaboration work. So you can always contact me either by email or through WhatsApp. Thank you.
Thank you, Dr. Emmanuel. I must admit that most doctors are very poor in understanding physics and mechanics, but you have done your best to explain so many things very lucidly. And it was nice to hear you comparing nanostructuring with the dough we prepare for chapatis and rolling of chapatis. That was a nice comparison indeed. A little some things which maybe we have missed is, you said that dental implants, definitely titanium for dental implants, but not for orthopedics. And what exactly, I mean, would nanostructure titanium be more applicable over here?
So if we look into the titanium especially, right, so there are different grades of titanium which are available in the market. Okay, so starting from the commercially pure titanium till very high strength titanium, and the commercially pure titanium is suitable for biomedical application because titanium has a very good biocompatibility. And the commercially available high strength titanium has several alloying elements added to it, like aluminum, vanadium. So because this aluminum and vanadium are the two major alloying elements which are added to stabilize certain microstructural feature of these materials. But the material which is alloyed with these elements, especially the one with vanadium, these are not, effectively a biomedical material. Okay, people still claim that grade 5 titanium can also be used, but especially this vanadium is not a biologically friendly material. So that is the reason why we cannot use a grade 5 material as a bio implant. And if at all we are using, then we may have to frequently change it. Right now, the reason why people have to alloy these things is primarily to improve the strength of the titanium. Yes. And now the alternate way is not to add any alloying element, but only to strengthen the material in its commercially pure form by just modifying the microstructure.
Without any addition of any other alloy.
Alloying elements.
Right, right. And what about the magnesium alloy? Do we have any nanostructured magnesium alloy presently, clinical applications for that?
Presently, I could say in our group and especially in India, it is only in research stage. And in our group, we are actually having a tie-up with one hospital in Chennai, where we are currently taking it up to a clinical trial. Right? I think I don't spell it correctly. Maxillofacial surgery, something like that.
Okay, maxillofacial surgery, right?
Yes.
Thank you.
So our objective is actually to use this nanostructured magnesium, because not only the mechanical properties but also the we call it biological corrosion. The rate of biological corrosion also can be tailored by modifying this microstructure, right? So to meet our requirement, whether the material should be absorbed within 6 months or it should take at least 1 year to absorb, that rate is something which we have to tailor when it comes to this biodegradable implant.
Right. That is true. That is true. Thank you so much for your very lucid lecture. Thank you.
Thank you.
Any question from the audience? Maybe further questions could be added to the chat box and we could proceed with the next lecture.
So we'll have our next lecture by Dr. Damanpreet Singh, who is a principal scientist in pharmacology at CSIR IHBT Palampur. And he'll be speaking on the topic zebrafish as a potential model in nanoscience and technology.
Here my slides.
Yes, sir, we are visible. Kindly share your screen.
The presentation is visible to all of you.
Yes, sir, we can see the screen, sir.
You can full screen now. Is it fine?
Yes, sir, please put on slideshow mode. Yeah.
Okay. So first of all, good afternoon to all of you. And firstly, I like to thank the organizer for inviting me for this wonderful event. And chairperson, Dr. Mahapatra, Dr. Agarwal, and Dr. Sandeep. Thank you for chairing this session. And as far as my topic is concerned, it is zebrafish as a potential model in nanoscience and technology. So before I start with my presentation, I just want to give a snapshot of our institute. So this is the beautiful look of our institute. You can see the front and our laboratory, and the third photograph is for the thing this place is famous for, that is tea, the green tea. So now coming to my topic, that is the use of zebrafish. I think we all are talking about therapeutics, diagnostics. And if we see the situation of nanoscience in India, so the nano mission, it was initiated by Government of India and the Department of Science and Technology in 2007. And now it is 2023. And we have around 17 years. We have started this nanoscience mission. And it's one of the main aim was to have therapeutics and diagnostics, because this nanotechnology play a great role in biomedical research. That's why it was one of the aim. But the problem is that the moment we have started with the nanoscience for this biomedical application until now, we have very less outcome out of this. The thing is, whether we don't have any better tool, we don't have much expertise in this field. But if we see on the PubMed, if we just add this word nanoscience with India, there are around 9,300 articles that are present. And it's obvious if it is NCBI, those all 9,000 plus articles, these are related to medical or biomedical sciences. But still the outcome is very less. And if I see from my point of view, I think that the main problem that why there is less outcome, it is the model that we don't have that are appropriate. And if we say in biomedical sciences for therapeutic application, the path is from preclinical to clinical. We mean we cannot bypass this preclinical phase. We cannot directly go into the clinical part. My pain is better expressed, already expressed by Dr. Ravi Kant, where he has clearly mentioned that there were some ethical issues. And that is, these ethical issues, these are not only with the clinical, these are also with the preclinical. Like our lab, we are much more focused with the preclinical, and we know what are the problems that we face when we have to start the preclinical study. And secondly, the investment that is required. I think we all are working in academic research, because I have seen majority of participants, they are from the academic research. And in academics, finance is a big issue. Means all these concerns say that we need a better models that are having that require less finance and give a better view to proceed further. If we see from morning to till now, we have seen a lot of biomedical preparation that are this nano preparation that are being tested. And what we have used in vitro models. And if we say in vitro models are really different from the clinical perspective, we cannot move from directly in vitro to clinical. We need this preclinical phase. And Now we are much more focused toward rat and mice and rabbits and dogs and cats, but still there are a lot of ethics with this CPCSEA that is involved. So I'll start my presentation. So my slides are moving.
Yes, sir.
Okay. So these are the contents of my presentation. It is the nanopharmaceuticals in India, status of animal use in biomedical research, importance and need of alternatives to animals, zebrafish introduction, application in nanoscience with our own examples, and little bit about how we can keep this zebrafish in our lab so that we can run a good research, and what are the troubleshoots and what are the limitations. So if we see about the nanomedicine, nanoformulation, this term, it may not be new to us, but still, if we say from Indian prospect, this term, it is new. It was this nanomedicine, it was the guideline that was framed in 2009 itself by Honorable Minister of Health, Dr. Harshvardhan, and it was approved in October 2019. We mean around we say around 4 to 5 years back. And that is the reason we don't have much more drugs or therapeutics or diagnostics that are developed in India, because we are not, we haven't initiated this part from long. We are just 4 to 5 years old in this case. And according to this, the drug that we have, they are the new chemical entity. But if we say about the nanomedicine, these are not the new drug as it is, but still CGI defined that all nano pharmaceutical preparation will be treated as investigational new drug permanently. It means they start with IND. But to complete this IND, we have to go for several preclinical studies to make that it is safe and it is efficacious. So if we say about the whole drug discovery and development part, we have target selection and validation, we have discovery, and we have development. And the model that I am going to discuss, it play a crucial role in all these steps. I will just explain this step by step. So if we say what are the importance of animals in biomedical research, or we say in preclinical research, they have the same organ, they have the same body as that of humans. They are susceptible to same diseases, many of cases. They have a short lifespan. It means I have to study the whole aging process. I need not to wait for 60, 70 years. The whole process can be done within short span, 2 years, 4 years, 5 years, or 10 years, because of the short lifespan of the animals. Environmentally, we can easily control. Maybe in a single room, I can maintain the condition of Europe. I can make the condition of hard desert. I can make the condition of any place by easily controlling the room conditions. And secondly, and another thing, that is the ethical. We cannot do each and every study in humans directly. We have to ensure the safety in the small living organism. And secondly, they have the similar biology as that of human, and in many cases, they are genetically similar to humans. In some cases, it is around 99%. So these are the major domains that need animals. We need animals, particularly in context of biomedical sciences, in disease pathogenesis, drug development, either it's preclinical testing through efficacy, safety, or it is a formulation design, particularly related to nanomedicine. And it is a source of pharmaceuticals, whether it is antibody, antivenom, hormone production, and raw material. And last, the more important, it is toward education. We have to make our second line ready to initiate this or to take the biomedical or the medical sciences forward. And this is the current status of the use of animal in biomedical research. So if we say around 65% of animals, all we use, these are the mouse, followed by rats, and then birds, and then fish, and there are some other rabbits, dogs, and cats. To primates we also use. And this is the number of animals that are used globally. And if we say China, it tops the list. But this is the only information that is available on public domain. There are a lot of studies that are being done or that have been performed, and they are not in the public domain. They are just a summary of theses or some reports. And if we say in this list, India comes around at 16th number. So these are the major concerns. Suppose I am sitting in a research institute and you are sitting in a medical institute. This concern, it affects the UGC banned the dissection in all the colleges. We cannot only show the next generation that this is the lung and this is the liver, this is how it look like and this is how it works. We need a live demonstration. These are just the top line headings of the topmost press reports. The government banned the use of live animals for education and research. This is for the academics. And if we say about the industry, it also gets suffered with these stringent guidelines, India to end household product test on animals. Government ban use of animals in tests for soaps and other such preparation. This won't say that these are safe, but the thing is, we cannot use animal just to check the safety of those things whose toxicity affect us not much more. So there are some principles which has been in place that just restrict the use of animals. That is called as 3R principle. The one is the reduction, then refinement and replacement. Reduction, we can reduce, but we cannot totally abolish the use of animals. I am using 6 per group. I can use 5, but I cannot use 1 per group. We can refine the protocol. If my protocol required 2 control, I can stick to 1, but still I cannot refine to that extent that we cannot use any number of animals. This is replacement. It's better that we can use an alternative to animals or to have some other computer stimulation. So next 2 R's that have been added, that is rehabilitation and reuse. We cannot rehabilitate the small animals and we cannot reuse the small animal like mouse. So we have to sacrifice research. We can replace, but replace with what? We have the 4 things as a replacement to animal. Either we can use computer assisted methods like computational models, or we can use in vitro cell culture, or we can use human disease structure. But still in vitro, it tells me what is happening at the target site, but it won't tell me much about how the drug is getting into that target site. It means I have to go for kinetics also. And for the human disease, apart from having the ethics committee from animal, IEC, I have to get the additional approval from IEC. It automatically increases the time of research. It also become difficult to get such approvals. So in academic research, we say we have the only option is to use a less sentient animal. It means they are less socially concerned, they feel less pain. And what is the list of these alternative to animals? So this CPCSEA, now it has been named as CCSEA, is the organization that restrict or that control the use of experiments on animals. And according to this, these 5 categories, these are considered as an alternative to animals. My just interest to tell about these alternative at this platform is that we have this option as a living organism in which we can test our nano formulation, nano diagnostic as an alternative. So in this list, zebrafish was the top, followed by C. elegans, Drosophila. There are some tunicates. Apart from this, there are some starfish that can be considered as an alternative to animals. So these guidelines, this communication, it came in 2019. But still the guidelines were missing. But in 2021, the CPCSEA come out with the guidelines on experiments on fish in 2021. And after that, a lot of work has been going on this fish, and particularly it has played a great role in nanomedicine. If we say for nano delivery, nano diagnostics, for kinetics and everything, this fish, it is being used. And if we see the status of this fish compared to other organisms, it is going to get increased day by day. If you search it out on PubMed, earlier in India, 5 to 6 years back, we were having only 2-3 labs, but now we have more than 100 labs. They are working on the use of this zebrafish for this biomedical purposes, and to some extent, each and every lab, they have used this model for nanoscience also. And if we see the usage of fish, it has been increased manifold since it comes in 2004 until 2013. And if we say by bulk, this model, it was used by firstly by University of Oregon in 1980s. And after that, establishing this model, now its use has been increased multifold. And these are some of the important things that make this model an ideal for drug discovery part as it is a vertebrate developmental model. It has all the senses like taste, touch, balance, and vision. It has fast development and is simple to maintain. So these are the stages of fish. I can say just within a span of few hours, this model is ready to be get used as an in vivo model compared to other rodent models. So you can see all the stages from 0 to 30 minutes. It is a single stage. If I want to have some knockout, I have to make some disease model. I can easily silence any gene. I can activate anything that I want to do. And you can see at 4 to 5 hours, the cell division starts, and at 15 to 18 hours, majority of the vital organs, they are ready. You can see the beating heart at this stage. So these are some of the other important things that make this model ideal for drug discovery, and particularly it is a transparent that allow the clear movement of the thing that I am interested. And apart from this, it is easy to go for in situ immunohistochemistry. I can easily bind any antibody to any protein that I want to see its expression. It show very fast embryogenesis, and direct mutagenesis is very easy in this model. So you can see just the larva, it is at 24 hours. So you can see all the vital organs, these are present. It means if I want to direct any of my nano diagnostic or therapeutic to any organ, I can do it in 24-hour larva as a in vivo model. It has eye, brain, heart, swim bladder, muscle, everything. And now coming to what exactly zebrafish it is. It is Denio belonging to family Cyprinidae. It has distinct horizontal strips that extend across the length of its entire body. It's originated from the Western India. It inhabits at the freshwater, streams, ponds, and canals. Its size is around 6 centimeters. It can live for 5 years in lab, but in wild it is around 3 years, and it is ideal for keeping in aquarium. My aim is just to show you this species, that what is the correct variety that is of our interest, because majority of the lab that I have visited, they have some other varieties and they are using that as a zebrafish. They are testing their drug, they are testing their disease models in the other varieties. The problem that come at the end of the day, that the information related to their antibodies or the commercially available antibodies or the primers that are available, NCPI, these are related to the wild type. But people are using some other variety. That is the reason they won't get the exact changes or exact pathogenesis for which they are more interested. So these are some of the wrong variety. The last one, that SF, it is a short fin zebrafish variety that is of our use, that we want to use for biomedical research. And this is another mutant that is long fin. Again, it is a wild type, but it has one mutation that after attaining the maturity, it get opened, and still it is considered a wild type. So these are some of the other different genus of fish that is sometimes considered as a zebrafish, but it is totally wrong. Our interest is this short fin. So this is the more identification of this fish. It has 4 to 5 blue horizontal strips as shown in the first figure, and the color should be intense. And this is the differentiation between male and female. The male, it looked like a little bit pinkish with a yellowish with cylindrical body, but compared to female, it has some belly shaped abdomen, obviously because of presence of eggs, and its color is little bit bluish and silver as compared to male. It shows some yellowish, a little bit golden type appearance. So it is important to have it to differentiate between male and female. So this is the importance of zebrafish in nanoscience. Why we want to use this model for this nanoscience? I think for whole biomedical sciences, this model, it has a great importance because the cost is very less. The division is very fast. The number of organisms that I get at the end of the day is very high. Second, the ethics that is associated with the use of this animal, this zebrafish, it is very less compared to the rodents. So in particularly with the nanoscience, it has ability that we can screen large number of nanoformulation quickly. Like we say nano. So it's obvious that we use some polymer, we use some nanomaterial, And we all know that the cost of all these nanoparticles or nano raw material, it is very high. So if I have to make 10 molecules or 10 different variants of this nano formulation, it's not either practically possible or not it is financially viable that I can test each and every in the rat model. No doubt I have checked in vitro, but still in vitro is practically have a lot of difference when we go in vivo. It is showing good response in vitro, but in vivo it gets failed. That is the reason that in India we don't have much more successful success in case of nanomedicine. We all are working, but at the end of the day, the success is very less because we don't follow the good screening path. We just screen using in vitro, and in vitro, sometimes it shows toxicity and sometimes it is ineffective. So to link in vitro and in vivo, we need something that can be used as a good screening tool. That means I have made the 10 prototypes, I can screen that 10 prototype in vivo. And then I select the best out of this, and then I can check it in higher animal model, maybe rat, rodent, and other dog and cats. So it is a good screening tool. In vitro and in vivo. It is difficult to maintain in vitro lab. And for in vivo, we all know we need a well-furnished animal house. But it is, compared to both of this, it is highly economical. We just need an aquarium. And some condition that I'll explain in my further slide that are required to make this fish surviving. It is high growth rate. A single fish, it gives number of eggs. If I say single 200 of organism ready by 10 days by a single fish. You can go for breeding again and again after 10 days. It has characterized developmental stages 0.5 to 5 millimeter. Developmental stages I have already shown. The interest of showing this size is that we can use simply stereo microscope to see its developmental stages. The cost of investment is very less. Genetical homology to human, 70% it has similarity to that of humans. It has X. All the organs, they are ready. It has easy availability of transgenic lines. I can get transgenic of each and every disease that I want. Because the major problem when we go with the chemically induced model, genetically they are dissimilar to the human. If I am saying Alzheimer, I induce Alzheimer using beta amyloid plaques in rodent. But here we can make the genetically modified fish with having developed Alzheimer at the birth itself. It's optical transparency. It remains transparent till day 5, but further if I want to make it transparent, I can use simply 1-phenyl-2-thiourea, simply called as PTU. It inhibits the melanogenesis. Apart from this, it exhibits all the physiological parameters like blood-brain barrier, cardiovascular system, immune system, and everything. So I can check everything related to these organs. So these are the model for preclinical investigation in nanomedicine. So fish, we can use it for toxicology, we can use it for pharmacokinetics, we use it for therapeutic efficacy, with several advantages using having external and rapid development, short reproductive cycle, transparent, low cost, easy maintenance, and genetical modification is very easy. But thing that has to be taken into consideration while selecting this model, it is a development stage. Yeah, I want to use embryo, I want to use larva, I want to use adult, and the route of administration. Because we cannot, sometimes we need specialized instrumentation to go for specific route, like for IV. And secondly, physicochemical properties of the test molecule. Because in many of the cases, we give our test molecule in system water, and we want it should be water soluble. So this is one of the example where this model, it has been used for a liver cancer. It is just a type of diagnostic as well as therapy. So here the liver damage, it was induced, the liver cancer, it was induced in 2 days larva, in 2 days, after 48 hours. And you can see the GSH, these are the 2 probes that were used to test the efficacy. One was with the hypoxia and second was with GSH. And it was TBD, it was conjugated polyethylene glycerol encapsulated platinum that is showing the fluorescence. You can see the size of the larva, the size of the liver. So you can easily differentiate. So here the drug was at the nano formulation was administered IV, and it has shown good conjugation with the liver tumor cell. So this is a beauty that everything is in front of our eye. For rat, rabbit, and other models, we need a good high resolution instrumentation, but here the purpose is solved with simple stereo zoom microscope. So another playing a great role is in case of neuroscience, because in rats and adults, and I will say in case of in vitro, we can see what is affecting at the site in the brain cell. But for getting the drug into the brain, I have to use different models. I have to use a cell line to see its penetration to the blood-brain barrier. And for that, I need 2 or 3 cell lines. And then I have to see what is happening at the target side. I need a special cell where the disease has been induced. And the beauty of this model is that the blood-brain barrier in this fish, it is very much similar to that of human as well as a rodent. This means that whatever I am getting the results in screening part will be replicated in rodents, and probably will be the same as that I want to see in humans. So you can see the structure of blood-brain barrier. It has all the same endothelial cell, pericytes, and glial processes. So this example, it is of glioma, where a particularly, you can see in the second image, the glioma has been established in the brain. And here the test molecule was the vincristine nanoparticles, and you can see that it has been suppressed. So these are some of our own examples, as Dr. Parvar and Dr. Amitabh, my friends, they have already described that our institute is working to utilize a bioresource for human consumption. And for this, our major focus is on nutraceuticals, where we are focusing on dietary flavonoids. And our group is working on epilepsy. We tried to screen all the major dietary flavonoids in fish model. So we have selected apigenin, hesperidin, naringenin, quercetin, rutin with valproic acid as a reference standard. And when we tested all these flavonoids in fish model, we get some interesting results. One of this flavonoid, hesperidin, it was showing the highest response as compared to valproic acid. And apigenin was one of that, it was found to be ineffective. So it was quite interesting, and the study it has indicated us that we can use this flavonoid for this condition as an add-on therapy for epilepsy. We cannot go for the per se therapy because obviously we need drugs for the management of epilepsy. So we use this model. So it is just a simple stereo zoom microscope, a simple web camera, and you can see the video, I hope it is working. So you can see the movement of larva. It is just 7 days larva, and it is showing generalized seizure. It is similar to that of human beings. So it is rotating on its own axis. It's a whirlpool-like movement, and we mimic it with the initiation of seizures in the case of human. And here you can see the full-blown tonic and clonic seizures in second slide, the third figure, the video that is going on. So you can see the jerky movement. This jerky movement is because of abnormal contraction of the muscle layer that is present on its back of the body. So it gives a similarity with that of human, and I can use this as a screening model for this particular disease. Since it is not the topic of this, I am just explaining how we have screened them. So here we have then identified that hesperidin is interacting with some receptors and some genes, because of which it is showing some therapeutic response. The whole study was done in zebrafish. So then thereafter we have we just gone through the literature, and we see what are the plants and what are the bioresources that are available that contain these flavonoids that we have already tested. And what we found that in Himachal itself, HPMC is one of the company that utilizes a lot of citrus fruits, and they generate lot of peel in turn that is generally dumped as a waste. And we screen each and every peel of those industrially important citrus plants, and we found that among all these citrus peels, all the flavonoids that we have screened, they are present in enormous amount. And our interest was on hesperidin, because in our earlier study we found that hesperidin, it showed a very good response. So we found that the quinoa peel, that is Citrus reticulata, it contained lot of hesperidin, and it was a potential agent that can be used for the development of nutraceutical as an add-on therapy to anti-epileptic drugs, so that we can have withdrawal better. But the problem that the extract that we have extracted, it was not showing the promising results. So it was probably because this flavonoid, they show very poor solubility. So then we collaborated with our formulation group, and they come out that we can make a nanomycins out of this that will have the both philic and phobic design, so that they can go better penetration into the brain and have the therapeutic response. So we make a solid dispersion out of this, and we tested the efficacy in first fish model, and then we replicated it in rodent model. So this was the whole a whole part that how we develop this solid dispersion. We use extract and a Solu Plus as a carrier, and then we use mix it, and we then follow the freeze drying, and thereafter we made a lot of 4 to 5 prototypes, and then we screen all these prototypes, and we come out with a specific prototype having a good in vitro solubility, in vitro dissolution, and it was fulfilling the criteria of the particle size and all other requirement. So this first figure, it is this A that is showing the Solu Plus itself, that is having large size, uneven, irregular shape. Then the last C, it is the dispersion of our interest that show homogeneous distribution of extract. And the second, this B, it is the just physical mixture of these compounds. And the particle size, it was found to be around 78 nanometers. And at lower, the A, it is showing the FTIR, means there was no shift, there was no spectral shift, and the release, it was sustainable. And when we tested these both product in fish model, you can see SDN10, SDN100, the last one, it is showing the better response in fish model compared to CRE100, that is the extract itself. Means its activity has been increased double. And for this, we use the fish model itself. And another, we have made lipoic acid conjugate with the tocopherol polyethylene glycol TPGS, and it was just a chemical reaction. We have conjugated this, and we tested its safety and efficacy in fish model. So we have made this conjugated, then we characterized using NMR. I am not showing much result out of this. And again, there was no shift there was no spectral shift. And this was a size distribution, everything that we want to develop out of this. We have a good conjugate out of this. And this was a biocompatibility study. We treated larvae with our conjugate, and this was a developmental. It was showing no abnormality till 20 micromolar, and then we tested its safety in efficacy. In fish model, this was a whole survival rate, hatching rate, nothing was affected. This indicated that our nano formulation was safe, was showing no toxicity. And then we tested in fish model, you can see again the efficacy of this conjugate. It was better as compared to lipoic acid itself, even at the gene level, even at the behavior level. So now further we are going to patent this particular conjugate. So all these work, it showed that we can use this model for screening, we can use it for safety, we can use it for efficacy. Now the next question is how we can maintain this model. What are the requirements that can be fulfilled so that we can use this model for academic research? Frankly speaking, that using this rat model nowadays, it is becoming costly, as well as many of the ethics committee, they don't allow you to use n number of animals. And when we go to publish our result, the reviewer, they say that we won't accept this paper with this less number of animals. They want better statistical comparison with more number of animals. And in case of fish, we can easily use 12 to 15 to 20 even number per group. So these are the general requirements. If you can fulfill this with any mean, you can keep this fish in your lab. And I say that a single shelf with 4x2 feet is sufficient to maintain whole of this facility, and it will give you lot number of results. So if your first requirement is the temperature, 25 to 31. I think the place where we are sitting, it is little bit cold. It is cold throughout the year. We need a heater. Maybe, I don't know what is the exact temperature at Raipur. If it is between this, you can easily keep this fish. And if not, you can maintain it using air conditioning and for heater, water heater, that is available easily at the pet shop. Conductivity 400 to 800 ohm. It can be simply maintained using sea salt, again available commercially. pH 7 to 8.5, you can easily maintain. For we use bicarbonate to maintain. Light and dark cycle, it is little bit important. It is 4 hours to 10 hours. It is not 12 as that of rodent. 14 hours light and 10 hours it is dark. And feeding, you can use pellet feed that is available commercially, but we use Artemia because it is a wild fish. It loves to hunt and eat. So we use Artemia because we are working on CNS pharmacology, and its show behavior like it's original in the wild. If it is, it gets the wild condition. And the breeding, we use 2:1. Each and every lab, they have their own SOPs, but we use 2 male and 1 female. And anesthetic, we can use Trican, we can use simply cryoanesthesia. I have just added this word anesthetic because in animals, the anesthetics that are approved by CPCSC, these are not commercially available easily because they fall under Schedule X, but here anesthesia is very easy. So this is just a breeding setup. I have just shown that if you can maintain this setup, it is easy to breed. So you can see the tank, bottom image, it shows you that there are 2 tanks. One, it is with a solid base, and another that is fit into that solid based tank. It has grooved at the bottom. Grooved is because the egg that will be laid, they go into the bottom. If there is no grooving, the female will lay egg and male will eat that. And this green color, these are just to create the natural environment. And we make this setup ready on the day, on a prior day on which we want to have this egg. And I have used 2:1. So there are 4 female and 2 male on one side or other side. And next, and these are divided by a shutter.
Sir, may I request you to kindly wind up, please?
Sure. I'm just trying to show you the setup of this breeding tank. So this is the setup that we have in our lab. So now regarding its application or ethics or regulatory approvals, these are the guidelines that are approved by OECD. It is test guideline 236. It is used for the toxicity determination in fish. So just my aim is to show you that this fish, it has already been approved by OECD to have toxicity using this model. So, bas, thank you.
Thank you, sir. It was a wonderful presentation. I have a few question. Can
Yes.
we go for clinical trials directly after preclinical trials in zebrafish, or do we need to do animal studies in mice or rats?
So, ma'am, there is need. I have already explained that this can be used as a screening tool. We have to go for rodent studies as per guidelines to have both this rodent and non-rodent before going to clinical trial or before filing the IND.
Okay. Thank you, sir.
Okay.
Any questions? You can please drop your question in the chat box. Thank you.
Thank you.
Thank you, sir, for the lecture. Our next speaker is Dr. Vikram Patyal, Principal Scientist, Toxicology and Pathology at CSIR IHBT Palampur. and he'll be speaking on nanotechnology in drug development, safety and toxicity concerns.
So very good afternoon. Am I audible?
Yes, sir.
So very good afternoon to all of you, respected dignitaries on the dais, all the respected faculty members, for the audience and students. So first of all, I want to thank the organizers for inviting me to deliver a talk at this platform. So I'm sharing my slides quickly. So is my screen visible?
Yes, sir.
So are the slides visible and they are moving?
Yes, sir. Please start, sir.
Okay. So thank you very much. So my topic for the talk is nanotechnology in drug development, safety and toxicity concerns. So as earlier speakers also describe the genesis of nanotechnology. So the concept arises with the lecture by the Nobel laureate Richard Feynman that there is a plenty of room at the bottom. And this is a science, engineering and technology conducted at the nanoscale, which is about 1 to 100 nanometers. So nanomaterials, they have unique properties with vast range of applications ranging from basic material science to personal care applications. So these properties, they are because of their different compositions, then their size, their shape, and their surface properties. So if we talk about the nanomedicine, so there is a clear definition of the nanomedicines by the global organizations as well as in the India. So as per the US FDA, the nanomedicine are the products in the nanoscale range and at least one dimension in the size range of 1 to 100 nanometer. So that can exhibit different properties, whether it is physical or biological or chemical, as compared to their large scale counterparts. So in India, as the previous speaker have mentioned, we have guideline and there's a clear definition for the nanopharmaceuticals. And these nanopharmaceuticals, they are the preparation containing the nanomaterials, which are intended for either internal use or external application in case of human for the purpose of therapeutics, diagnostic, or health benefit. So size is between 1 to 100 nanometer, at least one dimension. However, in addition to this, if the material exhibit the different properties which are attributable to its dimension, which is beyond nanoscale range up to 1000 nanometer. So the guideline says that the material should also be considered as nanomaterial. And the pharmaceuticals which are developed using these materials, they are also considered as nanopharmaceuticals. So further, it was emphasized that it is important to mention the size of the developed nanomaterial on the product which is made out of it, and the nano size should be same in all the testing conditions during the claimed stability period and the final product. So this is a slide showing the representation of nanomaterial. So if we can see that the tip of pencil, which we can see with the naked eyes, it is about 10⁶ nanometer or around 1000 micrometer range. So we can see that with our naked eyes. However, the different cells in like fibroblasts, red blood cells, or bacterial cells, they are in the range of 10⁻³ to 10⁵ nanometers, and we use the optical microscope to to visualize them. So below that, it is the range which we are talking about, and the nanomaterials, and it is the about 1 to 10 to the power 2 nanometers in which these nanoparticles they comes. So so nanomedicine products, they are of different types based on their for the application they are used. For example, they can be lipid-based nanocarriers, either liposomes, solid lipid nanoparticles, or they may be polymers, polymer-based nanocarriers like polymer micelles or polymeric nanoparticles. They may be drug conjugates like antibody drug conjugates or polymer drug conjugates, polymer protein conjugates, or the inorganic nanoparticles like metal nanoparticles or silica nanoparticles or viral nanoparticles. So there are different type of products, and they have different applications. So the major applications for which the nanomedicine they are used, for example, one is the drug delivery. For example, the drugs having the problem with the their bioavailability, their stability. So in that, this nanotechnology is used. The targeted therapy, which is used in case of cancer to target the medicine for the specific cells or specific targets. So that is one application. Then gene delivery is the one of the application. Molecular imaging, one of the major application of the of these products, and then detection and diagnosis, in which this nanotechnology plays an important role, like various contrasting agent developed for the MRI. So, so these are the different applications which nanomedicines have. So if we talk about the current scenario of the nanomedicine in the market, so mostly in the market, the nanomedicines, they are for different indications like for cancer, for skin diseases, ocular diseases, infections, then cardiovascular diseases, immunological disorders, blood disorders, or nervous disorders. So if we see the developmental phase, around 33% of the nanomedicine which are in the developmental phase, they are in the phase 1 clinical trials, then 21% in the phase 2, and around 15% they are launched in the market. So if we see with regard to the indication or disease, so around 53% of the nanomedicine, they are basically developed for cancer, followed by the infection around 40%, and then the other disorders like blood disorders and the other indications. So in case of material, so like liposome or lipid-based nanoparticles, they are leading with the 33%, and followed by the antibody drug conjugate around 15%, and then polymer drug protein conjugates and the other materials. So this is the current market scenario as per the recent report. And if we see what are the different advantages of nanomedicine which are claimed over their other counterparts, so they are claimed to have the high bioavailability, and on the basis of that, they have improved efficacy in comparison to the parent drug, and overall they have better capacity to overcome the cellular and physiological barriers. They can easily cross the blood-brain barrier or mucosal barrier in the body. They are site-specific, and they are target-specific. They allow the imaging and diagnostic application at the cellular level or the molecular level. They have control release, low dose in comparison to their parent drug, and multiple drugs can be given at the same time. So these are the different advantages of nanomedicine which are claimed. And here are few examples of our group and our laboratory. We have performed some preclinical studies and developed the few nanoparticle for the to study the preclinical efficacy of those drugs. So we have developed the dendrimer conjugated podophylotoxin. As you know that the natural podophylotoxin is an anti-cancer drug, and it has a function as a antimitotic agent, and it inhibits the tubulin polymerization. So, but the major problem with the podophylotoxin is the toxicity and the its side effects or the adverse effect. So here our group, the chemists from our collaborations, they have developed the dendrimer conjugated podophylotoxin. So dendrimer, they are the polymeric molecules, and they are conjugated with the drug, and they are supposed to increase the stability and solubility of the drug. So further, these developed molecule is characterized by using the NMR, and then the size measurement studies were performed using the transmission electron microscopy and ELS, and the in vitro drug release was studied. And it was found that in vitro condition, they developed dendrimer conjugate, shown the better stability and sustained release. So further, we want to find out whether there is a difference in the toxicity of the parent molecule and the developed conjugated dendrimer. So we performed a study in the mice in which both the parent and the developed conjugate was given for 14 days at different doses, and we have found that the mortality pattern in the parent drug, it has induced some mortality in the mouse, whereas the dendrimer conjugate drug mice, they have survived throughout the period. So indicating some kind of reduced toxicity of the parent drug. The other parameters like liver and kidney injury markers and histopathology also confirm the safety of the drug in the developed dendrimer conjugate in comparison to the parent drug. Further, we want to study whether this dendrimer conjugation, it has efficacy also. So a study was performed in a skin carcinogenesis model in which in mice, the skin tumor were developed using the DMB TPA, which are the carcinogens. It was a 14-week study, and after 6 weeks after the development of these skin tumors, the drug was administered to the mice, and we have found that the number of papilloma which were developed on the skin, they were reduced by the drug administration, indicating the efficacy of this developed dendrimer conjugate. So further, the histopathology studies confirmed that this severity of the histopathological lesions in the tissue, those were lower, and the drug also induced the apoptosis in the tumor tissue, which is responsible for the efficacy of this drug. So this confirmed that the developed product also have the efficacy in producing some kind of tumor. So further, in another model, like model of hepatocellular carcinoma, the efficacy of this depoter was also studied, and we have found that it was very much effective in lowering the different inflammatory markers in the liver and different fibrogenic markers like TGF-beta or alpha-SMA in the IHC as well as their gene expression, they are significantly lower, and also improve the histopathology of the liver tissue. So we concluded that that it is effective in lowering the lowering the progression or reducing progression of the tumor. So another example of this is the dendrimer conjugated istamostin, which is also an anti-cancer drug, and istamostin phosphate is a marketed drug, and it has problem with its solubility and stability and bioavailability. So we have developed a dendrimer conjugate, and similarly we have characterized and performed the drug release studies where the dendrimer conjugated drug shown the stability and sustained release of the drug. So further, we want to compare the parent drug and the developed dendrimer conjugated drug. So toxicity studies were performed in the mice, and however, both the parent and developed drug both were found almost the non-significant increase in the injury markers were found. However, histopathological, there were not much difference. So in this efficacy studies, again, we have found very good result and improved efficacy with respect to reduced number of papillomas, reduced severity of the histopathological lesions, and the expression of the proliferating cell nuclear antigen and the induction of the apoptosis. So with these two examples, we have found that the nanoparticles, they have role in reducing the toxicity as well as the improving the efficacy of the drug. In another attempt, we have developed the plant-based cellular nanocrystals, and they were impregnated with the silver nanoparticles, and they were used for the wound healing studies. So they were well characterized using the different techniques. The antimicrobial studies were performed in the bacteria like Staph aureus and Pseudomonas aeruginosa, and we have found that these developed nanoparticles, they have very good antibacterial efficacy, and they don't damage the bacterial cell membrane and reduce the number of the bacteria. So further, the wound healing studies were performed in the mice, where wound is was created on the dorsal surface of mice, and then the developed formulation they were applied, and then at different stages the histopathology was conducted, and we have found that the developed formulations, they have better, they have reduced the inflammation and increased the epithelialization and the wound healing in case of mice. The inflammatory parameters and the collagen deposition parameters, they were also measured in the skin tissue. So these were the example which were showing that nano particles, they have a role in the development of the medicines which have efficacy, better efficacy as compared to their other counterparts. So if we see the internationally approved drugs, so as there are number of drugs, more than 50 in the market, like liposome nanoparticles for the respiratory distress, pancreatic cancer, then polymeric nanoparticles for prostate cancer, chronic renal disease, or the micellar for the various indications, then nanocrystals as a immunosuppressants or bone substitute, then inorganic nanoparticles for iron deficiency in case of chronic kidney disease. So, FDA have approved a number of medicines which are already in the market. But with the approval of the authorities, there are also safety and toxicity concerns which are related to the nanomedicine. So there are examples in which there was withdrawal of the various drugs from the market, like Cineram is one of the example which was used as a contrasting agent in case of MRI. However, after some time, it was found that it led to many side effects in the patients, so it was withdrawn from the market. Similarly, Bind-014 as a anti-cancer nanomedicine made from the docetaxel, and it was approved by the FDA after the small level clinical trials, but however, it was failed at higher level, so which also led to the closure of the company. So major concerns which are related to the nanomedicines, like during their development, more focus is on the drug delivery, but the toxicity and pharmacokinetic parameters, they are not well studied. And because of their size and their surface properties, which are unknown, there are an unexpected toxic effect due to increased reactivity. Hypersensitivity reactions are there. Then tissue accumulation and slow clearance in case of nanomedicine, it was observed. And the reactive oxygen species generation is one of the main mechanism for inducing the oxidative stress and various toxicity in case of the nanomedicine. And also the cost, because they are relatively expensive. So if we see the mechanism of nanotoxicity, so nanomedicines or nanoparticles, they induce the toxicity at various levels, like they induce the molecular toxicity, cellular toxicity, tissue toxicity, or immunological responses they can induce. So, like in case of molecular toxicity, as the nanoparticle they enter the body, it is surrounded by the various biomolecules, which lead to the protein corona formation. Now, this protein corona formation, this alter the size of the nanoparticle, and also alter the its surface properties, which is responsible for its untoward effect or unknown side effects. Similarly, the interaction of the nanoparticle with the various proteins, it also lead to the unfolding of proteins, so which alter their function. For example, in case of iron oxide nanoparticles, when it binds with the transferrin, it leads to the unfolding, so this alter the iron transportation in the body. Another, the protein aggregation were reported within administration of the nanoparticles, like for example, the amyloid, which is responsible for various pathological conditions. So at the cellular level, nanoparticles, they are known to induce the reactive oxygen species, lead to the lipid peroxidation, and lead to cell membrane damage of the cells, as well as the membrane damage of the organelle. So this lead to the cellular toxicity. Whereas at the tissue level, some tissue toxicity also reported, for example, in case of the carbon nanotubes. So when endotracheal installation were given with the carbon nanotubes, so it was found the deposition of these carbon nanotube in the lungs and leading to the lung inflammation. Similarly, intravenously given positively charged lipid nanoparticles, they are found to cause the lipid toxicity. As the nanoparticles they enter the body, so they are cleared by the macrophages. So they are as they are engulfed by the macrophages, so they also lead to activation of the complement system in the body, which lead to the unknown immunological responses. For example, in case of doxil and taxol, their nanopreparation, they are found to induce the various skin allergies or hypersensitivity reaction in the patient. So these are the different mechanisms through which nanotoxicity was induced, and as the size of the nanoparticle is very small, so they can directly damage the genetic material or induce the genotoxicity. So the available safety test in the current situation for the development of the drug, so they are basically used as per the conventional therapeutic agents. So because nanoparticles, they behave differently, they are small molecules. So some of the tests, they are not directly applicable to the these nanoparticles or nanomedicine. For example, if we talk about the AIIMS tests for mutagenicity, so some of the nanoparticles which they are found to show the toxicity in case of the mammalian cells. However, in case of AIIMS test, which is performed using the bacteria Salmonella typhimurium, so in which it was not able to induce the mutagenicity. So that shows the there is a need to develop the new test for such studies. The other example is the carbon nanotubes. So in case of carbon nanotubes, the conventional test we use for the cytotoxicity MTT assay. So these carbon nanotubes, they are found to interact with this dye, and so its interaction with the MTT increase the, you can say, intensity of the color development and show more toxicity. So this shows the use of this test, how this type of test, how much they are applicable for the nanoparticles. Again, the in vitro test used for the cytotoxicity study. So in vitro studies we are using the high concentration which are not applicable in vivo. So these we have to take care while perform the toxicity study. Purity of the sample is very important, because some of the carbon material or carbon nano tubes, they also contain the hazardous metals which are responsible for their toxicity. So purity is also important. So major challenges with the regulation of the nanomaterials or nanomedicines, as there are the pharmacokinetics of the nanomedicine, they are different from the conventional drugs. So they have increased capacity to enter the blood-brain barrier, and they cause the compromisation of the normal brain function. And the issue is that at the smaller scale and larger scale, there is a difference in the stability of the drugs. Possible environmental effect with the non-biodegradable materials. Then systemic accumulation of the nanoparticles which lead to organ toxicity. They are potential genotoxic, as they directly can damage the genetic material. And there's a lack of unified set of global regulations. So then variation in the nanomorphology result in the characterization issues, and the definition and classification of the nanomaterial and nanomedicine, it is different for different countries. Still, we do not have the a single guideline for which is defining what is nanomaterial and what is nanomedicine. So global strategies to nanomedicine regulation, there are different bodies, like in USA, there is a FDA who is regulating, but again, FDA is taking the, there's not, I can say, the proper guideline, and they are just case by case, they are regulating the nanomedicines. Similarly, in UK, it is MHRA, European Union, EMU, and then Canada and Japan, they are a different agency, but all they lack the proper guidance document for the regulation of the nanomedicines. So in India, we can say recently or in 2019, our government came with the guidelines for the evaluation of nanopharmaceuticals, and developed by the Department of Biotechnology with the Indian Council of Medical Research and Central Drug Standard Control Organization, CDSCO, with the aim to ensure the quality, safety, and efficacy of the nanopharmaceuticals, and to encourage the commercialization of nanotechnology-based interventions in India. So, as per these regulations, they have defined the different category of nanopharmaceuticals. One is nano carriers loaded with active pharmaceutical ingredients, or the API, they are directly converted into nanoforms. So they can be further divided into different categories, like category 1, 2, 3, 4, on the basis of that whether these nanocarriers or these API, they are known or not known. So on the basis of these categories, there are specific guidelines, what type of studies we have to do for their development before the clinical trials and for going to the clinical trials. So, however, these guidelines, they do not apply for the conventional drugs with the incidental presence of the nanoparticles. So the conventional drugs, we have incidental presence of the nanoparticles, or the drug products containing the microorganism proteins, which are naturally present in the nanoscale range. So these guidelines are not for those type of drugs, and also not applicable to the medical devices, in vitro diagnostics, or tissue engineered products. So As per these guidelines, there are guidelines for the proper characterization of the nanomaterials and different physico-chemical properties. Then the different data, animal pharmacology data and animal toxicology data, what type of data is needed for going to the clinical trials. So there is a clinical drug, new drug and clinical trial rule 2019, in which all the tests are mentioned, what type of test we require to develop the drug, to develop the nanomedicine. For example, in case of animal pharmacology, specific pharmacological action studies, then the safety pharmacology studies, pharmacokinetics, they are required. Similarly, in case of toxicology data, we need systemic toxicity studies, male fertility studies, female reproduction and developmental toxicity, local toxicity, allergenicity studies, genotoxicity and carcinogenicity as per the regulatory requirements. So these are the studies which need to be performed in the animal models. And if there are guidelines, there are not, there are some type of toxicity tests, they are not mentioned in these guidelines. So in that case, we need to follow the US FDA or ICH guidelines for the pharmaceuticals or OECD guidelines for the chemicals. So after that, we can enter to the clinical trials and different phases of clinical trial involving the safety and the efficacy studies in the clinical
Excuse
trials.
me, sir.
Yes, I'm binding, ma'am.
This is a.
So as we have seen that the nanomedicines, they have shown the promising role in improving the efficacy and the targeted drug delivery and reducing the side effects of the drug. And we have seen the rapid growth of the nanotechnology in the medicines and specifically in the area of cancer. So there are around 50 or more nanomedicine in the market. However, still the failure rate is much more high in case of nanomedicine. So the need of the hour is to develop the proper guidance and proper guidelines by the regulatory bodies, so that more and more products they come to, or nanomedicine they come to the market. So in the end, I thank you all for your patience and listening. Thank you.
Thank you, sir. It was a very interesting topic and was much needed also. Thank you for giving insight about all the regulations, particularly in India. If there is any question from the audience, they may leave it in the chat box. Thank you so much. Over to the next speaker.
Thank you, ma'am. Thank you.
Thank you, sir. I'd like to invite Professor Alok Agrawal, sir, Dean Academics, Professor and Head, Department of Orthopedics, for his next lecture on role of nanotechnology in orthopedics.
Full screen है. Respected chairpersons, august faculty members, delegates, friends and students. From the morning, you have been hearing about nanotechnology. And now we are going and entering into specific fields, that how it will be helpful in orthopedics and ENT, like that. So reminding you again, Richard Feynman was the first to conceptualize the potential of nanotechnology, 1959. He described it as a field in which little has been done, but in which an enormous amount can be done in principle. It was defined originally by the National Nanotechnology Initiative as the study and controlled manipulation of individual atoms and molecules of size between 1 and 100 nanometers, which have therapeutic applications at the subcellular level. Fundamentally, at the nanometer scale, nano-engineered materials have the theoretical ability to target and modify these processes. At nanoscopic size, They have unique and advantageous surface characters compared to their original size. High surface area to volume ratio is responsible for the infinite property achieved by the smaller size. Mechanical, optical, magnetic, and chemical characters of nanoparticles achieved due to its particle size, which enable them as an ideal choice for wide application in the field of science, electronics, defense, cosmetics, and medicine. Applying these principles to orthopedics, bone, when broken down to the nanoscale, is naturally a nanostructure composite of collagen and hydroxyapatite. Bone matrix is composed of elastic collagen fibers and ground substance. It is the calcium and phosphate in the form of hydroxyapatite proteins with type 1 collagen fibrils and water. Dimensions of the mineral and organic constituents are of the nanometer scale. Bone substitutes developed by nanotechnology have improved strength and longevity of implants. Technological advances made in implant manufacture have made possible the development and applications of biosensors, sensitive diagnostic systems, and controlled drug delivery systems. So that is the hierarchical organization of bone over different length scales, right from macro to nano. As you can see, bone has a strong calcified outer compact layer, that is figure A, which comprises of many cylindrical Haversian systems or osteons, that is figure B. The resident cells are coated in the forest of cell membrane receptors that respond to a specific binding site, that is figure C, and the well-defined nano architecture of surrounding extracellular matrix, that is the collagen molecules and hydroxyapatite. Coming to nanotechnology in orthopedics, the role is in all diseases, diagnostic, implantable, treatment, modalities, and materials. So you can see right on this tree from the left, if there is a cartilage injury, you can treat it with it. You can have a target drug delivery. You can have an early diagnosis and imaging. You can have infection prevention. You can go for fracture healing. You can go for nanostructured coatings for a better implant. You can repair the tendon and ligaments and regenerations. You can go for neuronal regeneration. And you can also go for fusion materials. So nanotechnology provides a multitude of new tools for application in orthopedics. Osseointegration of implant materials, repair and regeneration of meniscus, osteochondral defects, and vertebral disc are the important applications. It has an important role in targeted drug delivery in treatment of bone cancers. Application of nanomaterial in orthopedic surgery are related to improving implant longevity, treatment of osteoporotic vertebral fractures, control of infections, treatment of orthopedic oncology, orthopedic tissue engineering, and in stem cell regenerative medicine. Nanomaterials develop superior physical chemical properties due to modifications physical characters and resultant energetics of the original materials. So what are the major implant applications? One is in reconstructive joint replacement, then in spinal implants, orthobiologics, and in trauma. Here is a slide showing nanotechnology in wound healing. The interleukin and growth factor signaling networks organize cell to cell and cell to extracellular matrix interactions to fully heal a wound. Any alterations in this sequence leads to non-healing of the chronic wound. Antimicrobial non-nano-based dressings, immunomodulating antimicrobial nanoparticles, genes, modifying silencing technologies, and growth factor releasing nanoparticles are the different modalities available for wound care. Here is a copyright from IIT Bombay, how the dressing looks. Nanoparticles and nanofibers for wound dressings have two layers of silver coating, high density polyethylene mesh, electrospin nanomembrane polyurethane, and silk fibrocin nanofibers be the most commonly used in wound dressings, as you can see here. Electrospun materials have the advantage of high surface area to volume ratio, wide range of pore size, and has high porosity. These factors favor cell attachment, cell adhesion, growth and proliferation. They also improve spreading of type 1 collagen. Porous structures also allow fluid exudates. Coming to reconstructive joint replacement, there is a greater absorption and interaction of selected proteins, which is enhanced for new bone growth and is stimulated by creating nanoscale roughness of the bone. Regeneration of defective bone tissue occurs by a combination of living cells and growth factors with a biomaterial nanoscaffold. Nanomaterial implants reduce infection rates due to the larger surface area, a healthy environment with antibiotics incorporated, and a better wound healing. Extracellular adhesion proteins interact with nanophase implant scaffolds and have osteoblast adhesions. There is a good new bone formation, better fusion between implant and bone when you use nanomaterial technology. Osseointegration is stimulation of rapid new bone formation that makes the implant firmly fixed with the bone. Material properties of implant and bone tissues should be matched. They must be in direct chemical and physical bonding to achieve a good osseointegration. There should be no fibrous tissue interface formation between bone and the implant. Stainless steel and cobalt chrome alloys are used for good mechanical properties, but stiffness of the solid materials results in stress shielding and bone resorption. Osseointegration minimizes stress and strain at the tissue implant interface for better performance and the longevity of implants. The risk of aseptic loosening is a major cause of failure of total hip replacement. Nanotextured material improves osteoblast adhesion, osseointegration, and is also effective in the treatment for bone defects. This illustration shows a magnified nano-engineered implant surface and its topographical interaction with adjacent bone. So the methods available to reduce the implant failure rates, avoid complications, and prolong the life of implants are by implant coating materials, surface character modifications, bone replacement materials, and tissue engineering techniques. Nanostructured diamond hydroxyapatite and metalloceramic coatings are the most common coating materials. Titanium and their alloys have good osseointegration properties and they are ideal implant materials. New bone growth is stimulated by surface roughened techniques like sandblasting and hydroxyapatite coating or by formation of titanium dioxide or titanium helical rosette nanotubules. Titanium nanotubules are the newer class of nanomaterial that have been used with good long-term results. So here you can see there is the calcium hydroxide solution. You add orthophosphoric acid to it. You go for an alkaline pH. You centrifuge for 24 hours and you get nano hydroxyapatite precipitate. You again centrifuge for 6,000 revolutions per minute and you get nano hydroxyapatite powder. And this nano hydroxyapatite has got its characters. And then you put it into a field with electricity and you can see how it gets accumulated near the metal, and the ones without the property are going far away. And that leads to the implant being good for implantation. And the technique has been used for assorted titanium pedicle screws coated in hydroxyapatite. These are the different ways in which you can go for surface modifications. Identification and creation of non-structured surfaces using nanofibers has led to an enhanced implant performance. Nanofiber alumina have an increased cytocompatibility properties. Alumina nanofibers in both delta and theta crystalline phase increase osteoblast cell activity and also calcium deposition. Coming to bone replacement materials, an injectable bone matrix with 100% synthetic nanoparticular hydroxyapatite in paste form is available which gets absorbed in a few months. Engineered synthetic bone hydroxyapatite nanocrystals have structures similar to the native bone. Synthetic replacement material for cancellous bone grafts with a tricalcium phosphate nanoparticle has been developed. They possess large surface areas, higher porosity, enhanced bioresorption, and vascular invasion. Nanocomposite scaffold implants composed of type 1 collagen and non-structured hydroxyapatite are used for the treatment of osteochondral defects of the knee joint. So here you can see on the left, that is nanochem hydroxyapatite, which is non-crystalline osteogenic powder. Then there is a paste and also transparent gel possible. The same is with beta tricalcium phosphate. Coming to tissue engineering, you must have seen crabs. Their hard surface is chitosan. So chitosan is a deacylated form of chitin, an abundantly available polysaccharide. Chitosan has minimal foreign body reaction with an intrinsic antibacterial property, which can be molded to various shapes, and it forms porous structures ideal for cell ingrowth and osteoconduction. Application of chitosan are as bone graft substitutes, chitosan-based hydrogels, wound healing bandages, drug delivery, and for gene delivery strategies. Gene therapies with chitosan will be a reality in the coming years. Nanostructured implants can also be used as fillers. So here you can see what happens with chitosan-based hydrogels. They are biodegradable, biocompatible, non-antigenic, non-toxic, biologically active, biological activity is good, they're antimicrobial, hemostatic. So these are the different functions, and they can be used for drug delivery also as antimicrobial agents, growth factors, with stem cells, peptides, and other schedules. On the right, you can see a wound, how nicely the wound is being there, vascularity is being reduced, scarring has been minimized, and the wound is healing with application of these nanoparticles. The same thing is coming with alginate nanocarriers, nanotubules, chitosan nanoparticles, nanocrystal polymeric agents, and other methods as seen. The other uses nanotechnology for prevention and treatment of osteoporosis. Well, an ideal injectable nanomaterial for vertebroplasty and kyphoplasty treatment must have high injectability and homogeneity during injection. We have been using polymethyl methacrylate bone cement. But if you add it with nanophase magnesium oxide and barium sulfate, then you get higher osteoblast adhesion densities than pure cement. This is how you see the picture. What is pure polymethyl methacrylate bone cement? What is conventional magnesium oxide? What is nano magnesium oxide? And what is nano barium sulfate? Coming to joint replacements, it can be used for evaluation after surgery, where you put it under radio frequency signals and a computer analysis and finds out where is it getting loose, where is it getting away from the bone, where is it developing porosity, is there any infection present or there is no infection present. All those things can be evaluated in this technology. Coming to spine, as you can see in the diagram, that is the spine nicely seen. You bend, you get a disc prolapse, you get a degenerated disc. There are two options. You fuse the spine to prevent pain, but the movements will be restricted, or you go for a nano-made disc and the patient is happy. And that is how it is. You take the cells, you go for a media, you culture it, implant the disc. So these are the novel-based therapies, including tissue engineering for intervertebral disc progenitor cells, such as mesenchymal stem cells, to undergo differentiation into a nucleus pulposus like phenotype regeneration. The combination of these scaffolds with growth factors, such as tissue growth factor beta, under certain conditions has shown promising results in achieving a functional graft for intervertebral disc regeneration. In addition to disc and nerve regeneration, nanotechnology may potentially facilitate spinal fusion and avoid the cost and potential complications associated with recombinant human bone morphogenic protein. In 2014, FDA approved the first interbody fusion device to feature nanotechnology, and it is called as Nanolock, as you can see. It is better than plain titanium meshes and cages that are being used. Peripheral nerve injury. Synthetic conduits using carbon nanotubes and nanoscaffolds offer more tunable mechanical properties than autografts and may enhance nerve regeneration through augmented surface topographical interactions. Carbon nanotubes have demonstrated the ability to promote axonal growth and even mimic some electrical properties of myelin. The nanoscale fibers that comprise these conduits have increased area available for protein absorption, stem cell migration, and axonal regeneration, and guided growth in addition to being easy and cheaply manufactured. Commonly used materials in conduit composition include collagen and silicon. Although not yet clinically mainstream, nanosynthetic material appear to be the future of nerve regeneration therapy. Here's an illustration showing the basic structure of a carbon nanotube. These materials are of significant interest in multiple realms of orthopedics, including nerve regeneration, implant scaffold engineering, and drug delivery. Coming to orthopedic oncology, the principle behind nanotechnology drug delivery as a treatment modality begins with the creation of a drug next. This structure is attached to a specific ligand, such as a monoclonal antibody, which is able to bind and enter the cancer cell. This allows for direct action of the chemotherapy on the desired target with reduced collateral toxicity to non-cancerous cell-loaded nanomolecules. In oncology, preservation of normal uninvolved structure is crucial for maintaining form and function. Gold nanoparticles have a special property called the enhanced permeability and retention effect. Certain such molecules of gold nanoparticles accumulate in cancer cells more than in normal cells. Gold nanoparticles have a huge surface area and surface plasmon resonance properties. Gold nanoparticles restrict cancer metastasis also. Lipid nanoparticle carriers offer an attractive method of treatment against osteogenic sarcoma, as they have been shown to have excellent bioavailability and can be orally administered. Conventional chemotherapeutic agents such as etoposide have been investigated using this technique in treating other types of cancer and have shown promising results. So here is a nanoparticle, and there is the drug-loaded nanoparticle. This diagram is demonstrating the fundamental principles of drug delivery using nanoparticles. This method not only allows for more precise drug targeting, but also greater control of drug release in treating bone cancers, prosthetic joint infections, and osteomyelitis. Then there are nanoselenium implants, which inhibit the growth of malignant osteoblasts while promoting healthy bone function at the implant tissue interface. It was identified that selenium nanomaterial, unlike untreated titanium implants, increased bone adhesion, calcium deposition, bone proliferation, and alkaline phosphatase activity. Coming to tendon healing, silver nanoparticles in the tendon healing process have been used along with in vitro and vivo using Achilles tendon. In the in vitro evaluation showed that the silver nanoparticles promote the proliferation of pyramid tenocytes to silver nanoparticles and the production of extracellular matrix components. The silver nanoparticles accelerated the tendon healing process and moderated the extracellular matrix. Then silver nanoparticles also showed an anti-phlogistic effect, reducing the formation of scar tissue and adhesions. If there will be scar tissue and adhesions, the tendon will not glide, giving movements. So this is the basic principle. And to summarize, you can isolate mesenchymal stem cells. They can differentiate into chondrocytes. You make them biomechanically active, and then you inject it into the meniscus, and you can see that it will heal. Same way, for tissue engineering, you go for a cell source selection. You go for a bioactive molecular selection. You go for a material selection. You decide on the scaffold, and you go for a scaffold manufacture. This one injected in a tendon gap will go for a tendon regeneration. Similar to putting stitches and putting a cast or tendinoachilles tear for 3 months, it can heal without surgery also. So that was the role of nanotechnology in orthopedics. Thank you.
Thank you, sir, for such an extensive and detailed presentation, starting with the history and then going into different ways where nanotechnology can help in the wound healing and bone replacement and everything. And we know that due to this titanium, or we take the magnesium or the platinum nanoparticles, because of their biocompatibility and biodegradability and osteoconductivity, they are being used and will be a great for the futuristic treatment of any of the patient. And I thank you again for such a nice presentation, sir. Thank you.
Thank you, madam. And now that we have got a MOU with IIT, and they're working more on the surfaces and materials for nanotechnology, I think AIIMS Raipur with IIT will be coming out with multiple patents in this field. Thank you.
Thank you, sir. I'd like to request the chairperson to kindly felicitate Dr. Alok Agrawal, sir. Thank you, sir. I like to request our Director and CEO, Professor Nitin Nagarkar, sir, to kindly felicitate the chairpersons. Thank you, sir. We'll be taking a short lunch break of half an hour, and we'll be continuing the session after that. Thanks. So we'll be starting the session post lunch. I request Professor Dr. Ramanjan Sinha sir, Professor Dr. Amit Chauhan sir, Professor Dr. Sandeep Keema sir to kindly chair the next session. Our first speaker will be Dr. Keshwan Karthikeyan, who is from School of Studies of Natural Resources, Department of Pharmacy from Guru Ghasidas Vishwavidyalaya, Bilaspur, Chhattisgarh. And he'll be speaking on cationic nanocarriers, a new avenue for ocular drug delivery.
Yes, good afternoon to all. First of all, I must say thanks to the organizer, especially the Dean sir, Agarwal sir, and Dr. Amit sir for giving this opportunity to share my research work. So through this presentation, I'm going to share my research experience from PhD to till date. So my topic is, am I audible? Okay, yeah. Cationic nanocarrier, a new avenue for ocular drug delivery. So myself, Dr. Kesavan Karthikeyan, Assistant Professor in Guru Ghasidas Central University, Bilaspur. My research group working on the development of a nanocarrier system for ocular drug delivery. So before going to discuss in detail about the nanocarrier system, just I want to give some basic information about anatomy of eye. Eye can be divided into two parts. Anterior segment, that is front side, posterior segment, back side of the eye. So this slide shows the various components of anterior segments like cornea, conjunctiva, aqueous humor or lens. So there are some components in posterior segments like retina, choroid, vitreous humor and optic nerves. So this slide is showing the common diseases which are affecting both anterior segment as well as the posterior segments. The common disease like allergic conjunctivitis, bacterial and viral conjunctivitis or uveitis are the common disease affecting the anterior segments. Age-related macular degeneration, diabetic retinopathy, diabetic macular edema, glaucoma. So these are the common disease which affect the posterior segment of the eye. So the major problem with the ocular bioavailability of the drug, there are a lot of barriers which will affect the ocular absorption or permeability of drug into the eye. The first main factors affecting the bioavailability of the drug is tear secretion or lacrimal film. So tears are the essential component of normal ocular function, but at the same time, which will the negative effect on the ocular absorption of drug. So drugs which may be an irritant to the eye, which may produce more amount of tear secretion, which will wash out the drug when you will administer the drug on the topical route, which will be 95% of drug will be washed out by the tear secretion. Additionally, tears are characterized by high turnover rate, which limit the ocular residence time of the drug. The second factor which affecting the ocular bioavailability of drug is cornea and conjunctiva due to the tight junction between the cells, which will prevent the entry of drug into the eye. So for example, cornea and conjunctiva, will compare cornea and conjunctiva. Conjunctiva little bit permeable than the cornea. We'll come to blood ocular barriers. There are two ways generally drugs are administered. One is local effect
So the topical administered from the eye. The another one is systemic route. Systemic route in the sense it may go for per oral route or parenteral route. So the two barriers which will affect the entry of drug molecule into the eye. The first one, blood aqueous barrier, which will affect the entry of drug which is administered on the topical route, which will prevent the entry of drug through the cornea or conjunctiva. Blood retinal barrier, which will affect the entry of drug through the systemic circulation, which are, if you are planning for administration of drug through the systemic administration, that are also not possible to reach the eye due to the blood retinal barrier. So we need to design a formulation which can enhance the bioavailability of the drug, whether it is topical route. So we need to go for only topical route, because topical route of administration is the first choice of ocular therapy, as it offers convenience, ease of application, and without any pain. So you can easily administer the formulation in the topical route. However, as we discussed earlier, the lot of factors which will influencing the absorption of drug. So we need to only 5% of drug only reaches to the inside the ocular region. 95% will be washed out by the what we discussed, 3 factors. So during my PhD, then we decided to design a formulation which can retain the ocular surface for more period, so that the drug can be enhanced, the drug can be bioavailable more than the conventional formulation. So I did my PhD under the guidance of Dr. J.K. Pandit sir in IIT BHU. So and then we decided to develop a hydrogel system. So inside to gelling hydrogel system. So inside to gelling hydrogel system means a formulation which is in liquid in nature. After administering to the eye, the solution form converted into gel form. So the sol to gel will takes place. The trigger of solution to gel conversion can be achieved in the ocular region by 3 ways. The one is ion activated, so otherwise pH activated. The third one, temperature activated. Ion activated means ions, example, gelan gum or sodium alginate gums are changes to solution form to gel form when it will contact with the ions like sodium, calcium. So as you know, the tears having electrolytes, sodium or calcium. So when we'll administer, it will be in solution form. After administration to the eye, which will contact the sodium or calcium, that will convert it into gel, which will retain on the ocular surface for long period. Similarly, there are pH sensitive polymers like chitosan, carbobol, then temperature sensitive polymers like poloxamer. So these poloxamer, which will be solution form till if the temperature is less than body temperature. If the temperature reaches to body temperature, then it will convert into solution to gel. So 3 ways we can trigger the formation of solution to gel. So we have developed a different hydrogel system. Then due to the development of technology, we need to go with latest technology. Then we decided to develop a nano carrier system. So nano carrier based ophthalmic delivery can overcome the various shortcomings of conventional formulation, like a normal conventional formulation. As we discussed, hydrogel also having some problems like blurred vision. So once the solution converted to gel, so which will visibility become a problem. So to overcome this type of conventional novel drug delivery system, so this nano carrier system can overcome all the problems related to conventional formulation, like eye irritation, which can enhance the permeability of the drug due to the size of the particle. So the design of mucoadhesive nanocarrier can interact the contact time on the ocular surface, which may lead to controlled and sustained release of the drug. So if you design a mucoadhesive means the ocular surface having mucus layer. So if you design a polymer, we can select the polymer which can interact with the mucus layer, which will enhance the retention of drug on the ocular surface, which will enhance the bioavailability of the drug. So especially during my PhD, I have selected microemulsion as a system. So microemulsion is nothing but a mixture of emulsion. I don't know whether you know about emulsion. Emulsion is a biphasic liquid preparation in which one liquid is dispersed into the another liquid. You can see the diagram there. There are two types of emulsion. One is oil disposed in water. The another one is water disposed in oil. So we can prepare two types of emulsion. Oil is disposed in water. The small amount of oil disposed in the large quantity of water, which is called oil in water emulsion. The another one is small amount of water disposed in a large amount of oil, which is water disposed in the oil. So oil disposed in the water generally used for water insoluble drug. So we can say water insoluble drug or lipophilic drug. Lipophilic drug means the drug will be soluble in lipid. Hydrophilic drug means the drug soluble in water. So oil in water system is suitable for lipophilic drug. The reason is drug present inside the system. So the drug will released slowly for prolonged period. So oil in water system generally used for lipophilic drug, water insoluble drug. Water dispersed in the oil, water in oil is used for hydrophilic drug because the drug will be dispersed in a system. So slowly drug will release to the top. If you administer on the topical, drug will be slowly released on the ocular surface, which will retain the due to the nano structure, which will more bioavailable than the conventional formulation. So this system, either oil in water or water in oil, so which can prolong the release of drug due to the structure of size, nano size, as well as the surfactant, the microemulsion containing surfactant. Surfactant is nothing but permeability enhancer. So due to the surfactant, the entry of drug through the ocular surface also enhanced. Small modification in the microemulsion system can develop a more bioavailable system. For example, the simple microemulsion, we can consider phase transition microemulsion. So phase transition microemulsion is nothing but a water in oil microemulsion, so which will be administered on the eye. So due to the secretion of tears, so initially the water dispersed in the oil quantity is high, water quantity is less. Once you administer on the eye, so which will be diluted with the tear fluid, which will change to water in oil emulsion to bicontinuous phase. Bicontinuous phase means where both oily phase or aqueous phase become equal. So oily phase, aqueous phase, when become equal, the consistency will change, viscosity will increase. So that will call a liquid crystalline system. So liquid crystalline system will be more viscous. Further dilution of the tear fluid, the same system will be converted into again oil in water system. So changes from water in oil, then bicontinuous stage, then what oil in water. So this will help the therapy of by therapy or combination therapy in case of ocular drug delivery. I will explain in clear in the next slide. One more thing is self microemulsifying drug delivery system. Self microemulsifying drug delivery system means is nothing but the same microemulsion without aqueous phase. Simply mixture of oil, surfactant. So when you administer on the eye, due to the secretion of tear in eye itself, which will form the microemulsion. So microemulsion is nothing but mixture of oil, water, and surfactant. So self microemulsion drug delivery system is simply mixture of oil and surfactant. Once you administer on the eye, due to the secretion of tears, where eye, which will promote the synthesis of microemulsion in the ocular surface. Okay, so this slide will explain how bitherapy means treatment with two drugs will be fruitful with the help of in situ phase transition microemulsion. So as I discussed initially, if you will develop a water in oil microemulsion, small amount of water dispersed in the large amount of oil, so which will sustain release the hydrophilic drug because the drug is present inside the system. The same system after administered on the eye, so due to the dilution with the tear fluid, which will convert it into biphasic system or LC system, liquid crystalline system. So where if you will administer both hydrophilic or lipophilic, we can accommodate both drug in the microemulsion because which containing both oily phase and aqueous phase, we can accommodate both hydrophilic drug and oil soluble drug in a single system. So during the LC system, both hydrophilic and lipophilic drug get sustained release. further dilution of system by the tear fluid, the system will be converted into oil in water system where the lipophilic drug will be sustained release. So I will give two examples where we can use bitherapy in ocular delivery. The first one, bacterial keratitis. So bacterial keratitis, so as you know, which is caused by bacteria. So common treatment prescribed by the doctor is the combination of fluoroquinolones and dexamethasone. So moxifloxacin and dexamethasone very commonly available in the eye drop form. So moxifloxacin is the water soluble drug. Dexamethasone is the water insoluble drug. We can say lipophilic drug. So if you prepare a inside to phase transition microemulsion, so these two, moxifloxacin will be soluble in water, dexamethasone will be soluble in oil. So this system will be more suitable for the treatment of bacterial keratitis. The another one, ocular cancer, so which will be treated with mitomycin C and cyclosporin A. Here mitomycin C is a water soluble drug, cyclosporin A is a water insoluble or lipophilic drug. So this combination therapy also more suitable with this particular system. Then we decided to develop a nanocarrier system. Previously, we were used some mucoadhesive polymer. Then we designed a system which can enhance the retention of drug on the ocular surface. Then we decided to prepare a positively charged nanocarrier system or cationic. That is my topic. Cationic nanocarrier system or positively charged nanocarrier system. So the reason behind the design of positively charged nanocarrier system, because the cornea, conjunctiva, which is covered with the mucous layers are negatively charged. So due to the negatively surface charge on the ocular eye, if you will administer positively charged or cationic charged nanocarrier, that can interact with the negatively charged surface due to the electrostatic interaction. So simply the electrostatic interaction, which will enhance the retention time of the drug as well as the permeability of the drug, which will enhance the bioavailability of drug. So we have developed a different positively charged nanocarrier system. We are going to discuss something, 4 system here: positively charged nanocapsules, cationic nanoemulsion, cationic liposome, or cationic nanomicelles. So these are the different nanocarrier system. So the reason behind the selection of nanocarrier, due to the electrostatic interaction with the negatively charged ocular surface, which will enhance the bioavailability of drug. In the first case of nanocapsule, so nanocapsule can be synthesized by the conventional method only, polyelectrolyte nanocapsule. Polyelectrolyte nanocapsule is nothing but, so I will show the diagram so you can easily understand. So if you take one anionic polymer and take one cationic polymer, simply mix anionic polymer with the cationic polymer in presence of drug, which will encapsulate the drug at same time, which will form a nanocapsule structure. So if you will mix accordingly, the mixing of order is very important. So when you will mix the anionic liquid to cationic liquid, then you will get the cationic on the surface. If you will do opposite direction, then you will get cationic anionic surface. So if you mix the anionic liquid into the cationic liquid, you will get the cationic surfaced nanocapsule. So we have selected chitosan as a cationic polymer, so which is more suitable, which is approved by FDA, biodegradable, cost-wise very effective, easily available. So for that reason, we have selected cationic polymer. We have developed different nanocapsules with the help of different anionic polymers with chitosan as a cationic polymers. So these are some publications related to polyelectrolyte nanocapsule for ocular drug delivery. The next, cationic lipid nanocarriers. So lipid, as you know, lipids are negatively charged. So cationic lipids is very easily to make. Simply you add chitosan to the cationic lipid system, which will form a cationic lipid. You simply prepare liposome, add chitosan, Then due to the electrostatic interaction of lipid with the chitosan, which will form cationic lipid nanocarrier. So in cationic lipid nanocarriers, we have designed a two system, cationic nanoemulsions and cationic liposome. So nanoemulsion, as we discussed earlier, so microemulsion, nanoemulsion, emulsion are nothing but same system, but there is small difference in each other. So normal emulsion is the emulsion where the globule size or particle size of the globule is macro size, micrometer. In nanoemulsion, microemulsions are nano range. The difference between nanoemulsion and microemulsion is a small difference. Nanoemulsions are kinetically stable. Microemulsions are thermodynamically stable. So kinetically stable means you need high speed homogenizer to make a nanoemulsion. But microemulsion thermodynamically stable means due to the chemical component of the surfactant. So you need some more quantity of surfactant. So which is called CMC, critical micellar concentration. You need to use the concentration higher level of CMC, so which will easily form the microemulsion. Now microemulsion size is comparatively very small to nanoemulsion. Nanoemulsion means the nanoemulsion size may be considered less than 1 micron emulsion will be considered nanoemulsion. But microemulsion means the particle size or globule size less than 100 nanometer will be considered as a microemulsion. So simply making microemulsion, what we discussed earlier, then add chitosan to that, which will form a cationic nanoemulsion or microemulsion. Similarly, liposome. Liposome is the bilayer system. Bilayer system means phospholipids, made up of phospholipids. Our body membranes are made up of phospholipids. Similarly, phospholipids used for the synthesis the system which is called liposomes. Liposome also negatively charged. Simply prepare liposome, then add cationic polymer chitosan, which will convert into cationic liposome. This particular liposome is used for gene therapy still 2 years back. But recent years there are number of different nanocarriers are used. So this only system previously used as a carrier for gene therapy. So finally, nanomicelles. There is small difference between microemulsion and micelles. Microemulsion is the oil, water, and surfactant. In micelles, there is no need of oil. Simply adding surfactant above the concentration of CMC, which will form a micelles. There is the difference between microemulsion and micelles. Micelles simply drug, water, surfactant. Microemulsion, drug, water, and oil. So there is no oil phase in the micelles. So we have prepared nanomicelles with TPGS and chitosan. TPGS is nothing but vitamin E derivative surfactant, which will be used along with the cationic polymer chitosan, which will form a cationic micelles. This is the latest publication, micelles used for the glaucoma therapy. Okay, now come to gene therapy. Gene therapy, which involves intracellular delivery of genetic material, either to block a dysfunctional gene or to deliver a gene as a therapeutic. So gene therapy is nothing but if any gene disturbed or mutation takes place in our body or any genetic disease particularly. So the genetic disease means there is some alteration in the gene. That is why the disease created. So if you find out what is the responsible gene the disease created, the mutation takes place, if you find out the gene, then we can deliver the specific gene to the target site where we can cure the genetic disease. So we can use this cationic nanocarrier as a carrier for delivering the genetic material. Genetic material in the sense like DNA, RNA. So to carry the genetic material, previously only viral vectors were used. There are two types of vector used for the gene therapy: viral based vector, non-viral vector, viral based vector. Viral based vector, as you know, most of vaccines are synthesized by viral based vectors. But recent years, some COVID vaccines are synthesized in non-viral vector based also. So viral vector based previously commonly used system. But the problem with the viral vector based system having the immunogenicity and the mutagenesis problem. So recent years, scientists are focusing on non-viral vector based system. Non-viral vector based means nothing but a nano carrier system. So in nano carrier system, positively charged nano carrier is more suitable for gene therapy because the plasmid is negatively charged due to the phosphate group in the DNA, which will easily interact with the positively charged nanocarrier, which will easily bind with the positively charged nanocarrier, as well as the cell membrane also made up of negatively charged, so which can easily enter into the cell when you use the positively charged nanocarrier system. Your successful gene delivery system should protect the negatively charged DNA from anionic cell surface repulsion. Cell surface repulsion means our cell is made up of negatively charged. The DNA also negatively charged. Due to the negative charge, there is a repulsion takes place. When you will combine the genetic material to a positively charged nanocarrier, which will become a positively charged. So there is the easy entry into the cell due to the electrostatic interaction. A typical non-viral vector which containing cationic compound that easily bind with genetic material because negatively charged, as well as the which will easily enter into the cell through electrostatic interaction. So 2 years back, I got a fellowship from DBT, Getting Fellowship, where I visited University of Iowa, where I learned some technique used for gene therapy or gene editing. So treating ocular disease, and in particularly, I have proposed in that fellowship is glaucoma. So because I am working in the ocular drug delivery system, so I have made collaboration with Professor Val Sheffield in genetic department in Carver College of Medicine, University of Iowa. So we designed a positively charged nanocarrier and bind with the genetic material which is responsible for glaucoma. The gene is myocilin gene. So we have designed a there is a technique CRISPR. CRISPR is the nothing but a combination of targeting RNA along with the enzyme, that enzyme will cut where you want to deliver the genetic material. So CRISPR is the technology, recent technology commonly used for gene editing. So with the advancement of genome editing, we can consider this nanocarrier system. So this CRISPR system also negatively charged. CRISPR is nothing but the combination of a gene along with some RNA. So both they are negatively charged complex. When you will combine with the complex with the positively charged nanocarrier system, which can efficiently deliver the genetic material to the target site. So as we discussed, the complex nanocarrier system will easily deliver the genetic material into the inside the cell for treatment of any genetic disease. So this can be extrapolate, or we can go with any other genetic disease, not only in ocular. So any genetic disease can be curable with this type of technique. So conclusion, effective management of ocular disease is the major challenge because of presence of I have discussed here 3 major barriers. There are more than number of barriers are there. Consequently, design of nanocarrier system with improved drug delivery property to ocular surface would be promising step towards the management of ocular disease. So nanocarrier, especially positively charged nanocarrier, will be more suitable for ocular drug delivery. So non-viral vector based CRISPR technology will be more suitable for gene therapy. Currently, we are working on that. So I have developed a system for the treatment of glaucoma. So now we are working for same CRISPR based system for the treatment of cancer as well. The proposed nanocarrier promote higher transfection efficiency due to the electrostatic interaction with the negatively charged genetic material. Okay. So if you have any query, I am ready to answer that.
Thank you, sir, for the wonderful lecture. I request the chairpersons to kindly present a memento to our speaker, sir. Thank you, sir. I'd like to invite the next speaker, Dr. Manju Agrawal. She's an assistant professor from Department of Pharmacology, PTJNM Medical College, Raipur. And she will be speaking on nanotechnology, pharmacological perspective.
Good afternoon, everyone. I first of all thank the organizing committee for inviting me and giving me an opportunity to speak on this very new topic, which is an integration of science, technology, and biomedicine. The previous speakers have already introduced the topic, and application of nanotechnology help to develop nanomedicine, and it exploits the improved and often novel physical, chemical, and biological properties of materials at nanometer scale. We know that the size of a nanoparticle should be between 10 to 100 nanometer, which is smaller than an antibody. And the viruses, it means they are not visible to the naked eye. In 1986, Matsumura and Maeda observed that anti-cancer protein bound to polymeric nanoparticles exhibited greater accumulation in tumor tissue than in healthy tissue. And in 1995, the first liposome-based nanostructured delivery of doxorubicin for cancer was approved by the FDA. So why use nanoparticles? Nanoparticles bridge the barrier of biological and physical sciences. Drugs with poor solubility, less absorption ability are usually tagged with nanoparticles because nanomedicines or nanoparticles help to encapsulate or attach disease markers, and nanosensors within or on the surface of the nanoparticle. These particles have enhanced permeability and retention, thereby reducing the dose and dosage frequency and improving the efficacy both of novel and old drugs. These nanoparticles enable tailored designs for precision medicine with minimal side effects and improved patient outcome. There are different types of nanoparticles available, and the nanomedicine can be a formulation of a single nanoparticle or a composite nanoparticle, means more than 1 or 2 or 3 nanoparticles used together. We classify the nanoparticles into polymer-based, lipid-based, non-polymeric, and nanocrystalline. The present drugs available are formulated with the help of dendrimers, micelles, drug conjugates, protein nanoparticles, and nanogels. Lipid-based nanoparticles are most frequently used, like liposomes, exosomes, and solid lipid particles. Non-polymeric nanoparticle includes carbon nanotubes, nanodiamonds, metallic nanoparticles, which include gold, silver, magnesium, etc. Then we have the quantum dots and silica-based nanoparticles. So we have all these nanoparticles in therapeutics. We can use nanotechnology for therapy, for drug delivery, for diagnosis, for regenerative medicine, and tissue engineering. So what are the properties that affect application of nanoparticles? The material used has a major impact, followed by the size and the shape. Next, the surface conjugation of various substances that will affect the bioavailability of the nanoparticle. So before we go on to use these nanoparticles, it is important that the surface of the nanoparticle is functionalized. This can be achieved by bioconjugation or passive adsorption of molecules or ligands onto the nanoparticle. So we will be discussing each nanoparticle and the available drug in therapeutics. Coming to the lipid-based nanoparticles, which include liposomes, exosomes, and solid lipid particles, we all know about the liposomal amphotericin B. We have all used it during the COVID-19 pandemic for mucormycosis. So what is the importance of liposomal amphotericin B in comparison to the conventional amphotericin B? This liposomal amphotericin B, available as ambisome, has improved pharmacokinetic properties and it is more stable in the circulation. There is decreased accumulation in the normal uninfected tissue, decreased mammalian cell toxicity, making it more safer than the conventional liposome. Similarly, we have liposomal irinotecan, which has improved circulation time, passive tumor targeting, and fewer side effects. Liposomal vincristine also offers the same advantage. The second step in liposomal drug is pegylation. Pegylation helps in better absorption of the drug. When pegylated, especially in doxorubicin for cancer therapy, this drug can cross penetration in vasculature, so there is improved tumor localization, increased blood circulation time, less vulnerable to the immune system, better tolerance than the conventional formulations, and favorable toxicity profile. Doxorubicin is well known for cardiac toxicity, so it poses better cardiac safety, less myelosuppression, and other side effects. Now, if we assess what are the sizes of nanoparticles that would be best, different formulations were prepared with different size nanoparticles, and nanoparticles with less than 100 nanometer had the best accumulation in the tumor tissue compared to the 200 and 400 size nanoparticles. So smaller the size, better the response. Other liposomal based approved drugs available are liposomal formulation of strabin-daunorubicin for acute myeloid leukemia, liposomal vincristine for Philadelphia chromosome negative acute lymphoblastic leukemia, and liposomal irinotecan, which is pegylated for metastatic secondary pancreatic cancer. These all drugs are available in the market and approved by FDA. Albumin-based nanoparticle formulated drug, that is paclitaxel, which we call NAB-paclitaxel. NAB stands for nanoparticle albumin-bound paclitaxel. It is a solvent-free nano version of the non-nanoparticle size paclitaxel available as Taxol. The advantage of this drug is that it has got passive targeting. It overcomes the P-glycoprotein mediated drug resistance, and it is used for advanced non-small cancer, lung cancer, breast cancer, and pancreatic cancer. In this COVID pandemic, we have seen 2 vaccines available in the market with lipid nano formulations. The 2 vaccines authorized by FDA and the EMA use lipid nanoparticles to carry the messenger RNA that codifies for the spike protein of SARS-CoV-2 virus. Owing to the size of the nanocarriers, which is between 50 to 200 nanometer, this formulation is referred to as nanovaccine. It is composed of nucleoside modified mRNA encoding for the virus, and people have used it in US and Europe. Available as Moderna vaccine and Pfizer BioNTech COVID-19 vaccine. The second type of lipid nanoparticle are exosomes. Exosomes are cell-derived nanovesicles that are present in our body. Artificial exosomes as drug delivery vehicles can be formulated, and therapeutic substances like protein, mRNA, and can be delivered to the recipient cells. They have phospholipid bilayer, which can directly fuse with the plasma membrane of the target cell, thus improving the cellular internalization of the encapsulated drug. They are the prospective vehicles for formulation of paclitaxel and doxorubicin. Macrophage-derived exosomes loaded with paclitaxel have exhibited 50 times higher cytotoxicity to drug-resistant cancer cells and stronger anti-neoplastic effect in mice with pulmonary metastasis. Similarly, polyethylene glycol paclitaxel-loaded exosomes have shown superior accumulation in cancer cells with improved therapeutic indices. The next type of nanoparticles are polymer-based nanoparticles, which include micelles, dendrimers, drug conjugates, nanogels, and protein nanoparticles. In the picture, you can see a drug when combined with a polymer will form a drug polymer complex. Now, this drug polymer complex can be of two types. It can be a nanocapsule in which the drug is either inside the capsule or on the surface of the capsule, or nanosphere, depending on the density of the nanoparticle. So again, the drug may be entrapped or may be on the surface. These polymer-based nanoparticles in natural polymers include alginate, chitosan, gelatin, and albumin. The formulations made with these polymers help in control release of the drug and protect drug and molecules from biological activity, thereby improving bioavailability and therapeutic index. The second lipid polymer-based nanoparticle are dendrimers. As you see in the picture, they are highly branched layers of polymers with terminal groups, and you can see there are multiple generations. At each branching, a generation is encoded to it. So in the formulation, it will be written G1, G2, G3, G4, depending on the number of layers in the dendrimer. They are made up of multiple types of substances, and anti-neoplastic agents like camtothecane, methotrexate, and 5-fluorouracil have been formulated with the help of dendrimer. We have a drug, polyamidoamine dendrimer conjugation with paclitaxel for breast cancer, imitinib in lung cancer, which has increased water solubility, improved targeting, and release in the neoplastic cells. Cisplatin with dendrimer is used in breast and ovarian neoplasm, and it has decreased half maximal inhibitory concentration and increased neoplastic cell apoptosis. The second type of polymer is dendriplex, which is a combination of dendrimer with nucleic acid. So dendriplexes have better ability to internalize and release nucleic acids. The co-delivery vector, which is produced by conjugation of branched polyethylene glycol with dendrimer, have two generations, and they load hydrophobic drugs with high loading capacity. Here in the picture, you see a dendrimer with of a doxorubicin dendrimer, which has been conjugated with doxorubicin and small interfering RNA, and the combination is used against tumors, which is more effective than the drug used alone without a polymer. The available agents are doxorubicin and paclitaxel. The other polymer-based nanoparticle drugs available are P-gylated antibody fragment of certolizumab for Crohn's disease, rheumatoid arthritis, psoriasis, and ankylosing spondylitis. Similarly, pegfilgrastim, which is a colony stimulating factor, which is available as Neulasta, is a monomethoxy peg aldehyde to a N-terminal methionine residue of filgrastim. This pegylation and nanoparticle formation increases the half-life of filgrastim which is about 3.5 hours without pegylation, and it is increased to 42 hours by pegylation and nanoformulation. Similarly, in 2017, factor 9, glycopegylated coagulation factor 9 has been approved for hemophilia with an extended half-life. Coming to the next polymer nanoparticle, chitosan. In the previous lecture, we have seen how chitosan improves the mucoadhesion and increases the drug residence time at the mucosal surface. So this formulation with chitosan can be used for ocular drug delivery. Fluconazole loaded chitosan is available for fungal keratitis. In the picture, you can see the, as in the previous speaker had said, chitosan is positively charged and the surface is negatively charged. So this will go and have better mucoadhesion. This can also be used for oral delivery. Doxorubicin has been formulated with chitosan for increased absorption in the small intestine. Tamoxifen chitosan formulation has been found to have 90 times increase in permeation at the target site. Itraconazole, an antifungal drug, which can be formulated as a dry powder with chitosan, has low solubility, which has better solubility when administered orally. We can also deliver drugs, vaccines through nasal mucosa, buccal mucosa, and vaginal drug delivery using this polymer. The present nasal drug delivery system, none of them use nanoparticles, but we look forward to have new drugs with chitosan as drug delivery vehicle and nasal drug delivery. The next polymer is oleogels. Spray-dried tenofovir alafenamide with chitosan nanoparticle loaded oleogel has been found to be long-acting, and it can be used as a depot system for HIV treatment. At present, tenofovir alafenamide is used as a daily dose regimen orally, but this depot preparation will have to be given once in a month. It is yet not approved, but maybe in near future we will have this preparation. Alginate is another nanoparticle which can be used, and it possesses biocompatibility. Rheological properties means it retards the release of the drug, biodegradability, marginal toxicity, and chemical versatility, which can be used for drug formulation. It can form stable gel in aqueous condition and is beneficial for drug delivery. In the market, many hydrogels are available for wound dressing, especially in burns, and hydrogel are also available for analgesic action. Hydrogel topical diclofenac sodium is available and has got better permeability and retention time at the site of application. Alginate cyclodextrin 5-fluorouracil nanogel, which has been prepared, has got enhanced chemotherapeutic effect by pressure controlled drug release for colon carcinoma. Similarly, alginate doxorubicin injectable hydrogel releases the drug gradually and has got effective concentration at the target site and overcomes the multi-drug resistance in cancer treatment of prostate carcinoma. Coming to micelles, micelles are amphiphilic block copolymers which can self-assemble to form a micelle with a hydrophobic core and a hydrophilic shell as seen in the diagram. The drugs can attach to the shell or to the capsule and can be delivered at the target site. The advantages are that they can carry water-insoluble drugs, they are more biocompatible, biodegradable, easily modified and functionalized. But the disadvantage is that they are more difficult to selectively target cancer cells and achieve optimal concentration. Coming to the non-polymeric nanoparticles, we have the metallic nanoparticle, carbon nanoparticle, quantum dot, and silica. Carbon nanotubes are the recent development, and they consist of rolled up sheets of graphene in which a layer of carbon atom is arranged in an hexagonal lattice. Docetaxel, doxorubicin, methotrexate, paclitaxel, and dexamethasone has been prepared using carbon nanotubes. These drugs are water soluble, biocompatible, and multifunctional. But the disadvantage is that the exact mechanism of action is not known, and they are very expensive for use in mass. The next nanoparticle are cubisomes, which are cubical in shape, and the drugs are encapsulated in the cubisomes as shown in the diagram. They can distinguish normal cells from cancer cells. They are having better biodistribution, more efficacy, and less side effect. At present, dantrolene sodium, which is used for malignant hyperthermia, nanosuspension is paired with a lyophilized formulation and makes it possible to reconstitute and deliver the drug within 10 seconds with high concentration and low, high volume and high concentration and low volume. It is still in the clinical trial phase. The drug encapsulated cefuroxime with prilocaine, which is a local anesthetic. They can be used for local infections like genitourinary infections, respiratory tract infections, and soft tissue infections. Coming to the quantum dots, these are semiconductor core material made of heavy metals like cadmium, selenide, and arsenide in the inner shell, and on the outer shell is the zinc and cadmium to prevent the toxicity of these heavy metals. They glow when stimulated by an external source such as UV light, and fluorescent assay for disease detection can be done. Quantum dots have been used as targeted doxorubicin drug delivery nanosystem for cancer cells with folic acid. Then coming to heavy metals in non-polymeric, metals in non-polymeric nanoparticles, iron formulations, ferrumoxitol and ferric carboxymaltose are available in the market, and they are less chances of anaphylactoid reaction and better tolerated, and larger amount of drug can be delivered to the patient at a single time. Now we have nanoparticles as next generation antibiotics. It means we can use these nanoparticles without conjugating it to any other biological agent. Nanoparticles such as silver, gold, zinc, copper, magnesium, etc., or supermagnetic iron can overcome resistant mechanism by different mechanisms. They can interact with the bacteria, leading to the disruption of the membrane potential and integrity, leading to cell death. They can also trigger the host immune response. These nanoparticles can inhibit the biofilm formation, and they can also inhibit the RNA and protein synthesis through the induction of intracellular effects. So as we are going towards antibiotic resistance and no new antibiotic developed in the last few years, we look forward to nanoparticles as next generation antibiotics. Nanoparticles can also be used to transfer drugs with higher dose at infected sites. Tetracycline silver nanoparticle complex interact more strongly with salmonella cell and causes more release, thereby creating a high concentration of silver near the bacterial cell that leads to the growth and inhibition of the bacteria. The side effect is its toxicity. This is not available. It is still in the clinical trial phase. In the previous lecture, we have heard about nanomaterial coating on biomedical implants. Zinc nanoparticles are available against biofilms. They are used to functionalize surface or reduce biofilm formation on medical implants, and they have antimicrobial activity by inhibiting cell membrane, release of zinc ions that alter the bacterial environment and reduce the oxidative stress. They are effective against both gram-positive and gram-negative strains, but exhibit greater sensitivity and susceptibility to gram-positive bacteria compared to gram-negative bacteria. Zinc nanoparticles can also be used for bioimaging because they are luminescent and exhibit unique size, optical, and structural features with higher level of brightness and photostability and biocompatibility than other organic fluorescent dyes. Graphene and folic acid zinc oxide nanocomposite has been used in cancer bioimaging. Nanoparticles can also be used for regenerative medicine, and they can be used as a substrate for cell growth and proliferation and deliver medicines and biological molecules at the target site. Different biodegradable nanoparticles are available like poly-L-lactic acid and polyglycolic acid. Nanoparticles can also be used for gene transfer because they have demonstrated better functionalized nanoparticles. They have better absorption and they have low immunogenicity and toxicity. Neutralization can be achieved by coating with hydrophilic polymers such as polyethylene glycol or chitosan. This gene therapy is still in clinical trial phase. Silica can also be used for gene transfer because this has got low toxicity. Amine-coated silica nanoparticles are efficient in transfecting genetic materials compared to the viral vectors. Because they have reduced immunogenicity, they have enhanced tumor transduction by 23 folds, but have reduced liver uptake and of tumor targeting by 210 folds. It means it is less taken up by the other tissue compared to the cancer tissue. The disadvantage is that the decreased delivery efficiency in the presence of serum containing media due to interaction with serum proteins. So we have discussed all different types of nanoparticles. The next generation nanoparticles are plants and microorganisms. Different methods like physical and chemical methods have been utilized for formulating the polymer or non-polymer based nanoparticles, which are costly and environmentally toxic. So these plant and microorganism based nanoparticles are the new components which will be less costly and less toxic. The challenges with the nano formulations is that most of the nano formulation drugs are of basically the same drugs we have seen in this presentation, mostly doxorubicin, paclitaxel, 5-fluorouracil. Not more than 25 formulations are available in the market. So we have to look forward to formulating more drugs with nanoparticle. Biocompatibility of nanodrug loaded particles and degradation of this nanoparticle is another issue. There is no guideline on standardization of nanoparticles. They are costly and difficult to manufacture for mass consumption. Nanotoxicity, biodistribution, and accumulation and clearance of these nanoparticles by human body is at present difficult to understand and predict. Tests for toxicity using validated methods tailored to the nanomaterial are not available. So this is still in the budding stage. So what are the future perspectives? We look forward to a design and development of multifunctional nanoparticle for integrated cancer imaging and therapy, release of drug payloads inside the targeted cell or organ that are responsive to pH, temperature, or enzymatic reactions, and designing and development of biocompatible nanoparticles to overcome non-specific organ uptake and reticuloendothelial system scavenging of these nanoparticles has to be understood. So what we learned is that nanoparticles can effectively solve the problem of targeting, local drug delivery, control release, sustained release, and reducing toxicity, but they do carry with it the side effect of the toxicity of nanoparticle itself. In future, we look forward to optimization of drug delivery in a more personalized medicine with the help of these nanoparticles. Thank you. Any questions?
Hello. Yeah. Thank you, Dr. Manju, for such a excellent presentation. The pharmacology. Thank you very much for your presentation.
Thank you, ma'am. I'd like to request the chairperson to kindly present a memento to Dr. Manju, ma'am. Thank you. I'd like to request Professor Dr. Alok Agrawal, sir, to kindly present a memento to the chairpersons. Thank you. For the next session, I'd like to request Professor Dr. Anil Goel, sir, and Dr. Atul Jindal, sir, to kindly chair the session. The first lecture will be by Dr. Rupa Mehta, ma'am. She's an additional professor at Department of ENT, AIIMS Raipur, and she will be speaking on nanotechnology in otology, current status and future directions.
This is the same one, no? Final.
So now we welcome Dr. Rupa. She is additional professor in Department of ENT Head and Neck Surgery. And she's going to talk on very exciting topic, that is role of nanotechnology in otology. What is the current status and the future directions? Looking forward for your talk, Rupa.
Thank you so much, sir. So a very good afternoon to all. We'll be discussing, since morning we have been discussing about nanotechnology in various fields. You know, Madam presented a very elaborate talk on in pharmacology. So we'll be seeing how it is applied in otology. So why otology? Because, you know, inner ear, the ear, as we all know, consists of three parts: external, middle, and inner. External, middle are amenable to medical and surgical treatment. But inner ear, since it is situated deeply in temporal bone, which is a very hard bone and has a very complex anatomy, composed of various membranes, various fluids, and very, very small, very delicate and small structures. So it is very, very difficult for the medicine to reach there and to reach there in effective quantity. So as we all know,
Inner ear disease is, like the signs and symptoms are hearing loss, tinnitus, vertigo, which are very disabling for the patient. So access to the sensory elements, that is the outer hair cells, the inner hair cells, the organ of corti, is restricted by various membranous partitions. And there are various, membranes and the barriers to this. And it is difficult to observe and obtain pathological tissues directly. Even if you have to administer drugs directly, it is like it itself entails a lot of destructive surgery. Diagnosis and treatment of inner ear disease has always been a major clinical problem. And there are so many multiple conditions of inner ear for which we have to raise our hands and say there is no treatment per se. So traditional treatments for surgical access to cochlea, even with operating microscopes, are marred by their inherent destructive nature. And as we all know, hearing loss is one of the most important sensory deficits, one of the most common. By 2050, disabling hearing loss due to sensorineural hearing loss will affect over 700 million individuals, which is a huge number. And we're just able to rehabilitate. We're not able to cure or treat these patients. So although common diagnosis and treatment of inner ear diseases is hampered by unique anatomy, limited access to inner ear due to relatively tight windows facing middle ear and blood inner ear barriers, which allow homeostasis of inner ear, but make it difficult to reach the drugs to reach the inner ear. So sensorineural hearing loss, as we said, is very common, can occur due to cochlear, that is the organ of corti and the outer hair cells, inner hair cells pathology, or retrocochlear, that is the spiral ganglion, the nerves, etc. So cells to brain can lead to SNHL. Although there is increased understanding, we understand the physiology, the pathophysiology, better, but delivery of these biomaterials to targeted sites is challenging. The leading causes of SNHL, as we all know, are advanced age, that is presbyacusis, ototoxic medications, the various cancer medications, the various malarial medications, then noise exposure, inherited disorders, and immune disorders. And almost all SNHL is irreversible. It is permanent. It causes permanent hearing loss. And current management is limited to sound amplification devices and cochlear implants. Nanotechnology provides a potential for a non-invasive and targeted approach to deliver various biomaterials to specific structures which are involved in the pathogenesis of multiple diseases. Cochlea, as I already told, is a very, very delicate, very, very complex, post-mitotic organ. It has very, very little regenerative abilities, very little proliferative abilities. And its sensory receptors are very, very delicate, comprising of various stereocilia and vibration-sensitive hair cells, their synapses and neurons that serve them. It is exquisitely fine-tuned, even with your eyes closed, you can hear, we can identify within seconds, you can identify 100 different voices. So it is very fine-tuned and responds within millions of seconds to displacement of atomic dimensions. These neurosensory structures are fragile and may be permanently lost due to genetic, due to environmental factors, and due to aging process. So the hearing loss can be due to loss of hair cells, the synaptic dysfunction, depletion of neural population within the cochlea, or dysfunction of stria vascularis or fibrocyte network. So in this picture, we can see the various components of the ear and how inner ear is situated deep within the recesses, and you have to traverse the external and middle ear to reach that. And even when we are doing it very delicately with the help of the latest technology, it is a destructive surgery, and there are multiple barriers for the drugs to reach there, to have optimum effect. So this is the picture of the cross section of cochlea showing its blood supply. It's an end arterial supply, and it's like labyrinthine artery, which gives the vestibular, cochlear, and spiral modular artery. And for the drugs to reach there, the systemic, when we administer drugs systemically, the drugs will reach the optimum, the optimum dosage for which it has to reach the area happens at the very high systemic doses with its consequent side effects. So nanomedicine helps us by utilizing various advanced drug delivery systems, new therapies, and in vivo imaging that overcomes the shortcomings of conventional medicine by targeted delivery, by delivering optimal dosages, and by controlling cargo release. Nanoparticles are small, they have high biocompatibility, good degradation, and simple synthesis. But however, there's still a lot of gap which needs to be bridged by research and various trials between biological research and clinical application. So as I said, they are small, they have high bioavailability and strong plasticity. They can be loaded with drugs, as Madam very nicely told, various types of nanoparticles and how they're loaded with drugs, or combined with deafness drugs to act as anti-inflammatory and antioxidant agents. They can carry genes, they can, non-viral vectors of genes into inner ear to facilitate gene transfection as tracers and imaging agents. and auxiliary CI electrical stimulation, which we shall see later in the course of the presentation. So these are the various nanoparticles which Madam already told, the various organic, inorganic, and other nanomaterials, cubosomes, nanocrystals, nanotubes, etc. They can be zero-dimensional, one-dimensional, two-dimensional, or three-dimensional. Multifunctional nanoparticle, again, very, very important. Multifunctional in the sense like, various conjugation of various structures can happen within the nanoparticle, so that we can target specific cells. So targeting ligands, which can be protein, which can bind proteins, antibodies, nucleic acids, small molecules. Then you have surface functionality that charges cationic charged nanoparticles. Then the composition and the shape. So targeting modification of nanoparticle-based delivery system helps increase circulation time and prevents degradation of drugs. Specific surface modification enables nanoparticle-based drug delivery system to reach a specific type of cell. And it is very, very particular for, very important for inner cells, because, for inner ear, because the various pathogenesis of various diseases involve specific cells. For example, ototoxicity will involve outer hair cells. So that way it is very, very important for targeted delivery. And ligands have a specific interaction with certain cells used for cell-specific targeting. So this is how for cochlear, retrocochlear hearing loss, we have this outer hair cells, the inner hair cells, the spiral ganglion cells, which can serve as targets for various nanoparticle conjugated neurotrophins or drugs. So therapeutic agents under consideration for inner ear delivery are antibiotics, anti-inflammatory agents, antioxidants, neurotrophins, and genes. So various treatment methods are systemic, then local, that is intratympanic injection, and surgical treatment. And for inner ear, the surgical treatment is very, very extensive, and it causes a lot of damage to the normal structures as well. So the biggest obstacle to delivery of drugs or neurotrophins or nerve growth factors is the blood labyrinth barrier. And the second thing is wash down. If you give intralocally, like intratympanically and locally in the round window, then it washes down through the eustachian tube. So nanoparticles can increase the retention time of drug and permeability of round window membrane or oval window. Local inner ear drug delivery technologies have been developed, such as microneedle introduction using nanoparticles and hydrogels as carriers. And they have been widely used in sensing, image enhancement, and transmission. So this diagram shows how, like intratympanic injections we give at to instill the drug into the middle ear so that it gets absorbed through the round window or the oval window into the inner ear. But most of it washes down the eustachian tube, and the permeability of round window membrane and oval window membrane is again very variable, and it becomes even more variable in various disease conditions in which we want the drug or the factors, the growth factors to act. And there are multiple membrane barriers, as we shall see later. So see, there are blood endolymph barrier, then perilymph endolymph barrier which are there in the various parts of the inner ear, which hamper the absorption of the drug. So this table, lists the mechanism and challenges of inner ear treatment and how nanoparticles, they overcome these by, keeping the drug more, more retention, more retention happens near the sites of absorption, and enhanced cellular uptake and targeted delivery happens. So transtympanic injection, best method for delivering drugs for inner ear disorders. They're the least invasive, and the drug is transferred via oval or round window membranes. NPs stabilize drug molecules and achieve site-specific targeting. And after administration, distribute in perilymph and endolymph compartments. These are the various types of nanoparticles which have been used in research for the various ailments of inner ear. In this picture, you can see how the nanoparticle, which is combined with the hydrogel, helps in better absorption, and then how the nanoparticle is released and the various active, biomaterials are released and absorbed in the inner ear. Now, inner ear imaging, as I already told, is very small, very delicate, and situated deep within the recesses of temporal bone. So it is very, very, difficult to image the fine structures of human inner ear. And the synchrotron radiation phase contrast imaging is proving to be transformative, but still it is experimental. So diagnosing hearing loss and vestibular malfunction, monitoring behaviors of the nanoparticles within inner ear, and evaluating outcomes of treatment is difficult as of now. And we need to do more research in terms of nanoparticle tracing and imaging. These are the various studies in which they have done nanoparticles tracing using nanoparticles. Now, gene therapy, very, very important. It is a safe and useful non-viral gene delivery system to deliver genes to patients with congenital or chronic hearing loss. And most of the children, you know, the genetic causes, as we all know, of genetic causes of hearing loss are very, very relevant, and they affect early in the infancy. So genes such as ATO1 can potentially regenerate hair cells. That happens in case of presbyocusis. However, the challenges are decreasing particle size while stably integrating gene into the particle, administering a gene nanoparticle complex to body, protecting gene from degrading enzymes, ensuring gene enters cytoplasm, escapes endosome and enters nucleus, and autofluorescence of inner ear, and vulnerability of fluorescence intensity to laser of confocal microscopes hampers the evaluation. So these are the common methods of inner ear gene therapy utilizing the various viruses, and the various studies which have been done in which the role of nanoparticles in gene therapy has been tested. Now, nanodialysis for cisplatin-induced ototoxicity. Cisplatin is a very, very common drug used in treatment of carcinomas, and it causes, it has been said that with the complete, it causes, with the complete therapy of cisplatin, it causes hearing loss in almost 90% of the patients. And even a single dose of chemotherapy, cisplatin chemotherapy can cause hearing loss in 1/3 of the patients. So various antioxidants, etc., have been used, but nanoparticle, combined glutathione therapy has been experimentally tried, in which, in this diagram, you can see, So with CCP, and this, the nanoparticle, this goes inside through the round window, and then it chelates, it combines with the cisplatin, and then it is with the magnetic effect, it is taken out and drained from the eustachian tube. So it is still experimental, but can be put to good use. The noise-induced hearing loss, again, very, very common cause of hearing loss amongst the young population, and it can also cause permanent inner ear damage. And there are various studies which have showed that delivery, a study by Gao et al. Found that delivery of solid lipid nanoparticle encapsulated edaravone inhibited free radical generation in cochlea and decreased hearing thresholds following noise-induced injury, although it did not demonstrate significant hearing recovery. Then it has also been, nanoparticles have also been used in retrocochlear hearing loss. Mayer et al. In his study demonstrated use of lipid nanoparticles to deliver rolipram to spiral ganglion cells. And they demonstrated increased neuronal survival and elongation of neurites outgrowth. Then implants and nanomedicine. Cochlear implants are very, very common form of rehabilitating young children with hearing loss. And it has been used in more than, I think, 5 lakhs of cochlear implant recipients are already there. But the bottleneck is that there is a gap between the electrode and the nerve, auditory neurons. So that is why these devices are limited through low frequency resolution and large stimulation currents are required. So a recently completed multinational interdisciplinary project called NanoCI, aimed at overcoming current limitations by creating a gapless interface between the auditory nerve fibers and cochlear implant electrode array, the NanoCI project. So here we can see in the first A and B images, the electrodes, how they go through the scala tympani in the cochlea and stimulate the nerve fibers, which are, which are far off. Like in the sense, you can see the green fibers are the nerve fibers. But in C and D images, when the electrodes are impregnated with the nanopolymer conjugated neurotrophin, that is the nerve growth factors, then the green thing, you can see the green line, which is the nerve fibers which has grown near the electrode. So the interface, the gapless interface happens, which leads to better resolution of the sound. So a gapless interface led to lower stimulation thresholds and larger dynamic range in vivo, and to reduce stimulation energy requirement. Challenges, as Agarwal madam also told, that there are challenges of engineering particles of optimum size, delivery to the site, efficacy, safety profile, how they are cleared from the body, how to image and monitor them, and making it accessible to all. So to conclude, modern nanotechnology offers a promising tool for efficient and non-invasive inner ear drug and biomaterial delivery. They can encapsulate various therapeutic agents like drugs, proteins, or genes, overcome anatomical obstacles, deliver them to target cells and even organelles, and improve the biodistribution in tissues. So, but there's a long way to go for real clinical applications. So thanks so much. Thank you.
Thanks, Dr. Rupa, for a nice and crispy presentation.
Thank you, sir.
And I think you have highlighted the importance of nanotech. If you look at the history of like evolution of ENT, starting from the hearing aids to the cochlear implant, and now you are talking about tympanic membrane injections and other biologicals. So it was a very nice presentation. Thank you, Dr. Rupa. Any questions? If it is there, we can.
Rupa, I just want to ask,
Yes.
Miss, are there any beyond animal trials on nanotechnology being used in ENT, not just otology, but ENT, head and neck tumors and
So
head
for
and neck area?
cancers, it has been done, but there is a difference between nanoparticles being used.
Apart from liposomal amphotericin.
Yes, sir, liposomal yes.
Apart from that, which you used left and right in the COVID era, but beyond
Yes.
that, any other nanotechnology used
So in
beyond animal studies?
Then head and neck cancers in vivo studies has been done because that there no the problem of the normal cells the death is not there, but here it is not. Still in vivo studies are very few, but in head and neck cancers few in vivo studies have been done. Thank you.
Thank you. I'd like to request the chairperson to please present a memento to Dr. Rupa ma'am.
So the next speaker is Dr. Amritabh Ghosh, Associate Professor, Department of Endocrinology. And he will be talking on a role of nanotechnology in diabetes mellitus wound healing. So over to Dr. Amritabh for next presentation.
Thank you very much, sir. I thank the organizers for allowing me to speak on this topic on this occasion. So my topic is the role of nanotechnology in diabetic wound healing. So the agenda is one brief introduction, the process of wound healing, how the microenvironment of diabetic foot ulcers are associated with non-healing, and then how nanomedical strategies are being developed for diabetic foot ulcer treatment. So we all know diabetic foot ulcer is a significant health and economic burden. So it's a significant health and economic burden. And lifetime incidence in a person with diabetes is more than 15%, and some studies have reported up to 34%. Leading cause of, it is the most common leading cause of non-traumatic lower limb amputation. And even if it heals, the recurrence rate is very high, up to 30 to 40% at the end of 1 year. So how does diabetes predispose to diabetic foot ulcers? And multiple pathways are involved with various pathways ultimately leading to increased oxidative stress, pro-inflammatory state, altered protein function, accumulation of advanced glycated end products, endothelial damage, and which ultimately leads to the microvascular complications and angiopathy. So one of the microvascular complications is neuropathy, which can be motor as well as sensory. Motor neuropathy leads to impaired joint mobility, foot deformities. Additionally, there is loss of protective sensation due to sensory neuropathy, which leads to increased risk of foot shear, pressure, and injury, which is unappreciated. There is also associated angiopathy, microangiopathy of the foot, as well as peripheral arterial occlusive diseases, leading to poor tissue perfusion. There is dry skin, which in turn again predisposes to ulcer formation. So there can be neuropathic ulcer, neuroischemic ulcer, ischemic ulcer, and often the patients end up in amputation. So normally the process of wound healing goes through continuous and overlapping phases of homeostasis, in which initially there is vasoconstriction, subsequently there is platelet addition and aggregation, which in turn leads to production of cytokines, and subsequently there is coagulation, leading to formation of fibrins and then thrombus, which closes the wound and provides a framework for tissue growth. So subsequently, there is an inflammation phase. Initially, the neutrophils predominate, and again, they are responsible for phagocytosis and release of cytokines. Subsequently, the macrophages take over. They are responsible for cleaning the neutrophils. Here, one thing is important is that macrophages M1 initially is the inflammatory macrophages. Subsequently, there is polarization to M2 macrophages, which in turn leads to progression to the proliferation phase, where there is migration of fibroblasts, granulation tissue formation, angiogenesis, and reepithelialization. In remodeling, type 3 collagen is replaced by type 1 collagen, extracellular matrix reorganization, and matrix contraction, ultimately leading to normal tensile development of better tensile strength of the wound and restoration towards normal tissue. So how is it perturbed in diabetic foot ulcers? Well, in the hemostatic phase, usually it is quick and not much affected. The problem starts from the subsequent phases, inflammation. So diabetic foot ulcer is usually associated with a chronic and persistent inflammatory stage. There is massive immune cell infiltration and chronic ongoing immune response. The hyperglycemic environment induces particularly this pro-inflammatory response. And additionally, the immune cells have reduced bactericidal and phagocytic activity, which renders the wound susceptible to infection. So diabetic foot infection is very common, particularly in tropical countries, and is the leading cause of amputation. Subsequent proliferation phase is also affected. There is increased fibroblast senescence, and which are again unresponsive to the stimulating growth factors. There is little vascular sprouting. So angiogenesis is also affected. Inadequate nutrient supply. This leads to inadequate nutrient supply. There is also decreased expression of hypoxemia inducible factor, which again promotes angiogenesis. There is increased matrix metalloproteinase expression, and reepithelialization also is affected. And in the remodeling phase, there is extracellular matrix decomposition and remodeling stagnation. So what are the main factors which lead to this alteration in the microenvironment? Hyperglycemia is involved. There is increased inflammation, there is persistent infection, there is hypoxia and ischemia, high protease activity, and wide range of pH, which impair the normal wound healing process. So, nanomedical strategies for diabetic foot ulcer treatment try to address these various impairments in wound healing. So, it tries to address the hyperglycemia, particularly at the wound, the inflammatory state at the wound, the infection, the hypoxia, the high protease levels, and wide-ranging pH. So, when it comes to hyperglycemia, usually the drugs which are currently available address the systemic hyperglycemia, particularly anti-diabetic drugs, the insulin. However, some of them again have low bioavailability. Nanomedicine tries to mitigate this by improving the physicochemical properties of the drugs and controlling their release. Also, certain metal organic frameworks are available. Okay, so which can deliver the certain drugs like metformin or curcumin, which is an anti-inflammatory agent, at the wound. So here the nanoparticles will act as a carrier. Another method which has been tried is the glucose oxidase method. It is combined with copper oxide nanoparticle based hydrogel spray, and when delivered to the wound, it results in production of glucuronic acid and hydrogen peroxide, which lowers the pH of the wound. At a lower pH, the peroxidase activity of copper oxide is stimulated, which leads to generation of hydroxyl radicals, which again has antibacterial activity. Also, copper has effect in angiogenesis. So ultimately, this promotes the wound healing. When wound is healed, the pH will again rise, and it will promote the catalase activity of the copper, and which leads to production of oxygen, which again is beneficial at the wound level. Hyperinflammation is another issue. So various antioxidants are available, particularly alpha lipoic acid, angiogenic drugs like VEGF are available for application. However, most of these are non-specific, have low bioavailability, and negative effects of long-term use with anti-inflammatory drugs. So nanomedicine tries to address it by microenvironment based targeted drug release and development of immunomodulatory nanomedicines, such as reactive oxygen species scavenging and modulation of immune cells. So in order to address this inflammation at the level of the wound, nanosystem should meet at least one of the following conditions to achieve anti-inflammation. One is promoting the macrophages towards polarization to M2 phenotype, down-regulating or up-regulating, like down-regulating the inflammatory cytokines, up-regulating the anti-inflammatory cytokines, and down-regulating the reactive oxygen species. So, various methods have been tried for promoting macrophage polarization to M2. conjugate glucomannan modified silica nanoparticles help in clustering mannose receptors on the surface of the macrophage. So depending on the types of receptors on the side on the surface of the macrophage, the M1 or M2 phenotype will be promoted. So presence of mannose receptors will promote the M2 phenotype. And then M2 phenotype in turn will promote the fibroblast proliferation, activation, extracellular matrix formation, revascularization. It can also help by delivering nitric oxide. Again, it will help in conversion of M1 to M2 type. Delivering of cytokines like interleukin 33 can also help in this transition. Biomaterials containing stem cells. So adipose derived stem cells can secrete various chemicals, and it can ultimately differentiate into cells which have capability of repairing. Exosomes can be utilized to carry microRNAs, which again induces phenotypic transformation. ATP delivering nanoliposomes, which can promote again cell recruitment, and here again adenosine will promote the macrophage transformation. High molecular weight hyaluronic acid can also be delivered, which will again promote the M2 phenotype. Again, anti-inflammatory cytokines can be delivered. Neurotensin is one of them. And often nanoparticles have self-activity, which have anti-inflammatory action, like chitosan coated cerium oxide nanocubes or heparin-based hydrogels. One microRNA, 146A, it can be, it is usually downregulated in diabetic foot ulcer, leading to increased pro-inflammatory cytokine production. So exogenous delivery of this microRNA can lead to suppression of the cytokine production. The other methodology that can be obtained is downregulating the reactive oxygen species. Here again, cerium dioxide, niacin, metal organic acid complex nanoparticles, manganese dioxide nanoenzymes. Manganese dioxide nanoenzymes will stimulate the catalase. Again, anti-inflammatory drugs or antioxidants can also be delivered. Similarly, gene therapy can be delivered. And again, hydrogen gas delivery also has a role. So, curcumin, which is coated in a, which is included in a nanoparticle and delivered in a hydrogel preparation to the wound site, has been shown to have regulate the inflammation and has healing activity. Persistent infection in a wound is another issue which has been tried to be addressed. Usually the treatment we administer are antibiotics and removal of biofilm by sharp debridement. The issues which we face are drug-resistant microbes and biofilm formation, which reduces the penetration of an efficacy of the antibiotics. And when it comes to debridement, although it is one of the accepted methodology, particularly we use sharp debridement. Sometimes in ischemic wounds, it can be painful. So antibiotic-free antimicrobial nanoenzymes, nanosystems have been developed. Particularly the nanomaterials for anti-infection therapy include silver, gold, copper, zinc oxide, graphene oxide, and graphene oxide silver nanocomposites. Particularly, which has been quite largely investigated, is the silver nanoparticles, which can adhere to or penetrate the cell wall membrane and inhibit bacterial cell respiration and destroy the protein structures. It also stimulates the functioning of the immune system and may be associated with activation of inflammasome, resulting in production of large number of neutrophils and WBCs to promote the bacterial clearance. Additionally, the silver nanoparticles could reduce the levels of matrix metalloproteinase at the wound. Near infrared therapy, that is a generation of the heat at the level of the wound, can also help in bacterial clearance. Nanoshells containing gold silver shells with copper oxide at the central core has been shown to have antibacterial activity against resistant bacteria by producing heat locally. As discussed previously, the glucose oxidase catalase enzyme system helps to generate hydroxyl radicals, which is beneficial in killing the bacteria at the local levels. Hypoxia and ischemia. The various therapies which are currently available are hyperbaric oxygen therapy, pro-angiogenic drugs such as growth factors, and often these are not sufficient and revascularization procedures are required. However, often it is high cost and is unable to sustain oxygen levels, and bioactive ingredients are susceptible to degeneration and inactivation with some of the agents. So, by incorporating the vascular endothelial growth factor or platelet-derived growth factor or other indirectly incorporating agents, delivery of the agents which can induce these factors, nanotechnology has tried to address this. So, hydrogen sulfide has been shown to promote wound healing. So, nanoparticles which deliver hydrogen sulfide through sodium compound has been tried again to address hypoxia. High protease levels at the level of wound are another issues. So various clinical treatment which are available are traditional dressing, human-derived collagen, which have been tried. However, nanotechnology can also help in this. Particularly chitosan-based multifunctional nanofiber dressings with antibacterial and antioxidant properties are being developed, and which also helps to absorb the secretions at the wound level. wide ranging pH, particularly the pH is reduced at the level of the wound, and pH switchable iron oxide nanoenzymes exert catalase and peroxide activity in neutral wound tissue and acidic biofilms respectively. And again, it helps to normalize the widely ranging pH, which is often seen in the wounds. So even nanotechnology has been tried to for prevention of diabetic foot complications. So this is one of the socks that is available, and this was a trial done in diabetics with diabetic neuropathy, which were randomized to socks with nanocapsules and socks without nanocapsules containing a hydrating agent. So those who received the socks containing the nanocapsules with the hydrating agent has significant improvement in clinical score, the hygrometry, the transepidermal water loss, skin temperature, and skin hardness with no adverse events. So I'll just summarize what are the therapeutic strategies of nanomedicines, how it tries to address the complication of diabetic foot ulcer. It tries to downregulate blood sugar at the level of the wound particularly, promote homeostasis, anti-inflammatory action, anti-infection action, regulate angiogenesis, extracellular matrix remodeling, and promote reepithelialization and regulate hypoxia. So this is a timeline of progression in diabetic foot ulcer nanomedicine. So in 2008, the recombinant human epidermal growth factor was delivered through nanofiber loading. Role of silver nanoparticles have widely been explored since then. And subsequently, nanofiber scaffolds were developed, which provide the scaffolding for tissue to grow. Gene therapy has been MMP response release delivery, exosome derived microRNA delivery, and recently porous polymer I'd just like to conclude by saying thank you.
Thank you, Doctor. It was an excellent, very comprehensive presentation where you have discussed about role of nanomedicine in diabetic podiatry, right from the management of local hyperglycemia management, hypoxemia, ischemia management, tissue repair and infection control and management. I would just like to ask two queries from you. Managing dry gangrene versus a clear-cut neuropathic wounds, it's relatively easy. But when you deal with a mixed kind of gangrene where there is both wet and dry gangrene, along with a component of neuropathy, is there any role of nanotechnology in identifying the perfuse boundaries where you can actually salvage tissues if you act in a better way? And again, an application of NIRS in similar kind of wounds.
Regarding the first question, yes, often ulcers infections are neuroischemic, and various methodologies are available for detection of ischemia. Particularly nanotechnology based therapies are being explored for treatment of the ischemia, hypoxia, through delivery of the agents which promote angiogenesis to the wound. Sorry, I couldn't get the second question. Can
Role of NIRS in identifying these gray zones of vascularity. No, NIRS actually, it's used very widely in critical care and anesthesia where they look at the flows in the vascular beds. So I'm not aware of the use of NIRS in diabetic foot and diabetic podiatry.
you're talking about infrared detection devices like infrared cameras?
Yeah, so infrared and nanoparticles along with that. So
yeah, actually infrared devices have been quite often explored in diabetic foot wounds. They have been explored to predict development of diabetic foot wounds, like temperature, local rise in temperature can indicate developing wound. Again, it is particularly important in patients with Charcot-Foot disease, where it can predict the whether the wound in the Charcot-Foot acute stage is resolving. Particularly, actually, in fact, I was involved in one study with thermometry, where I had monitored neuropathic ulcers healing. And we had found that temperature tends to decrease, however, it doesn't normalize when the wound heals. So further follow-up, even after reepithelialization, the wound tends to be warm, and further offloading and follow-up is required in those cases. Definitely, I'm not sure of the technologies, but yes, we have a role.
thank you. Is there any question from the house? Okay, thank you very much, sir. Thank you, sir.
Thank you, sir. I'd like to request the chairperson to please present a memento to Dr. Amrita Ghosh, sir.
So now I will invite Dr. Sarmishta Chakravarti. She is Associate Professor in the Department of ENT and Head and Neck Surgery. And she will be talking on role of nanoparticles in chronic rhinosinusitis and respiratory tract disorders regarding the current insights and the emerging trends. So over to Dr. Sarmishta.
Respected chairpersons, respected senior faculty members, and dear students, a very good afternoon to you all. And coming to the fag end of the session, I would like to keep my presentation crisp and brief. And my main aim will be to highlight the key features where the applicability of nanotechnology based on the domains of rhinology and laryngology and basic respiratory tract diseases. The initial few slides will be based mainly on the basic of nanotechnology. I think from the morning, every presentation must have included these things. So by this time, you must have all known Dr. Richard Feynman, who was the pioneer in the field of nanotechnology. He had actually mentioned this thing in a gathering of eminent persons. He had once mentioned that why not tried to write the entire 24 volumes of Encyclopedia Britannica on the head of a pin. So that is what he tried to emphasize, the concept of the world of nanoscale or nanotechnology. So basically, nanotechnology is all about shrinking down to nanoscale, where we walk and talk in the level of atoms and molecules. But this concept of nanotechnology is actually leading to development of microchips, ultra-fast computers, and any technology that you can think of. So making new things on this incredibly small scale is called nanotechnology, and the term basically means particles of the size 1 to 100 nanometer. So when we say, it basically means 1 billionth of a meter. So I'll not go deep into these initial slides, because I've realized these have been covered. Just to say that American physicist Richard Feynman first kickstarted the modern interest in nanotechnology. In 1974, Japanese professor Norio Taniguchi he coined the term nanotechnology. Then Dr. Eric Drexler, he first came out with the groundbreaking work and wrote a book on engines of creation, the coming era of nanotechnology in the year 1980. So that is how it has now progressed. In the year 1991, first carbon nanotubes were discovered. And of late, scientists in the space research organizations are on the verge of making carbon nanotubes as the primary portal of sending astronauts to space. So that is how technology is advancing. Now coming down to our own field, our own zone. So my discussion will mainly deliberate on 3 aspects. One is nano-based drug delivery system, which will mainly deal with the rhinological aspect. Second is nanoparticles impregnated during surgery and how they will affect in the outcome of the surgery. And third will be the future possibilities of its role in vaccination. So first is nanomedicine and nanodelivery systems are definitely a relatively new and rapidly developing science. The main reason why they are upcoming is because they can act at specific targeted sites in a controlled manner. So any material of the size 1 to 100 nanometer will come under nanoparticles. What are the nanoparticles? There are a variety of ranges which we will discuss. And depending on their use, whether they are used as carrier particles, they are used as drug delivery agents, they are given names like nanorobots. We have nanosensors, which are used as markers for diagnostic purposes. We also, the very first generation of nanoparticle-based therapy includes lipid systems like liposomes, which we are all aware of. It is FDA approved and it is now widely clinically used. Now, researchers have found out that these nanoparticles have immense role in nasal and airway inflammatory pathologies. Basically, the anatomy of head and neck is such that the role of nanoparticles or nanocarriers is even more relevant. That is because the anatomy of head and neck is very, as you all must be knowing, it has a very intricate anatomy, and targeting or delivering a drug at that particular site is very difficult. Compared to a GI system, any drug reaching into the nasal airway, the ear or the nose, or even for that matter, the throat is very difficult. So nanomedicine is even of greater role. Nanomedicine includes novel treatments, delivery mechanisms, and in vivo imaging. And that is because nanoparticles allow for controlled release and targeted therapy. So nanoparticles can cross biological barriers, which are otherwise inaccessible to traditional therapies. Second, it improves drug delivery and tissue specificity. And third, as I already mentioned, can be utilized for diagnostic purposes. So coming down to the major applications in rhinology, to begin with, we'll start with the very basic, which is nothing but nasal packing and nasal implants. We all know patients who come with epistaxis, bleeding from the nose, they require nasal packing. Immediate emergency treatment. Conventionally, we have tape gauzes with which we pack. As of now, we have atraumatic Merosil packs with which we are doing. They are relatively atraumatic and they harbor less infection, so they can be kept for a longer duration of time. Now, nanoparticles impregnated nasal packs they are completely absorbable. So completely absorbable means there is no need to remove. The patient is packed and then he can go home. So that is a very novel idea. And these are all into research, because that once any device we implant, we keep, and then if it is absorbable, there is practical benefits, but the issue remains of infection. That's why these are still into research. Absorbable stents, biodegradable polymers with extracellular matrices. So these are stents. Stents, we all are aware of stents which are in the cardiological aspect, we are aware. But as of now, upcoming research, there is also intranasal stents. These stents you can directly place into the sinuses during surgery, and these stents are impregnated with either steroid applications or antibiotics. So what happens? They are lodged and then kept in situ, and at a very low dose, they will generate the drug, so that it will cause a dual effect. The stent provides a macroscopic structural effort, and also it gives slow release of antibiotics or medications, so it has a therapeutic role. When compared with non-absorbable packings, absorbable packings have demonstrated reductions in post-operative synechia. That is, when the two raw surfaces stick together and they cause nasal blockage. So that post-operative synechia bleeding is less in case of absorbable nasal stents. So these stents are basically made of nanocrystals from calcium and sodium alginates. This is how they look microscopically. And once, if you, okay. So here, we don't have a pointer here, but I'd just like to highlight this thing. This is how the stent looks like. And intraoperatively, with the help of an applicator, the stent can be easily placed inside the nasal cavity. And the part we want to keep it dilated, the part exactly where we want the antibiotic to work, we can keep the stent. The stent will keep the opening or the ostium patent and give a very low dose targeted release of the medication. Third thing is a bioabsorbable polyglycolic acid stent that elutes mometazone furoate, which is a steroid. So this is a common condition which we all are aware of. A patient with chronic sinusitis, which is this is a normal sinus, and this is of course a diseased sinus with inflamed mucosa and also collection. These kind of patients present with chronic sinusitis or nasal polyposis. And nasal polyposis is a medical disease. Surgery is just a part of it. The patients require prolonged therapy, and this therapy often includes steroid therapy. So if we, instead of giving him or keeping him on long-term nasal steroids, the latest technology is just implanting a device in the sinus, which will very gradually release the medication. This is how the implant looks like. The picture in between, in the center, this picture shows a drug-eluting stent. That is, this drug, with the help of applicator, the stent is gradually inserted. The stent then actually opens up. Once it is inside the paranasal sinus, it's inside the nasal cavity, it opens up. And then this is the third picture. This is how it looks when it gradually releases the medication. The drug may last for 30 days, for 45 days, depending on the patient's requirement and the stent specifications. This is another type of drug-eluting stent that is known as Propel. So in this, what happens? After the sinus surgery is done, we have done the surgery. Surgery is required. There is no way out. But after surgery, these patients, because it's a medical illness, they will come back with polyps. So what we do is, we basically remove the polyp, open the ostium of the sinuses, and we keep it patent with the help of Propel as a type of, again, a stent, which will open up. And once it opens up, it delivers the medication. And after a time span of 30 to 45 days, the sinus will heal, the ostium will remain patent, and it will gradually dissolve. This is again another example how the ostium, the new ostium formed actually tends to close down. But if we keep an in situ stent, then the patency remains wide for a longer period of time. Then what are other applications in rhinology? This is supramagnetic iron oxide nanoparticles. So basically certain magnetic nanoparticles are also there, and they are magnetic nanoparticles are very good carriers of antibiotics. Amoxicillin clavulanate for rapid cellular uptake of antibiotic in the nasal mucosa. This kind of research actually tries to improve the therapeutic options we have for chronic rhinosinusitis. And coming to another aspect that we commonly come across in laryngology, is patients who have stenosis, laryngotracheal stenosis. Either it could be due to trauma or post-intubation. These patients, they have immense suffering because of prolonged intubation. They develop stenosis either in the subglottis, that is the part below the vocal cords, or at the level of trachea. So then they present with respiratory distress, stridor, and may require a tracheostomy. So these young patients often depend on tracheostomy lifelong if they are not treated. So the conventional therapy for these kind of patients is surgery, but based on nano carriers and nanoparticles coated stents, we have endotracheal tubes which are coated in PLGA, that is polyglycolic acid nanofibers, and loaded with mometazone furoate. So what happens? They cause reduction in laryngeal mucosal thickness post-intubation. So if we intubate the patient with these kind of stents, then the inflammatory reaction, the granulation tissue formation is much less. Thereby, the patient's chances of developing subglottic stenosis is substantially reduced. Now, these kind of trials are still in the research. The nasal stent I was talking about is already in FDA approved, but laryngeal stent is still in the trial phase. What we are talking right now is the future possibility. So these patients tend to have lesser chances of developing subglottic stenosis. So what happens? There is a pathological narrowing of the trachea, if you can see in the picture. So this mainly occurs because of altered immune response to the foreign body, which is the endotracheal tube, and thereby bacterial colonization of foreign body response occurs. So if we have an expandable metallic stent that helps to keep it patent, and we have medications loaded in it, then again it reduces the granulation proliferation. A revolutionized systemic side effect reduction method is nothing but a drug-eluting stent. And this is how the stents are prepared. This is the technicality of it, where we have 3-step polymer coating. And once this stent has been prepared with the polymer, then this polymer is being used to keep the stent in situ so that the patient has quick recovery and there is no need of lifelong or prolonged dependency on the tracheostomy tube. Another, of late, it has been found that rapamycin has shown to have immunosuppressive effects, also anti-fibroblast effect. So, but this rapamycin cannot be given systemically for a long period of time, but nanoparticles coated with rapamycin have shown promising effects in animal models. Nanoparticles have another aspect. They may help in the treatment of anosmia. Anosmia can be due to various causes, but of late, anosmia became a very important clinical aspect because post-COVID some patients developed significant anosmia and majority of the patients had some level of anosmia. Anosmia, we all know, is very difficult to treat. That is because the olfactory nerve, any topical medication is not able to cross the mucociliary clearance barrier of the nose and treat the disorder occurred in the olfactory nerve level. Now, nanoparticle technology has shown a role in the treatment of neurodegenerative disorder. Now, one barrier in the delivery of any type of nanoparticle therapy is mucociliary clearance. So, chitosan nanoparticles have been studied to overcome this barrier due to their positive charge and ability to bind to negatively charged mucus. So, this is the various nanoparticles which have emerged almost chronologically. The organic nanoparticles, chitosan-based nanoparticles, a very landmark study was done in 2012. Then inorganic nanoparticles, which are mainly antibiotic coated particles. Then we have nanofibers, which are used as drug carriers for mometazone furoate, which is a locally acting glucocorticoid. Then nanocrystals and nanoclusters. So this is nothing but a brief classification of the type of nanoparticles that we commonly use in the rhinology and laryngology aspects. They can be organic, inorganic, they can be carbon-based or composite. So these are the various organic nanoparticles which are used: liposomes, polymersomes, and then we have inorganic like gold nanoparticles, which have been covered by all specialities. There are certain interesting facts of nanoparticles or nanotechnology we all know. That is, when we come to the world of nano carriers or nanomolecules, then they are where the forces interacting between the atoms and the molecules is different from objects which we see in the macro world. This is a very important conclusion that we come. That is, when we deal with the world of atoms and molecules, what happens? Electromagnetic forces between the atoms and molecules is the major driving force, which decides how the molecules move, how they interact with each other. And this, if we create particles which are of the same size of atoms or molecules, then we can interact with those forces. And that is why they are able to bring about very significant effect in the field of clinical medicine. So these are nothing but carbon nanotubes. Nanodrugs have shown to have higher oral bioavailability because they exhibit typical uptake mechanism of absorptive endocytosis. So here, this is just almost a revision. Now coming to one very important aspect, which is COVID-19 infection. Now, of course, the concept of nano carrier mediated therapy given to the patients has not been done, but the scientific basis on which they work, this could lead to future possibilities wherein, because the size of nanoparticles is same as the size of viruses. So their interplay and the way they can inhibit these viruses can be significant. This is about coronavirus. We all know rapid mutation, tissue tropism, cross-species transmission, and adaptation. These are the main characteristics of these group of viruses. When we say the role of nanoparticles, then what happens? Currently, the diagnostic kits which were used for COVID-19 were mainly based on RT-PCR tests. Limited access to required equipment, necessity of skilled personnel, various restrictions were there. Nanomaterials can create effective interactions between the analyte and the sensor, owing to their large surface to volume ratio, because once an object is broken down to very small particles, the surface area increases. So the role of nanoparticles in COVID-19 has been brought down to 4 Ds. They have a role as disinfectant, as a preventive role. Why? Because coating of nanomaterial surfaces to inactivate the virus and prepare effective environmental disinfectants. Second is diagnostic role. Rapid and specific diagnostic tool for COVID-19. Then drug delivery, the use of nanomaterials to deliver antiviral agents, and development of mRNA vaccines. So since viruses are nanoparticles, so virus properties can be used to design virus-like structures. So this is intranasal delivery of polymer encapsulated antigens. So this can cause a strong immune response. So intranasal COVID-19 vaccine can be a possibility in future based on scientific research and implications how these nanomodulated carriers can have a role. So these are the basic strategies. Capability of nanomaterials to bypass the conventional restriction associated with antiviral agents. Nano biosensors for rapid detection. Antiviral surface coatings by introduction of nanomaterials and as disinfectant agents. Now coming to the last aspect of my presentation, that is a very common and especially more common in Raipur zone, which has a very dry climate and dusty climate, allergic rhinitis. So we all know that allergic rhinitis is again a disease, it's a hypersensitivity disease. So here the common presentations being nasal irritation, epistaxis, and when the common treatment given for any allergic disorder is anti-allergic medication and topical. If at a higher stage, then patient requires topical instillation of steroid sprays. What happens? These have bitter taste. They have their own side effects, and there is, in addition, there is decrease in drug residence in the nostrils due to muco, because there is a constant washout of the mucociliary by the mucociliary mechanism. So the drug stays for a less amount of time in the nasal mucosa. So then magnetic nanoparticles have a promising role in this kind of allergic rhinitis cases. Antibiotic impregnated magnetic nanoparticles, steroid as a steroid, which is again added to a nanocarrier, will act longer. The exposure of tissues to a magnetic field for 20 minutes. Iron containing nanoparticles had substantial residual stay in the nasal mucosa. So this is just in brief to show that this is where a normal nanoparticle, and this is a magnetic coated nanoparticle wherein the impregnation and the lasting effect is more of the medication. But this is in an experimental stage. So this is the final aspect. Clarithromycin is a representative drug of the macrolide group. It has both antibacterial and immunomodulatory effect. So it's commonly used in immunological conditions. So nano biomaterials, which are loaded with drugs and placed in the surgical cavity, can play a role in slow release of the drugs, promote drainage and physical isolation, also inhibit adhesions. So, but yes, every what goes up also has certain downfall. So here, one thing is because transition from micro to nanoparticles occurs, so the surface area increases. Because the surface area is increasing and the size is reducing, it can penetrate into any cell barrier. So that is the reason they can gain access into various cell organelles like nucleus, mitochondria, and trigger damage. They can initiate blood coagulation pathways. Silica sometimes, which is commonly used, can cause toxicity. It can cause oxidative stress, and inhaled particulate matter can directly reside in the lungs. So there are certain limitations. Again, potential route of entry via olfactory nerve into the brain. So these are the adverse effects which has to be kept in mind. These are my common references. So to conclude, it is a very promising tool. It requires extensive research, and converting it into from animal studies to translational research and bringing it into clinical medicine is a matter of time. But yes, the future possibilities are immense. So the next big thing is actually very small. Thank you.
Thank you, Dr. Sarmistan. Rightly said your concluding remarks. I also agree with that. The biggest things of these worlds are all simple. Right? So a very exciting and very comprehensive, excellent presentation. If any questions from the audience, we can take one or two questions. So basically what I learned in last 3 lectures, that most of these advances in the nanotechnology is restricted to, you can say, critical care or maybe most advanced things. So it is my urge that the same technology may be used for the purpose of diseases which are more prevalent in the community. Say, for example, if you talk about the pediatric point of view, I will say the malnutrition, pneumonia, because these are the biggest killer in the under 5 mortality. So I think we should take some initiative to develop some mechanism to prevent these 2 deadly diseases. Thank you.
Okay. Thank you.
Comments?
Thank you, ma'am. I'd like to request the chairperson to please present a memento to Dr. Sharmistha, ma'am. Thank you. I'd like to request Professor Alok Agrawal sir to please present a memento to our chairpersons. We really like Thank you. I'd like to request all the faculty members present to come on the stage for valedictory function.
So all the faculties can come on the stage and all the students can come besides. So we can have a group photograph. फोन करना पड़ेगा। आप कनेक्ट कर लीजिए। जूम का व्यू लगाओ ना। डॉक्टर नवीन ठक्कर को पिन अप करो। सही से बैठो। नहीं आ जाओ पीछे आ जाओ सब लोग। So dear friends, it has been a wonderful day where throughout the day we were doing deliberations on nanotechnology, a newer concept for the medical fraternity and people. I am highly thankful to our director, Professor Dr. Nitin M. Nagarkar sir, who had a dream of having 3 CMEs one by one on stem cells, on artificial intelligence, and now today on nanotechnology. And I know that all the people who are on the Zoom meeting, all the people who have attended this meeting, either on your YouTube or on IOTV or on Zoom link or being physically present, have learned tremendously on this occasion. I'm thankful to the National Academy of Medical Sciences, AIIMS Raipur, New Delhi also, because they were joint organizers in all these 3 functions. And today also they were here. To thank them, I will request our organizing co-secretary, Dr. Amit Chauhan sir, to thank with reading the biodata of President, National Academy of Medical Sciences, Professor Dr. S.K. Sarin.
Good evening, everyone. I will tell a brief biodata of our President, National Academy of Medical Sciences, Professor Dr. Shiv Kumar Sarin. Sir was bestowed with Padma Bhushan by the Government of India. Sir was given Henry Bockus Medal in 2017 by World Gastroenterology Organization. Sir received Professor Telfa Reynolds Memorial Oration at the University of Southern California. He delivered a lecture for Ralph Cohn Memorial at the Institute of Hepatology, London. And the World Academy of Medical Sciences conferred International Prize Award to him, and ESL National Recognition Award, Dhanvantari Medical Award, Ranbaxy Medical Sciences Award, and the Best Teacher Award. Sir was the founder editor of the prestigious Hepatology International Journal, recipient of Santi Swarup Bhatnagar Award, President of Indian Society of Gastroenterology, Indian Association for the Study of Liver, Asia Pacific Association of Liver, and Chairman Steering Committee of the Asia Pacific Association of Liver Association. Sir was involved in new treatment protocols for liver diseases, especially variceal bleeding, liver regeneration, hepatitis B, and acute and chronic liver failure. Sir has more than 630 international publications to his credit, has edited 13 books on liver, and has more than 87 chapters in various medical textbooks. Thank you.
Thank you, sir. Now we are having with us Professor Dr. Naveen Thakkar, who is the Indian Orthopedic Association National Secretary. Dear sir, we'll need your blessings.
Yes. Dr. Alok Agrawal ji and all the faculty and delegates for such a fantastic program on the nanotechnology, something new to be learned by orthopedic surgeons and many variety of faculty. And you are contributing a lot. I tried to join in the morning, but I could not join. And just now I could join it. And I think that it was a fantastic and successful program. And this will continue to make more and more knowledge and information to the Indian Orthopedic Association members. And on behalf of Indian Orthopedic Association, I'm thankful to organizers and Dr. Alok ji and all the faculty and all the delegates for a wonderful contribution. And we can evolve further and further by just sharing, contributing and learning together. It's a learning together process. So very good learning, very good academics. Thank you very much. Thank you, sir.
Thanks a lot, sir. Thanks. Thank you. And here's some representative from National Academy of Medical Sciences present here on YouTube or on Zoom. The learned faculty who is sitting on Zoom, if anyone wants to speak something about today's training, he is welcome to speak. Dear friends, we have with us Dr. Keshwan, the physical faculty from Bilaspur. So I'll request sir to speak something.
Thank you, sir. Very nice program arranged by Dean sir and Amit Sawan sir. I enjoyed the whole day. Actually, I reached first here at 8:30. I reached here. I attended all the session from morning to evening. I enjoyed a very nice, interesting, interacting session. Thank you very much for organizing team.
This CME on nanotechnology is as much learning experience for me, and also it stimulates the idea of research. So I believe that such CME should be continued to be planned in future also. Thank you, sir.
And who is the monitor from your batch?
Today I learned many things about nanotechnology. I saw the applications of nanotechnology in otology, pharmacology, and various fields of medical. And I think I have learned many things. And it was a great session. Thank you, sir.
I would like to thank our Dean sir and all the professors and delegates present over here. We had a wonderful session and a very learningful session. We hadn't thought like nanotechnology can be useful in our medical professions in the future as much, and we'll also try to make this field go to the further positive side. And thank you, everyone.
So dear friends, thank you. I'm happy that you people have learned something. And we all have to fill the post test and feedback so that we can get our certificates. They will be sent online. And if anyone wants any communication, he can do with our team if there's any problem in getting the online certificates. I'm thankful to the National Medical Commission, Chhattisgarh, which has given to the faculty 5 marks and to the delegates 2 hours of credit hours. So that's very important for all of us. Thank you. So we end with this. 3 cheers to AIIMS Raipur. Hip hip.
Hooray.
Hip hip hooray.
Hooray.
Hip hip hooray. Great.
Thank you, sir. Now we
bye.
Officially close the CME. Thank you.
Yeah.
Please close the Zoom link. Please wait, there is a high tea. Have high